Check, 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 check.
Check.
Hello.
Check. 1, 2, 3. So, very good morning, ladies and gentlemen. I hope everybody is doing good in the year 2024. Welcoming all of you to AgriVision 2024. Requesting all of you sitting at the back, please come in the front rows. All of you are requested to please join us in the front rows. Also, with that note, let us start this wonderful day, the first day of our AgriVision 2024. The theme of AgriVision 2024 is economic development through sustainable agriculture practice. Together, we can make it possible. We will be learning and discussing about agricultural practices designed to protect the environment, eliminate and reduce greenhouse effect, enhance soil quality, and fertile to protect crops and pressure earth nature resources for future generation. So these are the things that we are going to learn over the 3 days, where we have divided this entire 3 days into plenary session, technical session, poster presentation, farmers interaction, program exhibition, expo awards alongside the cultural night. To let us know more about this, let us have a person who is the backbone of this entire wonderful event. Let's welcome our principal scientist, ICAR NRRI, and our President of Society for Agricultural Research and Management, Dr. Arup Kumar Mukherjee, for his presidential remarks. Let's give it up for Sir. Give him a big round of applause, please.
Good morning, everybody. The journey started in 2018 when a young engineer, he approached our then director and at present the DG, Dr. Himanshu Pathak, for deputing a few scientists for a seminar which was organized in Puri in a hotel. Then our director, sir, he deputed myself, Dr. Pradhan, and most probably Dr. Pratap Bhattacharya, as I can remember. And after that, we thought there itself that let us continue this. So then we had organized another 5 seminars, international seminars, in 2 times in Institute of Life Sciences, once in Centurion University, then once in Robinson University also. And this time it is in a beautiful ambient places, which is within beautiful ambience of NRRI. And this time we are going to discuss about the environment, agriculture, sustainability, and human health, how they are interrelated. And then in 2021, we decided that let us make a society. Then Society of Agriculture Research and Management was formed. And 2022, it was registered under the Society Registration Act. And the entire credit goes to a single person who is none other than engineer Dr. Dibangshu sir is there. He is sitting here. So he is the person single-handedly managed everything. And I don't want to make my speech longer because I know that all of you are eagerly waiting for the main session. So I welcome you all in this gala event. And I am sure when the NRRI has taken the responsibility to make it successful, and this event will be highly successful, I welcome you all the delegates from different parts of India and from abroad also. I welcome for a pleasant stay at Cuttack, and also let them have a nice visit of Odisha. It's a beautiful state, and welcome you again, and thank you very much.
Thank you so much, sir, for your wonderful and kind words. And let's give it up for Engineer Divyanshuji for organizing such a wonderful event, which we have just started to begin. Let's give it up for him, everybody. Okay, so let's start with the first session, which is on the topic climate resilience agriculture. The chairman for the same is Professor Vaishnav Charan Tripathi, distinguished Professor Sharda University, Noida, and rapporteur is Dr. M.D. Shahid, senior scientist, ICR NRRI, Cuttack. So let's, Divyanshuji, are you going to announce this or shall I do that? The first session? Okay. The PSV Session-1, Advancing Sustainable Agriculture Through Process-Based Crop Modeling in the Era of Climate Change, which would be discussed by Dr. Vangimala Reddy, Research Leader, USDA ARS, ACSL, Beltsville, MA, USA. This session would be on video conferencing, so let's have that.
Hello, everyone. I just wanted to make sure if you are seeing the screen correctly. Okay. Can you hear me well? Am I audible?
Yes, ma'am, you are audible.
Okay. Okay, that's great. Okay, yeah, that's great to know. Okay, so first of all, I would like to thank Agri Vision 2024 for inviting us to deliver a talk. And I want to send a special thanks to Prasad Das, who helped us to change this presentation from in-person mode to virtual mode. Vangimala Reddy is a research leader at Adaptive Cropping System Laboratory at USDA ARS Beltsville. He was supposed to come there in person and deliver this lecture, but due to some reason, he couldn't travel. So on behalf of him, I'm going to do this presentation. My name is Sahila Begum. I'm a visiting agricultural engineer at USDA ARS, and I'm also a research assistant professor at the University of Nebraska-Lincoln. For today's talk, the topic is advancing sustainable agriculture through process-based crop modeling in the era of climate change. Let me start with the climate change term itself. Climate change refers to the evident long-term change in the climate patterns. We know if we look at the global temperature variation over the years, there is an increase in 1.1 degrees Celsius since the pre-industrial level. This is mainly due to the increased amount of greenhouse gases that have resulted in the global warming. And if you look at another factor, another greenhouse gas called as carbon dioxide, if you look at this graph here, which shows the increase in CO2 from 1960 to 2020, there is an increase from 320 ppm to currently it's around 420 ppm. This is also due to the increase in fossil fuel usage, deforestation, and land use changes. If you look at the methane concentration, that have rise up to 1923 ppm in 2022. This is mainly due to increase in production and transport of coal and natural gas, and also due to the livestock, from the livestock. With the increase in global temperature, there is also increase in ocean warming. If you look at the increase in ocean warming since 1880, there is an increase of 0.69 degrees Celsius. This is combined effect of both greenhouse gases, which have resulted in the global warming. So as the temperature in ocean increase, there is also an increase in sea level rise. This is data from 1993 to present. There is overall increase of 100 millimeters since 1993. So this is due to mainly two reasons. One is due to a process called thermal expansion, and there is, in addition to that, there is also melting of ice. When we talk about melting of ice, there is a mass variation in Antarctica since just estimated since 2002. We can see that there is a rate of change of 146 billion metric ton per year since 2002. This melting of ice sheets is a cumulative effect of both greenhouse gas increase, which have resulted in increase in temperature followed by the melting of ice. So should we be really worried? That's a question. And the answer is yes, we should be worried because it affects the life on the earth. So some of the key sectors that are affected by the climate change are agriculture, ecosystem, water resources, energy supply and use, transportation, human health, and society. So in 2022, the Intergovernmental Panel on Climate Change said this: The scientific evidence is unequivocal. That means there is no doubt there is climate change happening because there are enough and more evidence now. And climate change is a threat to human well-being and the health of the planet. Any further delay in concerted global action will miss the brief, rapidly closing window to secure a livable future. It just says that there is only a very limited time to take an action, and this action should be taken at a concerted global level. That means a collaborative level, collaborative effort at the global level is very necessary to tackle the adverse impact of climate change on the earth, various sectors on the earth. Now let's see what are the challenges posed by climate change on agriculture. As I mentioned before, due to the increase in greenhouse gases that have resulted in the overall increase in the temperature and climatic variation, there are extreme weather conditions that we are observing. Extreme weather condition includes extreme precipitation, extreme droughts, heat stress, etc. This have greatly influenced the agriculture in terms of production. The growth and development of crop itself is being affected. And there is also alteration in the growing season being observed in different parts of the world. And research are also showing that there is an increase in pests and diseases. With the increase in CO2 concentration, their growth and reproduction is also enhanced. And there is shift in agricultural zones. The soil degradation is happening, soil quality is reducing, and this is ultimately affecting the supply chain of the agricultural production. There is something that is complicating this challenge into a more complex stage. That is the increase in population. Currently in 2024, right now, we all know that the population is currently 8 billion, crossed 8 billion. And by 2050, the world population is set to surpass 9.1 billion. That is 34% than what the population is right now. And the food production for this increased population has to be increased by 70% to meet the need. So we already have the issue of climate change challenges. In addition to that, we also have to tackle this requirement of more food production in the future. So this both together makes the system complex to address. Here, I'm going to show some of the examples how climate change could affect the agriculture and is affecting the agriculture. Some of the examples are on different parts of the world and some are specific to US. In the first figure, if you can see, this shows the length of growing season in US. Length of growing season is the time between the last frost event in the spring to the first frost event in the fall. So that is called as a length of growing season. So if you look at from 1890 to 2020, the average length of growing season in USA have increased by more than 2 weeks. There is an increase in length of growing season by 2 weeks. So it can have both pros and cons to the agriculture. The advantage that is that it can allow farmers to diversify the crop, and it can also allow farmers to have multiple harvests from the same plot. But at the same time, the research have shown that with the increase in length of growing season, there is also an increase in the invasive species and weed growth, which can affect the agricultural production. With the increasing the length of growing season, it can also alter the ecosystem, range of and types of animal species and soil microbes, etc. And the right figure, this is to show the number of drought events from 1970 to 2020 and the projected increase from 2021 to 2030. As you can see here, there is a clear evidence that number of drought events are increasing. It increased from 7 in 1970 to currently around 22 currently around 19 in 2021. Just looking at the projection from 2000 to 2030, you can see that there is a drought have risen up to 30% globally since the year 2000. So drought, it can affect the plant growth and development, and also it can put more pressure on the irrigation requirements for the crop, for the agriculture. I would like to take your interest into this figure here. This figure represents the productivity of maize in US. So the y-axis on the left, it shows productivity in kilogram per hectare, whereas on the right side, it shows the total US production in million metric ton. If you look at this, you can see there is an increase over the years. This is mainly due to the technological improvement in terms of advanced machineries and technology in terms of using sensors for irrigation and new cultivars, etc. So obviously there is an increase in productivity, but at the same time, you can see in some years there are reduction in the yield, though the overall increase is in the positive direction due to extreme climatic conditions. Some years are drought, some years have wet spring, early frost, and unusual climatic events can reduce the yield. So which gives a clear hint that climate change, even if we have technological improvement in improving the overall productivity, the climate change can clearly lower the yield production, the productivity. As I mentioned to you before, there is extreme events are also increasing. So when extreme precipitation happens, and sometimes they can be flooding in the agricultural farm. So flooding can also affect the crop growth and development. I want to take your attention to this figure here. This shows how the plant growth and development under different duration of waterlogging. This is for the cotton. So the cotton was subjected to waterlogging at the 2-leaf stage, starting from 0 days of waterlogging, 2, 4, 6, 8, 10, 12, and 14 days of waterlogging. And then all the parameters were measured and analyzed in terms of growth and development, just highlighting just 2 of it, which shows that photosynthesis and the whole plant dry weight in continuously waterlogged condition were 75 and 78% less compared to 0 and 2 day waterlogged plants, which shows how much effect the waterlogging can bring into this crop growth and development. This is another experiment that shows the impact of weed growth under current and potential futures carbon dioxide concentration in the atmosphere. These are the control chambers in which one is set to 380 ppm of CO2 and the other is set to 680 ppm of carbon dioxide. And both plots were equally treated with herbicide. But when the CO2 increase, there is an evident increase in the amount of weed that is grown in the chamber, which shows that this may put more pressure on the use of herbicide in the future when the atmospheric carbon dioxide is increasing. The temperature, the right figure shows how the temperature affects the plant growth and development. This is the day and night temperature in cotton. So up to a temperature of 35 by 27 day and night temperature, the cotton plant is going to grow well. But once it crosses the optimum temperature to a higher temperature, plant growth is affected in a negative way. So up to a certain temperature, the effect can be linearly high, but if it crosses a threshold, then the plant growth and development can be affected. This effect is different for different crops. So in summary of climate change factors and general impacts on crop growth, in summary, the climate variable carbon dioxide is projected to increase. And this increase, the general impact on crop growth is that there can be an increased net photosynthesis and transpiration use efficiency. It can reduce the transpiration, increase canopy temperature, and reduce crop nutrient concentration. Temperature, which is also projected to increase both maximum and average temperatures, minimum and average temperature. A general impact is that it can help in reducing the frost risk, but at the same time, there can be increased stomatal conductance, photosynthesis, respiration and transpiration, faster growth and development, phenological shifts, reduced transpiration efficiency, and an increase in temperature could result in increased heat stress. The projected rainfall is observed to be varying across the globe in the future. So it have both positive and negative impacts on the crop growth. Solar radiation, the projected trends are reduction in solar radiation and increased diffused light fraction. The increase in diffused light fraction could result in increases photosynthesis and growth. So one way to understand how climate variable interactively affect plant growth and development on different processes of plant growth and development can be by using process-based crop simulation model, which can be a key tool in assessing and addressing the impact of future climate change. So process-based crop modeling involves simulating the growth and development of crop based on the underlying physical, biological, and environmental processes. So in process-based crop models, all the processes are mathematically represented at a mechanistic process-based level. So these models, the existing process-based crop models, have a mechanistic way of simulating the photosynthesis, respiration. It can have phenology, soil water dynamics, how much nutrient uptake and utilization. So climate impact in terms of when the temperature change, carbon dioxide change, vapor pressure change, relative humidity, all this impact can be represented back to how it affects the photosynthesis and then carbon partitioning, etc. And pest and disease simulation, crop management practices like irrigation and fertilizer, and stress factors, water, nitrogen, and heat stress can be mechanistically brought into the model. And canopy architecture, root growth and function, and crop genetics can also be incorporated in a process-based way. So this process-based crop modeling could be a potential solution. It can be a solution for understanding how this complex climate variation can affect the crop growth, and also help us in developing solutions to improve the yield, at the same time protecting the environment. So I'm going to show some of the application of process-based models towards climate change analysis. So this is a simulation that is carried out for the US, for the Eastern Seaboard region, which includes 13 states from Maine in the north to Virginia in the south. So in this simulation, the future climatic simulation scenarios have considered the shift in the carbon dioxide, temperature, and rainfall from the baseline of 1970 to 2000. So here, the bottom figure on the left shows the change in temperature and the precipitation for the simulation up to the mid-century. Here you can see that there is a shift in maximum daily air temperature. The average temperature have increased plus 2.9 degrees Celsius. And whereas in terms of precipitation, there is an increase in 10%. So how this increase in temperature and precipitation along with the variation in the COT affects the yield. This is simulated using process-based crop model called as SPUDSIM and MAZIM. So based on this two model simulation, it is observed that for the potato, the yield can decrease up to 42% in this zone, whereas for the maize, the yield can reduce up to 20% in this zone. So this give a spatial variation on how the yield variation could happen because of the climate change. Process-based models can also be used for analyzing the spatial variation as well as over the entire in a global level. For example, this is a study by Jonas in 2021, which have tried to understand the variation at a global scale, in which the simulation have shown that maize shows a decrease in the yield from -6% to -24%, mainly due to the warmer climate projections in the regions where maize is currently cultivated. Whereas for the wheat, which is more expanded in the high latitude, it have a positive projection in terms of yield. There is a yield gain from 9% to 18%, and this is linked to the higher carbon dioxide concentration, and also it is mainly located in a high latitude area where a positive increase in the temperature is helping it to grow well. So this kind of simulation can help us to know how the geographical location play a critical role in affecting the crop growth and development and also the yield. So for this also the process-based crop models can be used. More details can be read from this reference here. Okay, now coming to another application of process-based crop model is to know whether crop switching can help in the future, reducing the reduction in the yield because of the climate change in the future. So this is a study by Rising et al. In 2020. What they have tried to understand is if we are following the same cropping pattern currently followed to the future, then they estimated using the process-based crop model that there could be up to a drop of 31% in the yield for the 6 major crops that is grown in USA. But if systematically, if we are allocating or switching this crop to other locations which will positively help these crops to grow in a better way, then we can reduce the loss up to half. So the last figure, which is shown in 2070, when reallocating the crops to different locations, it can reduce the impact of yield reduction. So these kind of analysis can also be carried out using the process-based crop models. Last application, but not the least important one, the insects and pests. As I mentioned you before, insects and pests growth and reproduction is highly dependent on the environmental condition, like how much carbon dioxide and temperature and moisture, etc. So this is a study that have looked into crop, pest, and climate interactions to understand how much yield loss could be there in different regions in the world for due to the increase in the insect and pest damage on the crops. So in this study on wheat, rice, and maize, yield loss to insects was found to increase by 10 to 25% per degree Celsius of warming, and this is more influence or more hitting hard in the temperate zones compared to tropical and other zones. So now that we saw many application of process-based crop models, if we use it after understanding the fundamentals and the mathematical functions in the crop models and using it effectively can really help in understanding how the crop agriculture is impacted with the climate change, and also it can also show some light to what could be the possible alternative measures or possible management conditions or management practices or cultivar new genetic improvements that can be built to tackle the effect of climate change on agriculture. Along with the positive side, there are also some challenges and limitations for the process-based models. Process-based models can be really complex because they have mostly more complex interactions taken into account, like genetic, environment, and management interactions. When simulating this at a spatial larger scale, it can larger scale as well as a larger temporal scale, it can have model much complexity. And also there are always farm management and technological adaptation, new farm management practices and all. If it is coming, it's difficult to bring into the process-based models. And also some of the government policies and interventions can have an impact on agricultural practices, which is hard to predict or to incorporate into the process-based models. And process-based models, to have a holistic approach of looking to understand the effect of climate change, we may have to integrate it with other models or economic models or hydrological models. So that is also a challenge. And other limitations are technological and methodological constraints. Sometimes high computational demand is required to run complex models, especially at a larger scale. And the other constraint is lack of standardization. So process-based model for each crop will be different because crop growth and development is different under different temperature conditions, etc. So we cannot standardize the crop, but soil simulations can be standardized. And another limitation of process-based models are data availability and quality. Lack of detailed data for models. Process-based models, we need enough data about soil, crop, atmosphere, conditions over spatially and temporally. And also sometime for the model validation, historical data could also be one of the limitations. So what could be the future direction in process-based models? One could be the integration of advanced technologies like artificial intelligence and machine learning, and also bring in the results from the remote sensing into the models to improve the model prediction, and also integrate it with the global GIS system. And also there could be a component for big data analysis. And in future direction could also be for improving the model accuracy and resolution for better, and also better calibration and validation. And we could also think about tailoring models to specific regions on the earth, and also look for a global collaboration in terms of running the models and also sharing the data among different parts of the world. And also finally, if we can bring into a system where not just analyzing, but also helping in making the decision to improve the productivity, and at the same time not affecting the environment. For example, if we can increase the nitrogen nitrate fertilizer, and the production can go high, but leaching could be a problem if more nitrogen is applied. So a proper decision based on the existing scenarios, existing conditions and future conditions can also be incorporated into the process-based models. Just sharing you this quote: if agriculture goes wrong, nothing else will have a chance to go right, by MS Swaminathan. I'll stop here and I would like to take any questions, if any. Thank you.
Thank you. I don't have to stand up anymore. Thank you, Megam, madam, for giving a presentation here. Are this now open to questionings? Any questions, please? Yes.
Madam, excellent presentation. We really thoroughly enjoyed. But I have just one query. It's not question. That in most of your presentation, you have shown that due to climate change, the productivity of crops are increasing in most of the cases. Then should we worry, particularly being agriculture scientist, for climate change? Or we should worry because climate change will affect directly to human health. But as long as agriculture is concerned, how much we should be worried for that?
Okay, most of in the study, particularly, we have found that the wheat is having a positive impact, but most of the other crops will have a positive impact up to a certain temperature. But beyond that, if the temperature goes, it can have a detrimental effect on the crop growth. So there is impact, but there is always a limit up to which it can have a positive impact, but there is always a tipping point where it can have a negative impact on the crop. So, for example, in our lab, we are trying to grow plants at different temperature and carbon dioxide concentration. We are developing the process-based models by accounting for this kind of interaction between CO2 temperature and plant growth stages. So not all crops are having positive impact. It depends. It is complex, but it depends on the crop and as well as the climatic conditions. So I would say we should be worrying more than being happy that, okay, it will be just positive. So that's what I think. And also want to add that my research leader, Reddy, is also online, just to mention everyone that he's also here.
Yes. Thank you.
Yeah, good evening, Dr. Begum. Nice presentation. So my name is Varaprasad from Kansas State University. So my question is that does these simulation models take into account the impact of extreme events, particularly high temperature events, when they happen for a short period of time? Welcome, Dr. Reddy. Thank you.
Yeah, thank you. That's a very nice question. Yes. So what we are trying to see is, for example, for the cotton model, after pollination, the pollination can actually, for example, the temperature can improve the crop growth up to a certain stage, and also the fiber quality, everything can improve up to a particular temperature. But the problem is when there is extreme temperature beyond that, the flowering happens, but the ball will not retain on the cotton. So this kind of experiments we have to conduct in growth chambers, and if we can find that temperature, in what extreme temperature the ball start dropping, these kind of things we can incorporate into the process-based models. In that way, the extreme temperature stress or extreme drought stress impact on production and the plant growth can be incorporated. So some of the models have it, but some of the models, if not having, we can add it by first doing the control experiments and then making a relationship and adding that to the process-based models. So my answer is, for some crop models, it's already there, but some are not there, but it can be added.
Huh? Yeah. Those who are sitting back, please come forward. Okay? Please come. Those who are sitting in the back, please come forward. So people don't have shout. You can see the screen is better. Yeah. Please come and come. More advance. Yeah. Students also can come forward a little more. Yes. Send me sir. Yeah, please. Okay. Dr. Begum, any other question? Dr. Begum, I have one question if you don't mind. Hello? Are you there?
Yes.
Yeah. Okay, you have seen this model in probably mostly due to in US model, right? Maybe other models, but why a C4 plant maize, which have been a 1 degree temperature increase, we are predicting a loss by 15%. Okay, number one. And number two, similarly, wheat, which we have seen in experience in India, of course, you might be case in it might be case in US, other places in higher latitudes, but in India, we have 2-3 years ago we had a increase in temperature during spring, and that affected the wheat productivity so much that India has switched its rationing pattern from wheat to rice. Okay, saying that even 1 or 2 degree increase in temperature can impact wheat production to a large extent. Okay, but our experience probably should say that C4 plant like maize should have more productivity, even at 1 degree centigrade, because they are more tolerant to higher temperatures. Kindly say your opinion on this. Thank you.
Okay, great question. So the studies that I've looked into have shown that the maize shows a decrease from 6 to 24% is due to more warmer projections in the locations where maize is grown in the world. Whereas in terms of wheat, the increase is corresponding to the higher CO2 concentration rather than the increase in temperature. So higher CO2 concentration is the main reason where wheat is shown to have a yield gain compared to maize, where it is impacted by the warmer climate projections. So that could be one reason why wheat have an advantage, but maize have a reduction in the yield prediction in the future. And also the location also matters. So wheat is cultivated more in the global level. It is more cultivated into the high latitude region. So that is what I'm thinking could be the reason.
Well, that didn't answer a question. Anyway, is Dr. Reddy here? If he can answer this question better for us? Okay, he's not here, I guess. Anyway, and if you don't have any other question.
So we have one last question from the lady at the back.
Yeah, please come forward.
Good morning, everyone. I'm Arlene Alegre from the Philippines. So I'm particularly interested in diseases as a plant pathologist. So I would like to know if there are diseases that cause extreme devastation as a cause of climate change. Thank you.
Diseases, you mean the pest? Or insect pest?
Diseases.
So I don't know details about that, but overall what I know is the studies have shown that with the increase in CO2, the growth and reproduction of pests and insects are higher when compared to lower CO2 concentration. So that could be the one relationship that I could say. I don't know the complex way or what would be that disease that is more affecting the crop. So I'm not sure about it.
Okay, if there are no other question, we thank Dr. Begum and Dr. Reddy, for excellent presentation and work. I'm sure if they are here face to face would have been much better for us. But thank you very much for a great lecture.
Yeah, thank you again.
Thank you.
Yeah.
Well, now it is my pleasure to invite Dr. Prasad, who is in Kansas State University, and he has a lot of work on agriculture modeling, crop productivity. Break? Then break.
Check. So we have a slight change actually. We'll have his talk first and then we'll take the break.
Is it okay? All right. Okay. So, all right. So let us, Dr. Prasad, it is a great honor for us to be here, and and we can interact with him face to face. It's a great thing. And Dr. Dhankar is here. We will be enlightened to hear from him.
This laptop is up. Well, they're setting up the laptop. It's no, they're not able to project it there. I'm saying the slide should be projected there, not me. I don't know who changes it. Yeah, online can you just the screen has to be shown there. Okay. ये वाला screen वहाँ पे देखना. Okay, while we are waiting, I might respond to the scientist from the Philippines who asked for a question raised about the impact of climate change on the diseases. So just to let you know that one of the most impacted diseases from the climate change has been rust. For example, coffee rust is especially in the Central America region. It was only used to come at a lower altitude, but now it is coming at a higher altitude. So it has really impacted the whole coffee production in the Central America region. And also another one will be the blast. Blast would be also very much impacted by climate change, especially the increasing temperatures and the drought stress. And then the last one I would like to say is about the aflatoxin, because aflatoxin is caused by the Aspergillus infection, which happens with higher temperatures and drought stress, especially the groundnut and the peanuts and corn and sorghum are impacted by that. So those three are the classic examples globally which has impacted the climate change, particularly the high temperatures and the drought stress has impacted. But of course, you also know that under climate change, the intensity of the rainfall is going to be high, the humidity will be high. So that with the higher humidity, you'll also see a lot of other and spot, like leaf spot diseases. So that's one question. And then I think there was another question which the chairman asked related to the productivity of the maize and the wheat, and how maize being a C4 plant, why the productivity is decreased on those. But irrespective of that, most of the maize grown in the US is temperate maize, not a tropical maize. So the temperate maize is a little bit more sensitive to compare to that of a tropical maize. So it looks like my presentation is up. So good morning, everyone, and welcome to the Agri Vision. First, I would like to thank the organizers, Dr. Das, for giving me this fantastic opportunity today to speak to all of you today. And also wanted to thank the president of the SARM, Dr. Mukherjee, for inviting me. So let me get my presentation. Today, I'll be talking about the sustainable agricultural intensification to address sustainable development goals. What is the current status of the investments, of the research, and also what's the current status of what is needed in the future. So let me get my timer started. I have 20 minutes set up, so I'll make sure that I'll finish it on time. So first, before I go into my talk, I'll talk about the current status of SDGs, Sustainable Development Goals, food security, nutrition security, and then introduce you to the concept of sustainable agricultural intensification. What does it mean and what are the various components of that? And then talk about the current status of research and the current investments and the future needs. And then we'll give you global examples of how we can address some of the sustainable development goals, particularly the one related to hunger and nutrition. So first, the status of the sustainable development goals. So these are those of you, especially for the students who may not be aware of this. So Sustainable Development Goals is a continuation of a Millennium Development Goals. So after the Millennium Development Goals were completed, then in 2015, the Sustainable Development Goals were initiated, and they have to be achieved by 2030. So we are already halfway through. So there are 17 Sustainable Development Goals, which are listed here in this in this slide. Just focusing the first two, one is no poverty and then zero hunger. Those are the primarily ones which are looking at. And this year, last year, 2023, in July, there was a whole report which came out to tell us what is the status. We started them in 2015, halfway through. Where are we in terms of these 17 Sustainable Development Goals? Unfortunately, as you can see from the figure on the right here, so only 15% of the Sustainable Development Goals, we are on track. Whereas in the rest of the 48%, we are moderately and severely off track. And then 37% are stagnated or regression. So if we go at this pace, we will not be able to meet any of the Sustainable Development Goals. It's unfortunate to say that unless we really make a rapid change in the way we look at it and how we invest it and how we move forward in that direction. So midway through, only 15% has been reached. And then, okay, of those 17 Sustainable Development Goals, that was the overall combination of all 17 goals. Now, where are we at each of those development goals? This is a visual which gives you all the development goals on the left-hand side, and the green ones are on track. The yellow ones are fair progress. The red ones are stagnated, and then the gray ones are insufficient data. So I'm just telling you those which are mainly ag-related activities will have on those, especially the G1, no poverty, G2, zero hunger, Goal 3, good health and well-being, Goal 5, gender equity, Goal 8, decent work and economic growth, Goal 10, reduced inequalities, and Goal 13, climate actions. All of those are the ones which are less than 15%, not even at the 15%. So that's something to be worried about. And that is under the premise right now, today, 2023, this data, 820 million people do not have access to food. That is unacceptable. And majority of those are, 75% of those are women, and 80% are living in areas which is impacted by climate change. And that does not count the impact of COVID-19 and the current economic crisis and also the current conflicts going around the world. The moderate conservative number is that will add another 200 or 150 to 200 million people to the existing number. That means 1 billion people do not have access to food. Not only that, we are also threatened by malnutrition. Having food is one thing, but nutritional component of that is very important. And that's what I call it as a triple threat. So we have people, about 148 million children under the age of 5 who are stunted, which are too short for their age. 45 million are wasted, which are too thin for their age. And not only that, we also have 37 million people who are too heavy, obese, too heavy for their age. So that's a triple threat. So combine that, it's too much of an issue. And if you look at the micronutrient deficiency, the zinc and the iron, 2 billion people are malnourished in terms of micronutrient deficiencies. That is also unacceptable. So that's that with that premise. So now the concept of sustainable agricultural intensification. So the whole concept here, the goal is to provide access to safe, nutritious, healthy food at all times to all people from existing farmland without damaging our natural resources and ecosystem health. So that's some concept of sustainable agricultural intensification, because we have issues with food and nutritional security, but we don't have more land to bring into agriculture without devastating our forests. So we have to produce more from less or from existing farmland. The three important components to that: genetic intensification, which is on the red, which is focused on higher yields, resilience to pests and diseases, tolerant to abiotic stresses, higher nutrition, and both traditional and innovative new gene editing tools are on the table. The second one is agroecological intensification, that is focused on diversification, cropping systems, farming systems, improved agronomy, integrated nutrient, soil, water, and pest management practices. It's more about agronomy. How do we manage the ecosystem and the environment well? And the third one is socioeconomic intensification, which is focused on developing new markets, creating new opportunities, building social capital, and also understanding the barriers of adoption and institutional capacity building, because it's not lack of the technologies which is an issue, it's an adoption of those technologies at a scale where it can have an impact on food nutrition security. And all other innovative tools like digital, geospatial, artificial intelligence, nanotechnology, mechanization, precision agriculture, microbiome, income generation, entrepreneurship are components of all three aspects. So in brevity, what this does is this gives you the interaction between genotype by environment, by management, by social sciences, G by E by M by S interaction. Oftentimes, we only do 2 of the 4, sometimes maybe 3, but we always miss the most important component, which is the social science piece of it. And gender, youth, and communication and data science are an integral part of that. So my lab at Kansas State University is focused on addressing this. This is one of the Feed the Future Innovation Lab, which is one of the largest innovation lab. We started in 2014 and we'll be finishing up in this year. We have active research, education, and outreach programs around the world across multiple countries and continents. Currently, we are working in 13 different countries: Cambodia, Bangladesh in Asia, Ethiopia, Tanzania, Malawi in East Africa, Senegal, Burkina, Niger, Mali in West Africa, and then Honduras, Guatemala, and Haiti in the Central America and Latin America regions. So I won't go into the details of this, but we have a very large portfolio. In each of those countries, we have a research program focused on the need-based, what they want us to be working on. And we also have several consortium. One of the consortium is focused on mechanization. The second consortium is focused on digital tools and geospatial tools. The third one is a policy consortium. Fourth one is a soils consortium. So those are the 3 consortiums. And we also have centers of excellence on sustainable ag intensification and nutrition in Cambodia, in West Africa, in Coraf with Senegal. And then we have one in CMACH for Haiti, which is Center for Mitigation of Adoption, Resilience, and Climate Change. And we are in the process of getting one started in Guatemala as well. So anyone who wants to learn more about our lab, please visit our website and you'll have more information there. So we have about 130 scientists, 120 organizations with 20 US universities, 9 CGI organizations, and 250 graduate students. So that's our portfolio. So it's all the work is being done by those fantastic scholars. And I get the privilege of standing in front of you and sharing their results. So now I will give you the current status of sustainable ag intensification. So in 2018, Dr. Jules Priddy, who is father of sustainable intensification, because he was the one who coined the term sustainable intensification in 1995. He is our external advisory board member. So he did a survey to find out where are we in terms of sustainable ag intensification around the world. So they looked at these 7 types, which is the integrated pest management, conservation, agriculture, irrigated crops, pasture and forage redesign, trees, irrigation, water management, and intensive small patches, which is commercial vegetables or horticulture crops. So he estimated what was the status of those. So it's estimated that 163 million farms, which is 29% of the world, are practicing some form of sustainable ag intensification on 453 million hectares of land, which is 9% of the worldwide. So we are at a tipping point where it can make a significant impact on our food systems. agri-food systems. So that was 5 years ago. So those numbers I truly believe are significantly higher now. So that was the biophysical innovation, right? Those 7 biophysical innovations. So he also led another paper which talked about why it happened. What was the primary reason? We have the innovations, we have the technologies, but what was the reasons? So what he observed was that for the same 7 types, what was observed that there was an exponential increase in the number of social groups from year 2000 to 2020. Year 2000, there were only 500,000 of these social groups. But in 2020, that number increased to 8.5 million. So the whole of this social revolution behind, which allowed the adoption of these technologies, was very key. That's why when I said that in the G by E by M by S, S is very critical and important. So that's about the technologies. What's the status of those? Now, what's the status of funding? Because as I said, sustainable development goals. So we need to know how much we are investing in agriculture, how much we are investing in sustainable intensification, and what is the future requirement to meet some of the sustainable development goals. So there was an International Commission on Sustainable Ag Intensification formed in 2020 and finished in 2022, all during the COVID period. We never physically met. Entire commission ran online. So that's one of the adjustments we made because of the COVID-19 issue. I was very privileged to be part of this commission, and I chaired a couple of programs there. So there's a lot of reports, policy reports, and the detailed reports which are published, and all of them are available online for you to look at. If you're interested to just look at what the summary is, then the 2 pages will be 1 or 2 pages will be very valuable. But if you really want to look into the methodology and the entire report, all of those are available on those websites. And also, all of these have been published in a Frontiers in Sustainable Food Systems as a peer-reviewed journal article. So I'm just going to highlight 3 items so that we get to know what the status of this is at this point of time. So the first question was how much we are investing in agriculture research as a globe. And this is focused on Global South, which is developing low and middle income countries or developing economies. And I chaired this particular study, but it was conducted by Dalberg Asia. So we invest about, this all in Global South, okay? So $60 billion is invested in the innovation, only innovation. I'm only talking about innovation in research, $60 billion. You may think it's a lot, but it's very less. It's only 4.5% of the contribution of agriculture GDP. So we're only investing 4.5% of the agriculture which provides on innovation. So that's okay. The next question was, how much of that has to do anything to do with environmental goals, so the climate or the sustainable intensification, the environment piece of it? Only 7% of that $60 billion has anything to do with environment. And fascinating or shocking thing was half of that, only 3.5% has anything to do with social goals, which includes human dimension and the society. So with that type of investments, how can we meet sustainable development goals? So that's what the current status was at. That was the last 10 years from 2010 to 2020. Okay. So the next question was, how much is needed, right? So that's what is currently we're doing. So to at least meet some of the sustainable development goals and climate goals, so how much is needed? $15 billion a year from now to 2030. If you are able to invest that much money, we will be able to minimize the hunger, reduce the hunger to current levels of 10 to 15% to 5% in the rest of the world. And sub-Saharan Africa, we can bring it down to 11%. And we can minimize the carbon emissions of 242 million metric carbon equivalents. We can minimize the water use by 10%, and we can reduce the deforestation by 925,000 hectares. A mere $15 billion per year from now to 2030, we can actually make some progress in the sustainable development goals. And if there is a way, there is definitely a will. So that's the status. So now I want to give you examples of various things. So I'm at 12-minute mark. So in the next 12 minutes, I'll give you 11 or 12 examples of some of the things, innovations which we can do to address some of these sustainable development goals, especially when it relates to food security, nutrition security, climate security, and gender equity. I'll give you examples from all over the world. I'll start in Asia, or maybe I'll start in West Africa, but I'll provide you one example from different parts of the countries which we are working on. The first one is the crop livestock interaction. We'll start with West Africa, Burkina, Senegal, and Niger. We know for smallholder farmers, both crops and livestock are very critical. Most of the time, farmers will have components of both. They may have big cattle, or they may have pigs or poultry or aquaculture, but there is always some animal piece along with the agriculture or grain production systems. So we are looking at dual purpose crops, meaning that grain for human consumption, biomass for livestock, but there's always a need for both of them. So these are we have dual purpose sorghum, dual purpose pearl millet, and dual purpose cowpea, which we are promoting in the smallholder farmers. So the grain also has high zinc and high iron, so that they can have the nutritional piece of that. And those seeds are available. So that's one innovation which will address the food and nutrition security and also takes care of the livestock. And then now we'll move to the East Africa and Tanzania and Malawi. We're calling doubled up legume system. We all know about cereal and legumes, right? And then either we do rotation or we do intercropping, right? So here is an example of doubled up legume system where you have a maize in the middle, and then you have a pigeon pea, which is a taller legume and a perennial as well. And then that's an intercropping between cereal and pigeon pea. And pigeon pea is tall. Under the canopy of the pigeon pea, you can grow a shorter crop like groundnut or cowpea. So now you have 2 legumes and 1 cereal. So that's what we call a doubled up legume system. It's utilizing the below ground resources very effectively, the water and the nutrients, and also above ground in terms of the light interception. So that's a nice way of having a doubled up legume system, which has environmental benefits, also biological nitrogen fixation, and the perennial leaves drop, and then they provide the organic matter to the soil. So that's a doubled up legume systems. And then we know the issue with the rainfall. So now we're having high intensity rainfalls. The total rainfall in the future may increase, but it'll happen in a high intensity rainfalls, and there'll be a prolonged drought between the 2 rainfall events. So we have to capture this high intensity rainfall through different methodologies. One of them is through in situ water management, and you can also do through watersheds. But one of the examples of in situ water management is tide ridges, meaning that you have these ridges, and then you tie at a frequent intervals so that when the rain falls, they have more time for infiltration, and the water will actually go into the soil and not cause runoff and of nutrients and soil and other things. And then sometimes on the bunds, you can actually grow another species like pigeon pea or grass, which will also provide you with more biomass for your livestock feed. So that's in situ water harvesting. So that's just another example. And then most of the time, the watershed, another big approach, but watershed, you collect the water in the ponds, right? So how do you utilize those ponds? Water is through integration of vegetables in the home yard, but providing irrigation through that source using drip irrigation and conservation agriculture. So this, we are looking at conservation agriculture practices. The 3 principles are continuous soil cover, no-till, and rotations or diversification. So we're doing that. We are using the locally available mulch and putting it on the soil, and we are irrigating through drip irrigation system. So it is minimizing the water loss as well. So, and then of course, there are no tillage or minimum tillage only to remove the weeds, and we are seeing significant benefits. So I should acknowledge from each of these slides, the people who have done the work and the organizations with which we are collaborating are already shown in their logos. So I should have said that in my first slide, but I want to say it now and acknowledge all the people who are doing the work. So that's a conservation agriculture. And then we know when solar power is a big issue. So water lifting, women are the one who are lifting the water from the wells or nearby ponds, which is a very backbreaking event. So we are using solar power for water lifting and also water delivering through the drip irrigation systems. And we also have this sensor, small sensor placed there, which will tell you when to stop. Too much irrigation is also a major issue. So these simple innovations of solar power for water lifting, for delivering through drip irrigation systems and having a sensor when to stop. So those will improve the water productivity and minimize the waste and losses of water. So that's in Ethiopia, but can be, and also in Cambodia, but can be used globally as well. And in coming to Asia, in Bangladesh, we are working on high zinc rice, and this is a non-GMO traditional breeding done by Bangladesh Rice Research Institute, and this is a shorter duration variety. So we are able, if we are able to harvest quickly and also plant quickly using mechanization tools, you have a window of about maybe 60 days, and we are incorporating a new crop like mung bean or sesame or maize. So we are using high zinc short duration rice at the main, and then we are also mechanizing on the front end for quickly transplanting and planting, and also mechanizing at the end and that saves us time, about 15 days in the front and the back. And then you have 30 days between, so you can incorporate a new crop. And sometimes you're also integrating fish or aquaculture into the production systems. And then the perennials, in those home commercial vegetable gardens on the borders, we are having these perennial crops, especially Acacia pinnata, which provides high vitamin A. And then there are also wild gardens where different types of heights and different tree species, and which gives fruits at a different time of the year. So that way you can have some food security throughout the year. And then this is minimum till or no-till. I'm saying minimum till because for no-till, you require mechanization. You also required herbicide options. So those are limited in sub-Saharan Africa. So we are talking about minimum till. Sometimes we may not get the yield benefit, but the economic benefit of the labor saving is there. And then agroforestry is an integral part. This is a visual from Senegal. Our friend from Nigeria would easily recognize this director from Federal University there. So this is Fiderbia albida, and this is a cropping season. The tree is naked; it does not have any leaves. That means it's not competing with your main crop. And if you go to during the summer, the tree will be the only one which has a green, and then the rest of it is there. So it's not competing. That's called reverse phenology. And the trees have deeper rooting depth, so hydraulic lift. They're lifting the water level from the deeper level to the higher. So that's another. And there's also shrub systems as well. The Angera senegalensis and Pleostigma reticulata, these are trees and these are shrubs. So a nice way of integrating agroforestry systems. And the second last example is when we talk about greenhouse gases, rice, methane emissions, and nitrous oxide emissions are very critical. And application of displacement of urea in a brisket, not in a prill urea. So that will minimize the losses of nitrous oxide losses as well. And then putting it by hand at a place is very complicated. So there's a mechanized application of the displacement of fertilizer urea is there. And the last example I would like to give is on the pest management. So we typically use chemicals for pest management, but here's a technology developed from ICPA, which is called push and pull technology. So you have this maize as a main crop, and stem borer is your insect. You have intercrop of desmodium, which produces volatiles, which will repel the pest from the main crop. And on the borders, you have napier grass, which will attract the stem borer. So that way it's called push and pull. There's several examples which will follow this type of, and this is primarily because of the volatiles which these crops are producing. So that was my last example. So I'm at almost 20-minute mark. So in conclusion, sustainable intensification provides multiple options and a holistic solution to address food, nutrition, climate, water, and energy security. And engagement with the farmers, youth, value chain partners, private industry is critical and should occur from the start, not as an afterthought or not in the middle. And understanding the social networks and barriers are equally important rather than coming up with new innovations to remove the barriers of adoption. And focus should be on the creating enabling environment for scaling and innovation of technologies. And for this, interdisciplinary research and collaboration is critical and needed. Multiple disciplines going beyond biophysical sciences into social sciences, and better linkages of agriculture. We should promote agriculture as nutrition, agriculture as health, agriculture as food, agriculture as business, agriculture as technology. So that should be articulated very well. And then there is urgent need to increase the investments in innovation, building social capital, human resources, institutional capacity, and scaling to meet the sustainable development goals. With that, I would like to thank my team here who does all the great work. And then also would like to thank the organizers once again and SARM for giving me this fantastic opportunity. Sarve Jana Sukhino Bhavantu. Jai Hind. So I'm at 22-minute mark, so I don't know if there's time for questions or not.
Yeah.
I'll be glad to take any questions you might have.
Jai Hind.
Jai Hind, yeah.
Okay, Pradhan Corporate.
Please identify yourself, briefly your name, and then ask the question.
Hi, I'm Professor Om Prakash Dhankar from the University of Massachusetts Amherst, USA. And Dr. Varaprasad, thank you very much for such a broader aspects of the entire agriculture system, climate change and adaptation. My question to you is about the Bangladesh, the short-term, short duration rice and high zinc and iron. So Bangladesh is highly affected by the arsenic in rice, especially arsenic in rice.
Absolutely.
Because of the underground water contamination, and they're irrigating that. Has your team checked that arsenic in those high arsenic and zinc? Because arsenic and iron directly interact on the soil, on the root system, and high iron in the soil can prevent the arsenic uptake, or less iron can increase the arsenic uptake.
Thank you very much for that very important question, Dr. Om Prakash. We have not checked the arsenic concentrations in the grain or in the plant in these varieties which are being produced in the coastal communities. And you're absolutely right that our work is in the coastal zone and the polder communities where there is arsenic issues because of the ground of the soil itself is arsenic, and then also irrigation and the pollutants as well is an issue. So that we'll make sure that I'll inform that to our partners, and we'll measure the arsenic concentration in the grain and see if there is an issues. But there is also issues with acidity, and which has related to the iron as well, and low iron, and then also high iron, both ends rice. So that's a very important point.
And then on the extension on that one is when you said that with the short duration rice, there is a window of 60 days. So they are putting a short duration like a legume or something.
Yeah, legume or vegetables or...
And that's also another risk, food safety risk in that
right.
one is because more arsenic built up during the rice growth of the repeated irrigation, then the legumes will pick up that arsenic as well.
Absolutely, absolutely. Arsenic is a huge issue, heavy metals in entire country of Bangladesh. So that's a great question. Yeah. An important question as well.
Any other question?
Yes, there's a question at the back. Yeah, please, can you please pass the microphone to her? I think we have multiple microphones here on the table, so maybe distribute it.
Hello, my name is Dr. Christelle Ledroit from FIBL Syscom India. And I just have a question because I've seen that you have projects all over Africa, and in Asia you have in Bangladesh. How come you don't have any project in India?
Yes, excellent question. So this, as I said, my project is funded by USAID, US Agency for International Development, under the program called Feed the Future. So this is a whole of the government initiative, started with President Obama, then continued under Trump, and then continuing now. So at that time, they've identified 21 countries around the world based on food security, nutrition security, need, and the governance. So those are the 4 criteria. There are others, but those are the 4 important criteria to identify or pick a country. So India is the 5th largest economy in the world, and it is a rich nation. It's a distribution of the wealth is an issue. So at that point of time, through this Feed the Future program, India did not qualify, is not as poor enough to, and we don't want to be in that condition at all. But this program, Feed the Future, is not directly funding the innovation labs for this research, but USAID funds a lot of issues with health, especially when it comes to TB and also the AIDS. And then water and sanitation programs are well there. And there are also partner organizations which like the CGIAR organizations, which receive funding, and they do a lot of research. They're just the innovation labs, which are not working directly in India, but recently, last year, I think India is not the Feed the Future country, but an alliance country, because some of the research which we do here would be relevant to Nepal and Bangladesh, which are the Feed the Future countries. Good question. So that's my response. Yeah.
I have small question here. What about millets cultivation everywhere in the world? Because, time has come to think about alternative crop,
Sure.
and small crop.
Sure.
And millet can grow in countries like India, and many other African countries,
Yes.
So maybe it could be
Thank you very much for that question.
a future.
Yes, the millet is a very important millets, because millet is not a one crop. There's about 13 species which are qualified as millets, and the four biggest one, or the three biggest one, is sorghum or jawar, pearl millet or bajra, finger millet is the three ones, and then there's foxtail millet, there's teff, there is quinoa, and I can go on. There's about 13 or 14 different things. International Year of Millets was celebrated last year, 2023, under the leadership of India, supported by 9 other countries, and then it's a very good initiative. When it comes to climate resilience, millets are the most climate resilient crops because their water requirements is very less, anywhere from 400 millimeters and less. So that's wherever there is drought and opportunity, millets are a very good source of crop there. They're also very short duration. There's a lot of diversity. If we just look at sorghum, 45,000 accessions, just one crop. So there's a lot of genetic diversity in all of these millets. We need different durations, so we need to capture that. And then more importantly, like our previous speaker eloquently talked about the temperature increase, both the season-long temperature increase and the short duration. Right now, we are about 1.2 degrees Celsius warmer than what we were before. COP28, if you're following, our goal is to minimize the future by less than 1.5 degrees, or at least 2 degrees or less, but there's not good at this stage. At the current levels of investment, does not look like it's going to be happening. Maybe we'll be transitioning to fossil fuels, but then the most heat tolerant crop is millets. You asked about the temperature response, right? So if you look at the temperature response, or the ceiling temperatures of various cereals and legumes, so the first one is wheat, 32 degrees Celsius, rice is 35, corn or maize is 30, 33, 34, ceiling temperature at which there is no formation of grain or seed. And then pearl millet, sorghum is 38, pearl millet is 40 degrees Celsius. The most heat tolerant crop is among the cereals is pearl millet. And then so millets have a very good role moving forward, whether it's temperature stress or drought stress. And more importantly, they are nutri-cereals. They're very high in micronutrients. They're also very high in antioxidants. They have therapeutic values. Protein as well. Now you have high protein, not as high as wheat, but relative. And more importantly, it's also gluten-free. Those who are suffering from celiac disease, which are susceptible to wheat gluten, there's an alternative. So I truly believe that in future, millets will be the climate resilient crops, but we need to recognize one thing, especially with our friends in Africa and a lot of us in India. In India and Africa, millets are human food. Majority of the millets, sorghum or pearl millet, is consumed as a human food. Rest of the world are in developed nations like in Australia and especially in US, it's all livestock feed. Grain is fed to livestock. So I was shocked when I went to when I come from India and South India, from Andhra Pradesh and Telangana. When I took this position in Kansas, I never thought that you can feed the grain to the livestock. I thought it will be human food, but 85% of the grain sorghum is for livestock, 15% is for biofuels now, very few targeted human. But that is going to change because of the current projection of millets as a nutri-cereals. And if many of us, how many of you know quinoa? Raise your hand. Quinoa is a crop, right? So 10 years, 15 years ago, I never knew about anything about quinoa, but quinoa crashed through the market, become the health and innovative food. And I truly believe that millets will be the next quinoa. And if you look at quinoa, 2014 or 2013, don't quote me on this, 2013 or 2014 was celebrated as an International Year of Quinoa. Took 10 years and then it's a lot of investments. So I hope that next future will be the millets will be the nutri-cereals and also climate resilient cereals.
Thank you, Dr.
Long answer, sorry.
Thank you, Dr. Prasad, for excellent lecture.
Right.
We used to have more time to discuss, but thank you very much. We are now going for a tea break. We'll be back.
So just taking 5 minutes of everybody's. We want all of you on stage for a group picture first. Yes, all of us on stage for a group picture. He's already there. Yeah, that would be more convenient. Yeah, some of them, some of them can be there in the front. So, okay. Okay, however you are convenient. Yes. Can we please have the Agri Vision backdrop? Agri Vision backdrop. Agri Vision backdrop. Agri Vision main backdrop.
Photo जो देख लेंगे.
हाँ, तो main backdrop का दिया है ये set. हाँ, इतने कोड में इंडा. तो हमारा Agri Vision का backdrop था कोड. It Agri Vision. Logo इटा खाली. Backdrop था नहीं. Where's the backdrop? Thank you so much, everyone. Now we'll have a tea break for 15 minutes. We'll resume back the sessions up at time. We'll resume the session in the next 15 minutes. That would be around 11:35. Hello, check. We'll resume the session at 11:35 sharp. Thank you. Okay, it's a request for all of you to please go outside of this venue for the group picture. और जल्दी से वो क्लिक करके आप अंदर आ जाइए so that कि हम ये सेशन भी शुरू कर पाए. Group picture is already done here. Then okay. Okay, please settle down.
Dita algo kan mau, kalau tu sih, aku tempat aktif rokok. Kalau mana, kau ikut lepas lepas. On रखी थी कौन चश्मा ने कौन on कोई माइक ऑन ଅଛି माइक ऑन ଅଛି ଚେକ୍ କରୁଛନ୍ତି ମାଇକ୍ ଅନ୍ ଅଛି ନା ଏ ଅଡିଏନ୍ସ କୁ ଏକା ପାଖରେ ଦେଇ ଦେଉଛନ୍ତି କୋଉଟା ଦିଟା ମାଇକ୍ କାଢି ଏଠି ଇଣ୍ଡିକେଟର ଦେଖଉଛି ନା ମାନେ ସିଷ୍ଟମ ଅନ୍ ଅଛି ବୋଲି ସିଗ୍ନାଲ ଆଉଛି ଅନ୍ ଅଛି କଣ କରିବ ତମେ ମାଗିଲେ ମ୍ୟୁଟ୍ ଖୋଲିବ ତୁ ଅନ୍ କରୁଛୁ ନା ଅନ୍ ଥିଲା କଣ ଖୋଲିବ ऑडियंस ऑडियंस में डाउन ऑडियंस में डाउन ଗୋଟେ access ଦେଲାନି ପର୍ଯ୍ୟନ୍ତ ଭିଡିଓଟା କଲ ମିସ୍ କଲ୍ କଲ ବନ୍ଦ ହେଇଯିବ ପୁରା ନା ଆଗ ଖାଲି ଫ୍ୟାନ୍ ଗୁଡା ଅଫ୍ କରୁଥିଲ ଆମର ଯାହା ଅଛି ସବୁ ଠିକ ଅଛି ଆମର ତ ଭିଡିଓଟା ମାନେ ପ୍ରୋବ୍ଲେମ ଅଛି ନା ଆମ ଯୋଉ କ୍ୟାମେରାର କ୍ୟାପଚରଟା ସେଇଟା ଡିସପ୍ଲେ କରିପାରିବୁ ଆରେ ପୋକୋ କଣ କରିବା କହୁନୁ ବାୟୋଡାଇରେକ୍ସନାଲରେ ତ ଇଣ୍ଟରଫେସ କରି ଯାଉଛି କିନ୍ତୁ ଆଉଟ କେମିତି ହବ ଗୋଟେ କାମ କରିବୁ ଗୋଟେ SBI ଲୁପ୍ ଦେଲେ ବି ତ ଏତେ ପଶିବ ଏଠି ବାହାରି ଯିବ ମତେ ଆଉ ଗୋଟେ ସିଷ୍ଟମ ଦରକାର କୋଉଠି ଲଗେଇବୁ এটা 2 এটা 3 এটা 4 ତ 1 ମୁଁ ଏଠି ରଖୁଛି ଦେଖ ହେଲା ଏଠି 5 ରଖୁଛି ଦେଖ ଏଟା ରିପିଟ ହେଇଯାଉଛି କଣ ପାଇଁ ଏଟା ହଉଛି ଏଟା ରିପିଟ କଣ ପାଇଁ ହଉଛି
Check. Everyone, please join us inside the hall. We are about to begin the next plenary session. Check. Okay. So I would request Dr. Om Prakash Dhankar ji, Professor Crop Biotechnology, University of Massachusetts, Amherst, USA, to please join us on stage talking about the topic feeding and fueling in the future, metabolic engineering approaches for increasing oil yield and climate resilience in oilseed crops.
पॉइंट तो है नहीं, मैं वॉक करने का क्या फायदा है, तो वॉक को खड़ा तो मुझे यही होना।
आप रखिए, अगर आपको जरूरत पड़ी तो
ठीक है,
ये यहाँ से ऑन कर लीजिए।
ठीक है, ऑन है नहीं अभी,
क्योंकि ये वाला ऑन है।
ठीक है, इसको कहाँ से करना है,
मैं हेल्प कर,
ठीक है, बंद कर दो,
तो जब भी चाहिएगा मैं करूँगी।
हाँ बंद कर दो इसको, डिस्प्ले यहाँ से, yeah, okay, that's good. Let me get the good morning, and first of all, I would like to convey my sincere thanks to the organizer, especially to Dr. Dipen Das and Dr. Mukherjee to invite us here for this meeting. And it was in the beginning, and when Dr. Dipen asked me, please, on our behalf, please invite more other professors and faculty from US. And I tried almost like 6 or 7. 3 of us are here now in that group. So anyway, first of all, so now with this one, and this is my first visit to the Odisha. I was looking forward to the visit here, and I'm so far. Let's see. I'm going to enjoy the weather, and because the weather at my place in Amherst, Massachusetts, is -15. This morning was -15 degrees Celsius, and here I'm enjoying in upper 20s. That's good. Thank you. So with this, my topic is, I put it a little bit broader topic, feeding and fueling the future. But in this case, this talk here, I'm going to show mostly the metabolic engineering of oilseed crops, how to increase the oil content and seed yield in oilseed crops, and also how to increase the multiple abiotic stresses in those crops. So this is the University of Massachusetts, a very beautiful campus. And also we have perfect four seasons, and the fall time, autumn time in September, October is the best time to visit in Boston, Massachusetts area, because we have the best fall foliage. You can see these are not Photoshop images. These are the real images of the fall colors on the trees. So as you heard in the first 2 talks about the climate change adaptations and the impact of climate change. So and the first talk, Dr. Sohila and Dr. Varaprasad, they talk about all these issues about the population increase, as well as the impact of the population increase and climate change on the food security and food safety. So here, as you see this, and I'm going to summarize this in one slide. So currently, we already surpassed by 2022, we already surpassed 8 billion people, and by 2050, expected more than 9 billion people. And in order to feed those people, we need to produce more than 70% of the more food. But the 70% of the food need to come from the less arable land, less chemicals, less fertilizers, less water, and less labor. So there is a formidable challenge to feed the growing population with this. With the increasing population and limited resources. And not only that, also the food security and food safety, but the use of this, the new dimension toward using this biofuel phenomena, using the agricultural land to produce biomass and oilseed crop for the biofuel and biodiesel, that's also adding other extra kind of this burden, because you are shifting the agricultural land currently used for food production toward the biofuel production. So in my talk, I will be talking about these three things: how to increase the seed and oil yield in the oilseed crops by manipulating the lipid pathway, the tricyclic synthesis pathway. And I will give the example of camelina, which is also a mustard family member. And then I will show you what are the bottlenecks to identify the further increasing the oil contents. And then third one is to increase the climate resiliency of these crops so that you can enable those crops to grow on more marginal lands. So with this one here, because now we are shifting from the renewable, from the fossil fuel towards the renewable fuel energy, and the global demand for the biodiesel and biofuels like ethanol from the lignocellulosic biomass and biodiesel from the oilseed crops is increasing year by year. By 2022, it was basically significantly more than around 90 billion gallon per year is used for that one. And then when we started this one, we used camelina as a dedicated biofuel crop in US and many of the European countries. Camelina has been proposed as a dedicated biofuel crop because it's a non-food, cool season crops, can grow on marginal land. Also, the oil content is slightly higher. And in the US, 2012, US Navy, and several of those commercial airlines like Virgin Atlantic and KLM, they use this 50% blending of the camelina oil with the gasoline oil, and that's worked very well. So because of that one, like here, they use those in the fighter jets, and they start dubbing those fighter jets as a green hornets. The hornets, the fighter jets, they start calling those green hornets. So based on this, there is a significant interest in the public and private sector. And actually in US Navy want to run their entire fleet on the renewable energy in future. So we started with this one. And in the camelina, we had two aspects of those: how to increase the oil yield. So basically the first one is, of course, you need to increase the photosynthetic efficiency to capture more carbon. And then and in this one was the role, my co-PI, Professor Dennis Snell, he was involved in to capture more CO2, increase the photosynthetic efficiency. And my role was to flux that extra carbon from the photosynthesis to create a sink in the seed for the oil. And how we did that one, and this project was basically in my laboratory, was started with this postdoc Sudesh Sikara and master student Kenny from Ghana, and Parisa Akbari was technician from Iran, Hesham PhD student from Egypt, and Ben from US. So it's a very diverse and interdisciplinary team. So when you see the oil production in seeds, basically the oil production, oil synthesis and storage happens in the seeds. In the endoplasmic, in the fatty acid synthesis happens in the plastids. And then glycerol 3-phosphate synthesis happens in cytoplasm, and in the endoplasmic reticulum is the lipid synthesis happening in that. So through the glycolysis, the carbon is pulled through the glycolysis to make the fatty acids, and through the series of acylation, the fatty acids are acylated on the backbone of the glycerol 3-phosphate to the tricyclic. These are the fats, basically lipids.
So in this entire process, we found that these two genes are rate limiting steps, like glycerol 3-phosphate dehydrogenase and diacylglycerol acyltransferase are the rate limiting. We target these two genes initially. We overexpress these two genes in Camelina, and we put this under the DS-RAD fluorescent marker, so you can select those these transgenic lines without going through the hygromycin or any antibiotic selection. Just you throw UV light, and the transgenic seeds will glow red, and you can harvest. You can pick up those. So we overexpress those. And you can see these are the control seeds, and these are the transgenic lines, single transgenic lines and double transgenic lines. And here you can see the transgenic line seeds are almost 60 or 70% bigger in the size as compared to the wild type. And you can see here these double transgenic lines as compared to single transgenic lines. And this is the control wild type. The size was around more than 50% bigger. Also, the seed yield per plant was, as compared to control plants, was almost doubled as well per plant basis. And when we analyze those total oil contents, the transgenic lines expressing this two genes, DGAT and GPD1, they accumulated almost 16% higher oil content as compared to the control. Some of those individual lines also have higher, but the double transgenic lines on average were the best. And then seed yield per plant was also, as compared to the control, this is more than double. And then harvest index was more than double as well. So, and also we analyzed the fatty acid composition, and here you can see mainly the carbon C18-2, which is a linoleic acid, which is this omega kind of 3-6 kind of those. So this is basically was significantly increased, and this is a good for the human consumption for the edible oil, but also it has a good attributes for the jet fuel because oxidation state is more stable in that one. And so that we increase those, significant increase in the seed yield, significant increase in the oil yield by manipulating two genes. But we were looking for what are the bottlenecks? Why can't we produce 50% oil in those seeds as compared to 35 or 36%? So we look for those. So what we did was we used the omics approach. We used this double transgenic lines, and then we analyzed. We did the transcriptome, metabolome, and lipidome, and we mapped all the three data sets to identify the candidate genes. And through the candidate genes, and like here, to identify those, we use those different stages of seed development. For metabolic profiling, we use 3 different stages. And here is the RT-PCR stages, and then the RNA sequences. And we just, because this is we studied when the oil synthesis happens, what stage, what days, and then how the maturity happens. So we check here, and based on this one, we identify the different stages to analyze those metabolites and transcriptome. And just quickly showing, I'm not going in the details. In the transcriptome, you can see this in the individual lines at the 21-day stage or the 15-day stage. Day after flowering, there is thousands of genes were upregulated and thousands of genes were downregulated. So there is a big shift in the gene expression. And these are the Venn diagram shows some of the common at different stages compared to wild type. So there was a lot of changes. And when we did the metabolite analysis, we did a comprehensive metabolite analysis. We had around 246 metabolites analyzed. And this highlighted in the green are showing upregulated, highlighted in the red showing the downregulated, and in the black in the middle is unchanged. And compared to wild type with a single gene, compared to wild type with the other the DGAT gene, and compared to the wild type with the double transgenic gene. So there is a lot of metabolite shift as well. Overall, in nutshell, what we found is, this is a complex slide, but what we found is, because we were more interested in lipids, and here you can see this with the single gene here or double transgenic gene, there is a significant shift in the lipids, and some of those monosaccharides were higher, free fatty acids were polyunsaturated fatty acids were higher. So ultimately, what comes out is we found that the rate of fatty acid synthesis from plastid is limited. Accumulation of monosaccharic glycerol, breakdown of lipids is another limiting step. So push and basically the push fatty acids, what we need to do is increase the fatty acid, draw more carbon from photosynthesis, increase the fatty acid synthesis, and then push that fatty acid synthesis toward the tag accumulation, lipid synthesis, and then retention. So what Dr. Bara Prasad showed was push and pull, a kind of hypothesis. I'm showing the push and pull in a different direction. He was showing about the insect IPM, and here we are pushing the more fatty acid synthesis, pushing toward the tag accumulation, and then pulling the carbon to make more tag, and then retaining a push-pull retention hypothesis. We observe those. So those are the common genes that we found. These are very rate-limiting genes, and we are characterizing those genes further as well. But well, like I can say I'm Indian by heart. I live in US. I practice my education and my teaching in US, but I have my deep interest here. In India, India is importing 50% or more than 50% edible oil from abroad. And biofuel concept, using the oilseed crop for biofuel, it will not fly in India at all. It will backfire. So I was looking at the same pathway, same genes, everything same, how to increase the oil contents in the edible oil so India can become more self-reliant in edible oil. That's why the interest was. So we are translating this knowledge from the camelina into mustard, soybean, and sunflower. And why we are doing it? Because the edible oil consumption globally has tripled with all different oils, like palm oil, soybean oil, canola oil, sunflower, is tripled. There is a huge consumption because also there is a demand for more in the developing countries as well. And this is the scenario with India. This is the up to 2015 data. This is the red bars are basically the production in India. And this green line is consumption. So there is a huge gap between the production and consumption. So in order to meet that gap, we need to develop more the oilseed crop varieties that can produce more oil, and like mustards and groundnut and all those, and sunflower, soybean. So that's and we are doing it, transferring all those genes already. We have done in mustard. I'm not going to show much of the data there for time being, because of time, but we are already moving ahead with that one. Now I'll be shifting my focus in the last 2 minutes about the increasing this. You can increase oil contents, you can increase the seed yield, but you need to grow them more on the marginal lands so that you can use also the land degradation is happening all over the world. And then you need to put more fertilizers and more the chemicals, and drought and salinity are big challenges as well, and high temperature. So in this case, basically, we have a large, several families of gene families, novel stress-associated gene families. And in this case, I will show you only one example from camelina, from the data it came out, is how to increase the oil, the drought and salinity tolerance. But this team basically working on those, all these aspects of those abiotic stresses. And here, this is the impact of climate change scenario in agriculture by 2080. And one of the questions what Dr. Tripathi was asking with Dr. Begum was that some of those, there is a positive attributes of the increase in temperature and CO2, but that positive attribute is mostly in the colder region, when there is a more ice melting and temperature is the agriculture production, the more land is become available. So that's the only fraction of those certain countries in the cold regions. But 75% of the world is basically getting a negative impact of the climate change on agriculture. And then also all these all those this high temperature stress, drought stress, high salinity, heavy metal toxicity, and all these are impacting the crop production. And water is going to be the #1 limited resource for the food production, agriculture production in the future. Currently, 70 to 75% of the fresh water globally is consumed in agriculture alone. This is the green line showing that this is the total human municipal use and all those. But 70% is used in agriculture alone. And farmers are digging up tube wells and pulling the water from the shallow aquifers. And due to the over-exploitation of those underground aquifers, the water table has significantly receded. And India actually is the worst in this situation. India, 85% of the fresh water in India is used in agriculture, and almost 54% of India is water stressed now currently. So what we found is one gene. We found that from this Camelina database, that work we did the transcriptome metabolomic, we found one of the genes is called wax synthase. And then we characterize the wax synthase because mostly the plants, when they have the waxy layers on the leaf surface, it prevents the water loss, transpiration. And basically, most of the water loss happen through stomata, but cuticle loss through the cuticle is also around 25% is residual transpiration happen through that one. And so if we increase that the waxy layers on the leaf surface, it can increase the water retention and increase drought tolerance as well. So we overexpress this wax synthase gene first in Arabidopsis, then in Camelina, and we then generate transgenic lines. And here you can see this is one of the wild type non-transgenic line, and these are the two transgenic lines. The line number 1 and line number 4 and 9. You can see on the leaf surface by overexpression of WSD wax synthase gene, there is an extra layer of wax on the surfaces, and this is stem, and this is very smooth. Then there are some waxy seeds there as well. And when we quantify the total wax loading, you can see in the inset here, compared to wild type, these transgenic lines has almost like a 30% more wax deposition on the leaf surface. And we analyze all these different profiles of the lipid waxes, different component, fatty acid alcohol, alkanes, and all those were increased as well. So what is the impact of this increasing the wax on the leaf surface? So we analyzed the Arabidopsis plants first, model system plants. These are the wild type plants, and these are 4 transgenic lines. And this is watered equally well. And then we withhold the water for 10 days. And after withholding water for 10 days, here the wild type lines are shriveled. They are severely wilted, whereas transgenic lines are mostly green and healthy. And then after additional 4 days, after 14 days, and then rewatering, wild type plants could not recover, that they are dead. And transgenic plants, many of the transgenic lines recovered. And also these transgenic lines, compared to a wild type, they have significantly less water loss from the surface and less chlorophyll leachability as well. So it shows that it's a drought tolerance, significant drought tolerance in Arabidopsis. Also, we look at that whether this increase in wax also increase the tolerance to other stresses like salinity. And here the plants, wild type plants, the plants were exposed to 150 millimolar salt for 3 days and then recover in the clean water. And here, after recovery, the wild type plants could not recover, only few recover, and then transgenic plants all recover, and they're flowering and producing seeds very easily as well. So this increase in wax on the leaf surface and stem surface increase the drought as well as salinity stress tolerance in Arabidopsis. And then we translate this into Camelina. From model system, my lab is doing both basic research and then we translate in the real crops. So we translate in Camelina. Here is the after 2 weeks water treatment, after 4 weeks. These are the no droughts. Basically, these are their water equally well. And then 75% water withholding. After 75% water withholding, you can see after 4 weeks, the wild type plant is dead. Transgenics are still green and producing seeds and these pods. So, and these were the all different attributes under water stress, 50% water withholding, 70% water withholding. The fresh weight was high, number of panicles were high, number of seed yield was significantly higher, as well as the seed size was significantly higher. So it shows that this wax synthase could be very effectively used to create transgenic lines that can increase the drought and salinity tolerance. In fact, one of the commercial maize line developed by DuPont is basically AquaGuard. Naturally they selected through the this natural selection or mutation. It's basically extra wax layer on that one. So it could be very effective. So in conclusion here, what I showed you here is we increase the oil content in Camelina around 16%. We increase the seed yield almost double. And then we translate this, we identified those by changing 2 genes, then we identify more genes, the bottlenecks to increase the oil contents and seed yield. and then we are characterizing those genes. We are translating this approach in edible oil seed crops to increase the oil contents in the edible oils, especially in India and other countries. And then we overexpress these wax synthase genes to increase the drought and salinity tolerance. And with that one, this work was, of course, I'm standing here just to show the work done by the brilliant member of my team. It's their work, their hard work. I'm just basically presenting their work. So it's currently, some of the work was done basically, the current postdoc and two postdocs, and these are the PhD students in the lab, and some of those are the undergraduate students. They also do independent projects. And most of the work in the Kamdina was done by Hesham and Sudesh Sikara. And then my collaborator, Jason White, he's about to come here now. He just reached. He will be giving talk in the afternoon. And Dr. Bhavsan Jing from my department, and some other professors and the faculties in India also. I have the strong collaboration with Jose Puthur at Calicut University, R.D. Tripathi at Lucknow, and Aswini Parikh at JNU, and Dr. Upendra Kumar now. He was at HAU Hisar, now he's at in the Rohilkhand University, Bareilly. And all this funding from the different sources. I will stop here for for any questions. Thank you. Maybe I was a little fast, maybe I went a little over the time, but I try to focus more on the data side.
Yes, yes, that's very good. You know. So, any question first from the audience? Yes.
Sir, first of all, I congratulate to you for delivering very nice presentations, very practical presentations, and many things you have highlighted that is very international type of things. The particularly oil productions in India, you have shown one graph that you have mentioned in India the huge gap is there for the production and consumption, and you have shown many techniques to increase the oil productivity or the yield productivity of a particular oilseed crops. So what is your opinion, as per your opinion, your experience, what are the constants in India? That's why the oil productivity is less in India as compared to USA.
You know more than me here about the issues in India. But very first thing is because we have not diversified the agriculture in India much yet, because majority of that rice wheat cycle is the one that's that need to be disrupted. So farmers need to grow more oilseed crops, and Prime Minister Modi been promoting this for saying from since 2014 he been saying that we need to increase the oil production, edible oil for in those crops. And some of those impediments are the low yield, the climate adaptation is another issue. So maybe not only, I'm not saying that the transgenic crops are the only solutions, but genome editing now with the CRISPR-Cas will be the key. And several labs, I know that in NIPGR and all those, and Dr. Deepak Pental in Delhi, they have done this one, and they knock down some of the same genes that I highlighted, those limiting factors. They are working on those. They knock down those genes through the genome editing, and there is a significant increase. And once those crops can be in the pipeline, those should be that should address some of those concerns.
So, sir, through the varietal improvement, we can increase the oil productivity in India also. Yeah, is there any climatic conditions that we cannot reach the oil productivity that US has in the oilseed crops? So that is there, or we can reach to the productivity level that is US has in particularly oilseed crops.
Well, US has basically more in the soybean. US canola is less than US is more focused on soybean. Canada is more on the canola side. So, but adaptation, increasing the crop production on the margin lands or wastelands, and increasing the yield and increasing the tolerance to different stresses so that farmers can have higher yield are the key factors. And actually, this is currently just going on in the Supreme Court yesterday and day before yesterday, that the GM mustard case was heard again. And now Supreme Court has decided that we will see what could be delivered in the interest of farmers. So this is debate, but the modern technologies need to be adopted, need to be put in practice in agriculture here in India, and to address. It cannot be you cannot be one silver bullet. You need to address from multiple fronts. Increasing diversification, increasing more oil production, more oil seed crops, maybe increasing the range of the production in the crops that they are not currently used in the land for the oil seed production, and waste could be could address some of those.
Thank you. Thank you.
Yeah.
Thank you very much.
There are multiple fronts actually. Yeah.
Okay. Thank you, sir.
Any other question? Okay, if there's no question, I have some questions.
Please.
Number one, if you increase put three genes together, that is wax farming and and other two genes you talked about, lipid synthesis genes. Will it enhance both productivity and as well as resistance to water stress?
Yes, actually, we have a stacked or pyramid, not 3 genes. We have pyramided 5 genes now. Some of those, the 2 genes that I showed you, the rate limiting stack, and then other genes in the push-pull retention, kind of those 2 genes from that, and one of this wax synthase. We put 5 genes in camelina now, and we are characterizing those. So there is a technology available for gene stacking approaches. So we are using those, and you can put those. And we have yet to see what the impact will be. But I can say that these two genes, the double transgenic lines, it's not only validated from the greenhouse, from the lab to the greenhouse, but we did the field trials for the open field trials of the transgenic camelina in Massachusetts. That was the first open field trial in Massachusetts of any transgenic crops, and we did it. Actually, in fact, the productivity was much more higher in the field than in the greenhouse. Yeah, there might be several factors: more sunlight, more CO2, kind of those. Yeah.
Okay. But none of these none of the genes are responsible for photosynthetic genes.
No, these were not.
But they are enhancing photosynthesis indirectly, you say?
So these were not. So the point was, the project was at two stages of the project. One was to increase the photosynthetic efficiency. Second was to pull the extra carbon from photosynthesis to create a sink in the oil. That was my project and my collaborator. So ultimately, aim was that the gene he will characterize for increasing the photosynthetic efficiency, mostly bicarbonate transporters and all those to recapture the waste, CO2 waste in that, and putting those genes along with my genes. That was the ultimate strategy. So, but unfortunately, he passed away last year, and Dr. Dennis Snell, we lost him. And it was a tremendous loss, personal loss to me, because he was my strong collaborator. He moved from UMass Amherst, he moved to Michigan State, and then because of cancer, and we lost him. So the project was stalled at the moment, so we didn't move further because the photosynthetic efficiency project was taken by a company, and the company is also working with us to put those together. So a private company, Yield 10, will be doing that, those basically. Yeah.
Right. Thank you very much for the excellent lecture. Let us give him a big hand now. Yeah. Thank you very much, Dr. Ankar.
Thank you so much, sir. Now we'll be having Dr. Soren Das, Agricultural and Ecological Research Unit, IISR, Kolkata, and he'll be talking on the topic, abiotic stress management in plants, innate behaviors in mangroves. Give him a big round of applause, ladies and gentlemen.
General editing. This you cannot do a general editing.
Genome editing.
Well, you can now.
You can knock down. Enhancement, expression of enhancement is possible, but with increasing the promoter activity. Yeah, but that's the only possible. Knock down, deletion, insertion, disruption on that what. We have a fluorescent tag. We can just throw UV light and transmit it will glow red. Yes, sir.
Yes, sir. Already in the
Yeah.
So if you like,
the plants itself, I think there is some stress always imposing the plant. It have some response to its growth and development, right, like this. But stresses, I'm mostly talking about the abiotic stress, not the biotic stress at all. Plant stress is defined as some changes which may be environmental, which may be imposed by mankind itself, which issue for their ideal development and response. There are some two types of stresses. One is biotic, another is abiotic. Biotic stresses are, that the microorganism, viruses, parasitic plants, and some insects like this. But environmental stresses conditions are much more broader in the sense. Environmental factors can change by human itself. It is some great extent. Soil nutrition, water logging is a major stress of the plant, we know. And mostly I'm talking about the salinity stress. Abiotic stress is defined as the negative impact of non-living factors on living organisms in a specific environment. It is a common example. It is found that the major crop loss, it is somehow estimated about 65% crop loss may be causes from the abiotic stresses only. And it may be some biotic stress also, but in crop plant, it's found that the abiotic stresses are mostly depleted the crop productivity as well. Mostly the plant sustainability affected on priority three constant: plant adaptability itself, another is environment, and another is demographic compulsions. Sometimes we need some more development without looking after its future constant. That's why the forest products, we just acquire the forest products and something forest unplanned resource of forest products may damage the forest itself. And sustainability, plant adaptability means there are some inherent characteristics of some plants itself, By those, they cope up the harsh environmental factor of the environment. And thirdly, it is not a matter of joke that environmental factor mostly governed by somehow mankind itself. Anthropologic pressure is somehow mostly affected on the earth itself, by which the environmental stress begin some increased day by day. So abiotic stress tolerance of the plant, it is a multifaceted concern that morphological adaptation must be there. It's inherent. There are some adaptive characteristics, some anatomical specialties must be there, some physiological activity also, and lastly, genetic and enzymatic, by which the polymorphic expression of a plant itself, it should be there, by which they can cope up with the changed environment easily. Firstly, in plants, the first line of defense against abiotic stress is in their roots, no doubt. Different plant species have unique adaptation when exposed to variety of abiotic stress condition. Mangrove plants have developed some complex morphological adaptation allowing survival and success in their stresses habit, in high saline habit. Mangrove plants, you know, it is the coastal barrier, coastal first defense line from the against the sea surges in tropical world. We have the mangrove vegetation is in highest formation, in highest biological factor in Sundarban. I think Sundarban region is the largest mangrove block in the world. Though it is 2/3 portion under Bangladesh and 1/3 portion in India, still there are salinity difference in the total Sundarban, by which the plant colonization of the such species diversity in Indian part of Sundarban is somehow in more stressful condition, as which this mangrove plant thrives in the estuary, not in saline condition. In estuary, that means there have to some sweet water in mix in the sea mouth where the plant thrives. But in Sundarban region, we get the sweet water influx from the river side. In Indian part, it contribute only by the Ganges. But in Bangladesh region, it contribute through the Ganges, which is named as Podda, and another is Meghna, that is come from the upper stream of the India, upper side of the northeast portion of the India, Brahmaputra, and goes through the Bangladesh, named Meghna, and two stream collides in Bangladesh Sundarban, that is somehow sweet water influx in somehow reach in Bangladesh portion. That's why the salinity level in the Bangladesh portion of the Sundarban is somehow lower, and Indian part is somehow higher. One question arises that mangrove plant itself have some inherent characteristics to withstand the salinity. What is their abiotic stress? Now, the salinity stress they can withstand, yes, in reality so, but there are some have upper limit to some extent. Experimentally, it was proved that 15 to 25 ppt they can withstand saline condition, they withstand easily to some extent by some plant, not all the. Some important plant like Shundari, Heritiera fomis. It is on which the name Sundarban, Shundari. It is somehow extinct in Sundarban region now, as because of the salinity. Okay, let's come to the point. And one thing is leaf succulents. Leaf have some succulent feature in the all mangrove plants. These leaf succulents is only for its due to presence of some water storage tissue and some terminal tracheids acts as a water storage tissue. Mangrove leaves are succulent in nature with high water content, and expressionally provided the leaf thick succulence is increased along the increment of salinity of the substratum. Four species we took for an example: Avicennia, Bruguiera, Sidiops, and Heritiera fomis. You see that Heritiera fomis have the lowest leaf succulence than the other three mangroves. That means Heritiera fomis anatomically, on the basis of the leaf succulence, at least somehow weaker than the other three mangroves. Heritiera fomis is called Shundari. Next, leaf succulence is a common feature in most of the salt stress plant. Cuticle thickness increased in saline stress imposed substratum. Distinct water storage zone present in the hypodermal layer of the leaves. Stomata with thick cuticular outgrowth forming a beak-like structure which help to restrict transpiration. To withstand water stress, terminal tracheids act as a capillary water storage device in leaves. They also provide mechanical strength of the leaf. That means the coriaceous nature of the leaves to just protect from the herbivorous animal. You see here the stomatal feature. There are oh, no, I can go by, no, not yet. Is where?
This pointer.
Pointer.
Yeah, so you can use this one to show. No, that's not working.
That's
there.
not working there. Right?
Yeah, that's not working there. There is a laser pointer here. Pointer option, laser pointer. So you can use this one now.
Oh, right. Thank you very much. You see, in Bruguiera, is a mangrove, typical mangrove in say Kakra. There are some cuticular out in cross section of the leaf stomata and rhizophora also. There are simply some cuticular outgrowth and forming a beak-like structure, which is called ledges. Beak-like structure means it closes the stomatal pore on its own need. This is a feature of stomatal structure which restricts the water loss of the leaf. And there are some excess hypodermal layer which control the water content here and make the leaf succulent. And there are certain leaf gland, which is a leaf gland in cereals like this. This leaf gland accumulates excess salt to its glandular form and ultimately exert in the outer side, by which they can cope up with the high saline condition of the environment, substratum rather. Why we are talking about, I am talking about the to some extent excess on leaf, as when there is a paper, the root brain hypothesis of some plants, leaf considered as the brain of the plant. That's why any stress condition you can judge from its leaf itself, leaf structure, leaf accumulating substances like these. Another thing is salinity mediate specific leaf area. It is changing with the increment of the saline condition. You see, the Bruguiera gymnorrhiza, Exocaria agalocha hereditaria fomis, in this thing, specific leaf area is much more higher in saline condition than in its non-saline condition. We made an experiment, the same plant placed in a saline condition, normal, and placed it in a mesophytic condition. In that experiment, we studied that the specific leaf area, which is specific leaf area, is a function of leaf thickness and leaf density, which is come from the leaf density estimated as a ratio of the fresh mass of its volume. This SLA plays an important role, trade-off between the source of accumulation, resource of accumulation, and constraints imposed the leaf structure. Low SLA is a typical characteristics of the plant grown in poorly resourced and high light environment where conservation of water and nutrition must be promoted to ensure its ecological success. That's why the SLA in the mesophytic condition, it is somehow higher in all the plants, all 5 plants studied here, than the normal one. That means these are the saline condition, these are the mesophytic condition. This brown is mesophytic condition and the blue is saline condition. That means in SLA is somehow lesser in salinity condition. Now come to its stem structure, wood anatomy. You see, the more number of vessel element per unit area, shorter diameter and length, recognized as efficient stress management practices in the plant itself. In the 4 plants, Avicennia, Bruguiera, Cereus, and Heritiera, the frequency of the vessels is much lesser and the average diameter of the vessels is much bigger. Small diameter and much more frequent. It is the efficient characteristics of the salt tolerant condition. In that sense, Heritiera fomis inherently to some extent weaker in salt tolerancy. That means if Heritiera fomis is now in Sundarban, it's mostly some extinct plant. Almost it is in ICU and endangered species also. If we want to rescue the plant, we have to plan differently. So stress adaptation in physiological.
Could you go a little fast? Could you go a little faster?
Okay, no problem. Stress adaptation in physiological process. The elevated rhizosporic solute concentration and retarded water uptake, including induced considerable high by the means water potential deposition into excess compatible solute in leaf that may dehydrate the cytosolic condition of the cell. The cell itself accumulates some competent molecules which are not stresses for itself. That means some amino acids they accumulate within the cell sap to access its water potential to withstand the salinity condition. Water potential in the root is much more higher than the high tensed somatic cell of the leaf or itself. Otherwise, nutrient cannot go up from the substrate. Osmotic potential of the root and leaf, a comparison is there. Considerable difference occurs in osmotic potential of leaf and root excess in Heritiera. Relation between osmotic potential and the water content of the leaves. Leaf osmotic potential is more negative, where the amount of water storage tissue is less. Leaf osmotic potential is less negative, where the storage of tissue content is higher. In Heritiera, in all the aspects, it is somehow weaker in condition to the other mangrove species which are dominating in the Sundarban area. Degree of salt tolerance can be attributed to the accumulation and compatible solute in the cell sap, dual effect of osmotic adjustment for the cellular homeostasis, uptake of inorganic ion, occurrence of excess inorganic ion of the vacuoles. The cytoplasm has to confront biphasic stresses, internal stresses caused by the accumulation of ions and external stresses. But if the plant synthesizes compatible solutes, which can organic solutes within the cell sap, plants accumulate de novo some organic compatible solutes like amino acids, proline, and alanine, tyrosine, soluble sugar, polyads, betaine, glycine, betaine, and to adjust the osmotic potential. And moreover, these organic solutes do not interfere the normal metabolic processes. This is an experiment that incidence of free amino acids in leaves in different salinity level. If the salinity level rises, some plants this accumulation of this compatible solutes may rise up to some extent, but Heritiera fomis is somehow weaker in this context. Another thing is stress include a drought or desiccation, salt, chilling, heat stroke, heavy metal, UV radiation like this. The enhancement production of ROS, that is a simple metabolic process during stress, can poses a threat to the cell and may stress condition enhance the expression of ROS scavenging enzyme. That means if ROS is always getting high, then the cell itself is somehow in stress, and major scavenging enzyme should be there to scavenge the excessive ROS.
Could you conclude?
Right. Some oxidative stress enzyme also there. some peroxidase, superoxide dismutase like this, they are also somehow in lesser condition in hereditary form. That's why the plant is inherently weaker than the other Sundarban species. That means abiotic stress in somehow reactive oxygen species to over reduction of the photosynthetic electron transport chain causes generation of active and oxygen species, antioxidant, polyphenols, flavonoids, flavonol, antioxidant enzyme like these, stressful scavenging like this. These are all the stress adaptive stress condition to make mechanism to cope up with the effective stress in the Sundarban plant. That means integrated approach should need towards the understanding of the process that some of the improve of the plant communities under abiotic stresses. Thanks.
Thank you very much. Due to lack of time, we don't have a
Okay.
session now, question session now. I've given your presentation. Have you given the presentation? I'm going. Now a great pleasure to invite Dr. John C. White. Jason White, sorry. Jason C. White, sorry. Okay. He is the director, Central Agriculture Experience Center, Connecticut.
Which one's up?
Okay. Well, thank you for the invitation to speak here. I literally just arrived from the United States with a 8-hour layover in Delhi, so I may fall asleep before you do. So what I'd like to do is talk about some of the work we've been doing specifically on. Let me see if I can get a pointer. On using nanobiotechnology to increase the stress resilience of different crop species. So some of what we were just hearing about reactive oxygen species is going to be very relevant to what we're talking about here. Now, I noticed in looking at the agenda that I'm actually one of a very small number of people talking about nanotechnology. So I think in terms of the way to think about nanotechnology is basically when you have a material that's at the nano scale, in other words, the particle size is below 100 nanometers, that material, most often we're going to be talking about nutrients, that nanoscale nutrient behaves differently, both physically and chemically, and also even biologically. So that's a behavior that we're going to take advantage of. So the nanotechnology program at the Connecticut Agricultural Experiment Station in the US is about 15 years old. And the project actually started in nanotoxicology. My PhD is in environmental toxicology. So when I started working with engineered nanomaterials, it was mostly out of a concern that these materials were going to have a negative effect on crops, on agriculture. So we started investigating a lot of different questions related to how these materials were going to negatively impact plants. And what we noticed as we got into this was that reactive oxygen species, ROS, were a critical part of the toxicity of these materials to plants. But what we also noticed after a number of years was that under certain circumstances, these materials were actually not only not toxic, but they were conveying benefit to plants. So that's when this applications-driven research started. This work's funded mostly by the U.S. Department of Agriculture, although I'm part of two centers, which I'll mention briefly. But the other interesting thing that we noticed is that in some of the instances where these materials were conveying benefit to plants, reactive oxygen species were also really important. So we started thinking more broadly about reactive oxygen species. And we published this paper in Nature Food in 2022. And a lot of this was looking retrospectively at a lot of the data that we had, specifically around ROS, reactive oxygen species. And as we just heard, these are metabolites that are generated. They're generated actually during basic cell metabolism, but they're also generated under stress, including exposure to xenobiotics. And as we know, these reactive oxygen species, if the concentration is too high, they're going to damage biomolecules, organelles, so forth and so on. So cells, plants, animals, bacteria, otherwise, we've evolved pathways to kind of quench these reactive oxygen species. And those are some of the pathways you were just hearing about. But what's also important is that these reactive oxygen species are also signals. It's a signal to the plant that a stress is happening, and then the response to that signal is a significant upregulation of all of these stress-related pathways. So we started thinking about this in two different ways, and that's what you see up here on the right. So can we use nanomaterials that are able to scavenge reactive oxygen species, and can we also use nanomaterials that are actually going to trigger the reactive oxygen species and the subsequent response? Can we use that together to basically control homeostasis in these cells, in these organisms? And can we use that for our benefit? Because we know that if we're upregulating these stress response pathways, that's going to convey tolerance to abiotic and biotic stress. So this first pathway, which we have a lot more data on, is recovery. That's ROS scavenging. Plants exposed to something, it causes stress. We can supply a nanoscale micronutrient to alleviate that stress, to scavenge those ROS. What's interesting, and I'll finish with this data in about 15 minutes, is this preventative strategy. Can we actually expose a plant to a material that will induce stress response and prepare the plant for a stress that's actually going to happen in the future? And when you think about climate resilient agriculture, that's going to be something that's going to be interesting. But taking a step back, as I said, we got a lot of this retrospectively looking at our data. So in 2014 is when we started working on using nanoscale micronutrients as a means to induce tolerance to biotic stress, to pathogens. So we started working with fungal pathogens. In the US, we spend about $600 million a year on fungicides, and we're still losing about $200 million in revenue to fungal diseases. So we started considering micronutrients because these are the elements, these are the nutrients that are really important to these stress-related pathways. But we know that there's difficulty in getting sufficient amounts of these into the tissues where you need them. So for a soil-borne pathogen that's going to infect by the roots, you need these micronutrients in root tissue. So that's where we have to get them. So we actually started asking, well, what if we supply these nutrients in nanoscale form? We know nanomaterials are smaller, they're more active, they're more reactive, they're more mobile. So we aren't, even though we're working with disease systems, we're not targeting the pathogen directly. We're targeting this plant response, this stress-related response. So this was the first paper we published in this space. We published it in 2016 in a journal called Environmental Science Nano. We work in an agricultural experiment station, so we want to come up with strategies that farmers are actually going to use. So our strategy was pretty straightforward. We do a foliar application to seedlings, and then the seedlings are transplanted to the field. That's how a lot of agriculture actually happens in New England in the United States. So we looked at different micronutrients, copper, manganese, and zinc in different forms, the nanoscale form and the conventional forms. So we're adding 100 ppm, which sounds like a lot, but it's a foliar application. So we're only adding a couple of milliliters, which means we're well below a milligram of these nutrients. So, and the short of the long is what you see is, so this is field data. This is eggplant and tomato under infection with Fusarium, which is a fungal pathogen. Focus on the green because that's the copper. And what we saw was, and the middle green bar is the nanoscale copper. With that foliar treatment, months later, we're getting significantly greater numbers of fruit being produced per plant. This is eggplant data. This is a measure of disease. So we're getting significantly less disease, and we're getting much more copper, well, significantly more copper in the roots. And I'm not showing you the data, but the alleviation of stress and ROS specifically what was implicated. So because this is a field study, we knew that we spent about $44 on nanoscale copper. That's what our cost was for the material. And in terms of yield, we were increasing economic return for the farmer by about $10,000 per acre, or the way I prefer to think of it in terms of increased food that we're producing. So one of the other, as I mentioned, I'm in a National Science Foundation funded center. It's called the Center for Sustainable Nanotechnology, and there's a significant initiative in that center. The center runs for 13 years, and we're just starting our 12th year now, so we're almost done. But there's a significant effort in looking at how we can control the chemistry of these micronutrients to optimize the benefit, optimize the recovery from biotic stress in this case. So we took commercial copper oxide nanoparticles, which are just the same materials we were just looking at. And then we also made a material where we changed the morphology. We made it a sheet. We changed the composition. We added a phosphate. We wanted to see how tuning that chemistry would alter the response. And so this is another field study. This is with watermelon and another Fusarium pathogen. This paper was published in 2018. Same exact strategy, foliar application to seedlings. Then the seedlings go out into the field. And in terms of disease response, what you see is the yellow is the commercial material. You got to get up to a couple of hundred ppm to see a decrease in disease, or yellow down here, an increase in biomass. But look at the dark green bars. That's our custom copper phosphate nanosheet. At a 10-fold lower concentration, we're getting a significant reduction in disease and a significant increase in biomass. So by tuning the chemistry of your material, you can dramatically increase its efficacy. So this was the paper that we really kind of worked out this mechanism relating it to reactive oxygen species and stress. We published this in Nature Nanotechnology at the end of 2020. We switched to soybean and the Fusarium that affects soybean, which is causes soybean sudden death syndrome. This is just a greenhouse study, but we looked at a couple different kinds of commercial, a couple of different kinds of custom copper nanomaterials, because again, this is work done within the center. And so this is just a greenhouse study, but what we see is that when that fungal pathogen is present, you get about a 60 to 70% reduction in yield, photosynthetic output. You see significant oxidative stress as measured by a number of different parameters. But importantly, the nanoscale copper treatments significantly alleviate that stress. They increase biomass, they restore the levels of photosynthesis and other damage. So what we did here was this was the first time we really started looking at gene expression. We looked at almost 24 defense and RLS related genes in the roots. So we're doing a foliar application and we're seeing the response in the roots, and all of these pathways were significantly upregulated. But what was interesting is the extent to which they were upregulated was a direct function of the properties of the different types of copper that we were using. And we were actually able to describe that computationally. There's a computational chemist group in this center. And that's what ended up getting us the cover, I think, of this journal. So it's not just all about copper. One of the other materials we're looking a lot at is sulfur. And this is a lot of work we're doing in collaboration with Professor Om Prakash-Dankar, who's sitting in the first row here at the University of Massachusetts. So this is a USDA funded project. This was a paper we published in ACS Nano at the end of 2022. And in this case, sulfur is really interesting because in the United States, 25 years ago, nobody cared about sulfur because there was enough sulfur coming down in acid precipitation that there were no deficiencies in soil. But we had the Clean Air Act, which was passed, and now many farmers in the United States have to add sulfur to soil as an amendment. The problem is when you add sulfur to soil, you get significant changes in pH in the microbiome. So we were interested in what would happen if we added that sulfur in nanoscale form. And again, this is specifically in disease systems. So we looked at a coated and an uncoated form. This is tomato and a fungal pathogen. My postdoc on this, who's now a staff scientist with us, measured all sorts of endpoints over about a 2-week exposure period. So it was a time-dependent harvesting of these plants over those 2 weeks, looking at basically every parameter you could think of, including some time-dependent gene expression and metabolomics by high-resolution LCMS. So in terms of what we saw, when tomato is infected with Fusarium, we get an 87% decrease in biomass. This is just a greenhouse study. I'll show you the field study in a minute. But what we saw was our 2 nanoscale forms of copper or 2 nanoscale forms of sulfur were the most effective at reversing the impacts of disease, increasing biomass. The other forms of sulfur actually do pretty well, but as I said, those other forms of sulfur are what are causing all the problems in soil. So what we noticed was that when you looked at the ratio, looked at the distribution of sulfur in the different tissues as a function of treatment type, and in this case, this was a soil treatment because, as I said, that's how growers are adding it. But we saw particle size specific translocation ratios. We looked at 2-photon microscopy and gene expression, and it really looks like when you have sulfur in nanoscale form, you're actually using a different uptake pathway. You're not really using the sulfate pathway, which most plants are using. You're using this elemental pathway, which ends up being much more efficient for the plant, particularly under disease. So the time-dependent gene expression and time-dependent metabolomics ended up being really interesting. As I said, I don't know if you can see this, but we're harvesting plants at 4, 8, and 16 days. And what we saw was that, and this all was a function of nanoscale treatment, that the gene expression really wasn't impacted at 4 days. Many of the stress-related genes were upregulated at 8 days, and then dropped off at 16 days, whereas the metabolites, there was a delay, obviously, because of the central dogma. We didn't really see much metabolites response until we got to day 16. But again, the pathways that were most impacted, that were most interesting, were all of those surrounding stress and reactive oxygen species. So this was a field study. It had the exact same design as that ACS nano paper we were just looking at, except none of the endpoints. The only thing we were looking at was yield because it was a field study. And this was published in 2022 as well. But the results were basically the same. We got a significant increase in disease suppression and increase in biomass when we did this nanoscale sulfur amendment. One of the things that we also see, and we've seen this across systems now, is we see essentially a nano-enabled biofortification of the edible tissue. When you upregulate these pathways, many micronutrients tend to increase, and we suspect many organic nutrients are as well. But again, here's the math. Here's the practical aspect of this. This is how you sell it to a farmer. We're only spending about $33 an acre on nanoscale sulfur. And in so doing, you know, we more than doubled the yield of tomato per acre in a healthy system, from 4,900 kilograms to almost 12,000. And even in the disease system, we're still doubling our yield. And we're doing this in the complete absence of any fungicides at all. So it's not all just about biotic stress. We're also interested in abiotic stress and how you can control ROS homeostasis to alleviate that. And again, we were just hearing a lot about abiotic stress. This was a paper we published in 2019 where we're looking at drought. So in this case, the D is not disease, it's drought. And we're looking at zinc, nanoscale zinc. This is sorghum. So when you expose zinc, when you expose sorghum to drought conditions, you see a decrease in yield and many of these other important parameters. But if you have supplied nanoscale zinc in the soil at relatively low concentrations, many of these negative impacts are reversed. And this is all a function of the zinc being accumulated in the plant and upregulating these stress-related pathways and helping to alleviate the ROS damage that the drought conditions causing. We see a similar situation with cerium. And in this case, the abiotic stress we're looking at is salinity. Now, cerium is interesting because it's actually not a nutrient. It's not required, but it's a known antioxidant. So again, honing right in on ROS homeostasis. In this case, we used cotton because we had money from the cotton board, so we had to use cotton. And it was a seed treatment in this case with a coated cerium oxide, but the same exact thing. When you expose those germinating seeds to salinity stress, if they were pre-treated with that nanoscale antioxidant, and I should mention all of these effects that we're talking about are nanoscale specific. We always have the conventional controls, the bulk and the ionic, and they never work or almost never work. So when that nanoscale cerium is present, you get increased growth, you get increased root vigor under the stress. When you actually measure the ROS, you find less ROS if the seeds were pre-treated with cerium. And when you look at what was being increased in terms of gene expression, it's all of these same pathways that we're talking about. Excuse me. So we have a lot of work going on in this space. This is just papers we published in the last 2 years, looking at different micronutrients for the most part, and how they can impact ROS homeostasis under a variety of conditions, stressed, non-stressed. You just see some of what we're looking at here. Molybdenum, because it's for molybdenum and nickel because of their impact on nitrogen fixation, magnesium, lanthanum. What we've also started to do is look at more green or biosynthesized nanoparticles. This is a topic that's not very popular or not looked at very much in the United States, but in lots of other countries, including India, biosynthesis of these nanomaterials is quite intensely being studied. And the reason is you get a different kind of material. You don't just get nanoscale selenium, you get nanoscale selenium with all of these biomolecules associated with it, and it behaves differently. So as I said, lots of interest. So I'm just going to finish on the reverse. So the first half or the first 2/3 of this was you have a plant, it's exposed to a stress, disease, salinity, drought, and you can supply nutrients in a nanoscale form, and you will get a nanoscale specific alleviation of that stress through ROS quenching. So can you do the other thing? Can you actually pre-treat plants or seeds with a material, a nanoscale material that's not going to be overly stressful, that's not going to cause damage, but it's going to be, the dose is going to be appropriate to actually just upregulate all of these pathways. So then when that plant later, weeks, months later, is exposed to a stress, it's almost like a vaccination. It is prepared to handle that stress. So this was one of 2 papers we published in this space. This was published in ACS Nano last year, at the end of last year. And the nanomaterial that we're using to induce stress is silver. We know this causes an ROS response. So what we need to do is we need to find a dose that's low enough that it doesn't actually cause toxicity, but it's high enough that it upregulates all these pathways. And in this case, we're using rice. So we call this stress training. We train them as seeds or seed priming or leaf priming, or together seed and leaf priming. And so then those trained seeds are grown, and then weeks later, they're exposed to either rice blast or cold stress. And then we do the metabolomics and the transcriptomics to figure out what's going on inside of the plant. So I know I'm almost out of time. I'll finish quickly. So this is the data. So up here is the disease response. So this is just lesion size on the leaves. And what you see is regardless of how you did that stress training, when the rice is exposed to that fungus, literally weeks later, you see significant reductions in disease across the board. And the greatest reductions in disease are when you do the seed priming and the leaf priming together. You're looking at a 70–80% reduction in disease presence. Here's your cold stress data. It wasn't as dramatic, but still you see an increased tolerance with the seed and leaf priming, increased tolerance to cold stress. Again, and this is cold stress that's literally happening almost, I think it's 28 days after the treatment. So I won't dwell on this too much, but as you may suspect, all of the pathways that are being upregulated are these same exact pathways. They're all related to ROS scavenging or stress response. And you see that in terms of gene expression, and you see it in terms of metabolites. So the second study, which was just published in December, we switched to corn. We switched to our stress-inducing agent. We included silver again, but we wanted to switch to an actual nutrient so we could maybe get some additional benefit. And then the other thing was we looked at single stressors, but we also looked at multiple stressors together. So if you prime a seed with a corn seed with iron and then expose that corn plant a month later to drought and cold or drought and saline conditions. When you looked at the single drought conditions, corn behaved the same way as rice did. You get that stress alleviation, you get that priming effect, and you get it with not just silver, but you get it with iron. But what's really interesting is when you look at multiple stresses, you see increased tolerance. Again, weeks later to multiple stresses. And what's really interesting, and we haven't published this yet, is this seems to be transgenerational. We actually collected the maize seeds, even though we published this paper, we collected the maize seeds and grew them out under stress conditions in the F1 generation, and they're still exhibiting some of this tolerance. So hopefully in 1 minute too long, I convinced you that nanotechnology does have significant potential here. There's plenty of ways to use nanotechnology poorly in agriculture. I think we're working on some ways that are actually safe and sustainable and can exert really significant benefits to plants. And being able to control that ROS homeostasis, control when and how those pathways are upregulated, really you can have some dramatic effects on yield under stressful conditions. So we've got a very large group of collaborators, and I listed most of them up there. And Om, sitting in the front row, is one of my best collaborators and best friends. So it's always good to come to a meeting with him. But I will stop there.
Thank you. Thank you. Any very, very interesting lecture.
Right before lunch.
Yeah.
Bharat Prasad from Kansas State University. Very nice presentation, Jason. High quality publications and supported with the data. So my question is related to the stress memory, which you talked about at the end of your presentations. So you slightly touched about the transgenerational things. I was wanting to know if there is a memory. Does these nanoparticles uptake go into the seed, and then it still keeps the memory, and in the next generation, they're going to provide stress tolerance, whether it is biotic or abiotic stresses, kind of epigenetics of this stress and nano interaction.
Yeah, as I alluded to, and we've only done the experiment once, so it hasn't been replicated yet. But those maize seeds, when they were exposed to subsequent stresses in the F1 generation, there was an equivalent level of tolerance to single stressors. We didn't look at multiple stressors, so there's clearly something going on. But you raise a good point about the nanomaterial itself, because a nanomaterial of copper or sulfur or iron is completely useless to a plant. The biologically active form is the ion, right? I mean, that's what's needed. So there's a significant mechanistic gap in our understanding. We are supplying these nanoscale nutrients either as a foliar treatment or a seed treatment, or in all these different combinations, and we know under certain conditions, there are some nanoparticles getting into plants, but a nanoparticle in a plant is not going to give a plant any benefit. So that's when this chemistry of the material becomes really important, because you can tune its dissolution. I've got a collaborator at the University of California. He actually just published a paper in Small where he took he took carbon dots, and when he coated them with sucrose, he could actually put the carbon dots on the leaf, and he got 70% transport of the sucrose related carbon dots to the roots, because the phloem transporters were recognizing the sugars. So if you really need to load something into the roots, coat it with a sugar, because that's what's going to be translocated. So this ability to understand the chemistry, I think, is really going to be critical, because you can control where the material is going to go.
Okay.
Quick follow-up. So the way it is manufactured, you said, has an impact on the biological activity. So if you were using the biological ways of manufacturing these nanomaterial, will it have a more transgenerational positive impacts? That's number one question. Second one is, have you looked at the translocation of these nanoparticles, fluorescence using, are they ending up in the seed to have an impact on the next generation or not?
Yeah, so to answer your first question, the transgenerational impacts with the biosynthesized nanoparticles, I think so, because the little bit of work that we've done so far does suggest that when you take that nanoparticle and generate it by a biosynthetic process, in some ways you get a completely different behavior because of all of these polyphenols and all these, you know, plant nutrient or plant metabolites that are associated with it. So I would suspect if you're going to anything that you're going to see with a chemically synthesized material, I think there's a pretty good chance it's going to be greater with a biosynthesized one. We have no data to support that yet, but that's just my hypothesis. The second question, which I just forgot. The tracking. Yeah, we have tried to do that. In the 2 sulfur studies, we used 2-photon microscopy. And sure enough, when you put sulfur nanoparticles in the soil, you can see sulfur nanoparticles coming into the roots. And 2-photon microscopy is nice because it's a non-destructive process. So we actually have videos. We have movies where we can watch the particles coming in. That's another reason we're using carbon dots, because confocal fluorescence microscopy can be used to track those. So you definitely do see particles moving into plants, which is generally something you want to avoid or minimize. But in some cases that might be what you need, because if you're doing a soil treatment and you want these materials in the leaves, then the best way to get that is have the particle move all the way and then dissolve when it gets there. But you need to be very careful, because if you start loading nanoparticles into rice grains, there's a lot of people who are going to be upset that you've loaded nanoparticles into rice grains. So that's why way back in the beginning, that's why we started focusing in on micronutrients, because we knew if we saw a benefit, the benefit was not because of the nanoparticle itself. It's because the nanoparticle enabled us to get to a better location or to do something different, but it's still all about the dissolution. I've got collaborators who are using carbon nanotubes in agriculture and still want to use silver. I think that's a terrible idea, because they're not materials that are natively found in plants. So it's inherently a higher risk. And if the risk is higher, the regulatory community is going to look at it with greater scrutiny as well. They should.
Well, I have also a lot of questions, but due to lack of constraint of time, we skip the next of the questions now. So thank you very much, for an excellent lecture. We all are really delighted. No, we have many questions for you.
I'll be here all week.
Certainly, we'll ask you after the lecture.
Okay.
Thank you. Thank you very much.
So the next session is by Dr. Ajit Kumar Shashani, Director, ICAR-National and Botanical Research Institute, Lucknow, India. He'll be talking on the topic metabolic engineering for climate resistance agriculture. This would be on the video conference. Please watch on screen.
Slides are visible.
Yes, sir, it is visible. You can make it slideshow, sir.
Is it okay now? It's slideshow mode. I think it is slideshow mode. It's okay.
Yes, sir. It is okay, sir.
Okay. Okay, very good afternoon to everyone. Should I start now or you will be telling something?
You may start now, sir.
Okay. So let me start. So respected chairman, sir, and the audience. So I was speaking something about the metabolic.
Dr. Sasani, are you able to hear me?
Yes, yes.
Hello?
Yes, yes, I am able to hear you.
Yes, sir, you are audible. Please go ahead with the slides.
Okay.
Able to hear us, but his voice is not coming out. Yeah.
So chairman, sir, is telling something?
Yes, go ahead. Thank you.
Okay. I'm audible loud and clear, or I'm not clear?
Yes, very clear.
Okay.
Very close.
Okay, wonderful. So I'll be telling something about the metabolic engineering for climate resilient agriculture. So everywhere, if you see the sustainable development goals, every seminar you go, you will be finding out everywhere, all the people, all of us, we saw this. But here I am showing this to mean that agriculture is related to most of the SDGs. So out of 17, maybe 10 of them, they are directly related to agriculture. Having said so, the concentration and distribution of food insecurity by severity that differs greatly across the region of the world. We also know that, starting from the Asia to Africa to Latin America, Northern America, Europe, so everywhere. So if you see, so there is lots of food insecurity, and it is severe. I'm just showing this to build my lecture. Otherwise, everybody knows about this. So this is another slide for food security, India insecurity. India lies in that. So we say that we are secure in food, but we are secure in only carbohydrates, not in proteins as well as oilseeds. And we are working for that to be self-reliant on protein as well as oilseeds. So coming further, 8.9% of the world population lives in hunger. Either you admit it or deny it. This is the data. Also, food security, this says that it should be available, it should be properly assessed by the population, and utilization and stability, resilience and stability. These all things are all the dimensions of food security. Again, if you see the green revolution of India, and now, if you compare, the total area of cultivation remain stagnant. It has not increased or not increased much. A very little increase is there. But our productivity has gone very high. You consider in fruits and vegetables, crop plants, food grain production, spices production, cotton production, oilseed production, sugarcane production. What does it mean? It means that we are able to increase our productivity. But that productivity is not sufficient enough to feed the complete population in due course of time, the coming years, that if you consider the coming years. So already I have told that to meet the food security needs, this is Syngenta data, 2050 food production must be increased by 70%. Another dimension, always in every seminar as a symposium, we say about climate change and how we are working on the climate change. That I am telling, that I will be telling. A very small part of it, how we are contributing to mitigate the climate change. But we talk about climate change always and everywhere. So if you see the breeders, how much time I have, sir? Chairman, sir, how much time I have?
20 minutes, sir.
20 minutes, okay. So if you see from the starting of the Green Revolution, we have bred crops for yield to feed our population. Only the characters we have focused, our breeders, they have focused for yield, just like high grain weight, high grain number, more number of tillers, more number of panicles, and so on. So this is the characters we have concentrated. And we breeders, when we think that we will be doing something, so focus on some characters, we focus on that characters and discard all other materials generally. So climate change, I told you, there are many dimensions to it. So there are carbon dioxide increase, methane increase. So we know everything all about heat wave, cold wave, flood, cyclone, coastal inundation. And again, the crops area affected due to climate change in different years. There is uncertainty. The climate change has come such a way that the climate change has come such a way that we cannot predict when the stresses will occur to the crop plant. These stresses are again having many dimensions. It may be cold stress, it may be water stress, it may be water logging, it may be salt stress, whatever it may be. So these are all abiotic stresses, and biotic stresses are then also there. So you might have marked in recent days. So the plants which are the crop plants which are not host to specific insect, they are becoming the host to the insects right now. So different type of insect. Suppose the insect affecting a vegetable crop now affecting the cereal crop also. Also many insects are polyphagous in nature, so they affect all the crops. Many fungi are they're across the taxa they infect. So there is the climate change. It is affecting the crop production as from the slides, maize and wheat. It has been proven, and the data say like that. Also, the climate change has affected the nutrient availability to us through the plant. So the uptake of nutrients has also been decreased in due course of the climate change. I will give a concept here, the synteny. The synteny, we know that in a chromosome, two genes are linked. So there is synteneous. But I will talk about the modern synteny, that is the pathway synteny and metabolic synteny. If you consider different plants, then the pathways are there. Then I'm coming now to the secondary metabolites. I will just rush through because the time is very short. So similarly, the metabolic synteny. I take the example of beta-ocimine. When we analyze many plants, in 377 plant beta-ocimine is there. It may be in higher quantity in some plants. It may be very trace amount of some plants. That means beta-ocimine is present across 377 plants. Like this linalool, this is also present in many plants. So when we say that, and campheine, this is present in 401 plants. When I say that the pathways are conserved. But somehow, when the plants are grown in different ecological niche during the time of evolution, so they might have lost or gained some of the functions, and by which the production of the secondary metabolites are there. So here, one example of, again, the phenyl propanoids and all other things, rosmarinic acid, stilbenes, catechins. So these pathways are occurring in almost all the plants. But whether we are getting the product or not, that determines where the plant is growing, what is the ecological niche. And this is another representation, don't confuse yourself. So the pathways are occurring in many plants that I have depicted that. Again, synteny is observed in different microRNA. So if you see the synteny of the pathway molecules and the genes, they are occurring in across a wide range of plants. Then metabolic channeling. So another concept is metabolic channeling. So we now know about the synteny that occurs. Now let us know about the metabolic channeling. The metabolic channeling, if you see the red line, the upstairs, these are our primary metabolites, these are the secondary metabolites, terpenoids, polyketides, alkaloids, phenylpropanoids. And the name of the plants that have been denoted here, which are producing monoterpenoids, sesquiterpenoids, diterpenoids, triterpenoids. These plants are producing, I have written down the plants, but the plants which are being under the sesquiterpenoids, they are not producing monoterpenoids. It is wrong to say. So they are producing monoterpenoids also, but the dominant product, they are producing sesquiterpenoids. So they have evolved such a way in different ecological niche to protect themselves. We know that we have a very good immune system. Plant, they don't have the immune system comparable to us. We produce antibodies, they don't. For that reason, they have evolved such a way that they have to protect with the help of secondary metabolites and all other materials present within them. So the channeling, so now we say the channeling, the channels means where they are being stored, through which the molecules are being produced. So there are latex vessels, latex cells, trichomes, different places where they are being stored below the endodermis, specialized cells. So there are some packets in like tagetas. So these things happens in many plants. Again, this is our work. So there is channeling. Two genotypes in poppy. One produce latex, another doesn't produce. Because it doesn't produce because it doesn't get space. for the latex vessels. Latex vessels are not formed properly in this plant because the cellulose enzymes, they are defective in nature. Here, the menthol production in a right-hand side panel upstairs. So there are plastids involved, endoplasmic reticulum involved, mitochondria involved, cytoplasm involved. So some limonene is produced in plastid, then it is transported to endoplasmic reticulum. Then from there, isopiperetinol. Then in mitochondria, it is being converted to isopiperetinol, oxidized. Then isopiperetinol comes to the cytoplasm, and cytoplasm produces menthol. So that is one way of channeling. So in different plants, again, I will not go into detail. So then come to the metabolic network. In the metabolic network, some of the networks are confined to specific organelles. Like some of the oxidation steps, they are confined to peroxisome. This is our paper in which the oxidation steps in the benzoate biosynthesis, oxidized benzoate biosynthesis, they are confined to the peroxisome. Again, if you previously people were knowing that phenylpropionate is the main route to produce different benzoates. But now we could prove that in higher plants like petunia, all other flower where smells are being produced, aerogenate dehydrage is the main route to produce different benzoates. That has been proven. Again, the channel, when I say the ethanolamide biosynthesis, a series of paper we have, and this series of paper we have proved that through cycloatenol, campestyrol, cetostyrol, sigmastyrol, ethanolides are being produced. This is through RNAi, this is through by blocking, by providing some precursors, as well as genome editing. We have proved that. These are also virus VIX, virus mediated gene VIX method. We have analyzed in how the metabolites are being channeled. Again, we have also told that there are transcription factors which control the whole pathway, like Warki transcription factor. So this controls the whole pathway. So the pathway is now somehow we know how these are being controlled and how the ethanolides are being produced. So first I told the synteny, many plants is produced. Then I told about the metabolite channeling. In medicinal and aromatic plants, we call them, and this is another example. I will not go into that again. Through defective intron splicing, how the speciations has occurred, and through the chemotaxonomy, we are differentiating different species. That's menthofuran biosynthesis. It is present only in one species, Mentha piperita. The stress response. In CMAP, there is a locust infestation 2 years back during Corona time. You might have observed or read in some newspaper. Here, many medicinal plants, they were growing very well, but crop plants, they were being destroyed. So that is one example how locust, they are repelled by the medicinal aromatic plant. That means metabolite, secondary metabolite, they have some property to repel this, either the locust, insects, or the bacteria and fungi. So they have their immune system just like our immune system. I told our immune system depends upon the antibodies and all other subsidiary factors, and their immune system depends upon the secondary metabolites. So we classify the plants like hydrophytes, insectivorous, xerophytes, lions, fungi. There are classifications like Hodgkinson classification, Bass's classifications, and there are many other classifications we generally follow in the classical taxonomy. But we give them halophytes, gymnosperms, algae, fungi. We tell them lions, epiphytes, insectivorous, xerophytes. They grow in the region where water scarcity is there. Then, and function-wise, they grow in extremity, salinity, drought, shock. They actually withstand the shock, diseases. They face diseases, symbiosis, association, attraction. You tell the name. Many things the plant faces in nature. So they have in their armory the terpenoids, flavonoids. I told about the secondary metabolites. And we differentiate the plants, medicinal and aromatic. How and why? I say that all plants are medicinal. They give aroma. The aroma means small molecules. They diffuse from one place to another. but they are also medicinal many times. So there are Anacardiaceae family, Myrtaceae family, Ericaceae family, Laminicaceae family. They are generally being used for preparing the biopesticides nowadays. So the cereals, pulses, oilseeds, fruits, vegetables, they have also synteny, they have also the metabolic channeling, they have the secondary metabolites, but they are in very less amount. And during the course of breeding, we have concentrated on only yield and yield. So, and we have somehow not given importance to these metabolites. So secondary metabolites, if you think they deter the insect or repel the insect, antioxidants they may disrupt the life cycle, kill or attract. So they are insecticidal activity, antimicrobial activity. So there are many during the corona time, we formulated essential mixtures scientifically, and we could test that this is 99% inhibitory, or it doesn't allow the coronavirus to replicate. So these are the constituents, and the examples of inhibition of different animal viruses are there, but we have studied less for the plant viruses. I have found only tomato leaf viruses from the literature of all the molecules. Similarly, the essential oil components, the secondary metabolites, they kill many animal bacteria, fungi. And when we analyze the total transcriptome of osmum metabolomics in response to different type of stresses, also the Artemisia transcriptome, different type of stresses, abiotic stresses, we found many commonality. Many master regulators, which are actually, if they are expressed in higher amount, then it gives tolerant, the plant become tolerant to many stresses. So these are the common transcription factor, in addition to specific transcription factor, specific pathway which are being induced during the stresses. So one example is cis-hexanal. Cis-hexanal, when it increase in the plant, if you induce it, then it produce callus. There is callus formation, and this callus formation, it gives resistance to tomato yellow leaf curl virus. So that is one of the example. Another example, isoprene and caryophyllene, they confer resistance by different pathway, signaling pathway. So salicylic, they produce salicylic acid and jasmonic acid, and they induces different pathway for tolerance, resistance. Then this is about the biotic stresses I am talking about. This is our own reference. There are some cytochrome genes which induces specific metabolites in Udania and which give resistance to Pseudomonas. This is again our reference. If you induce the farnesyl synthase, this confer tolerance against bacteria pathogen and potato and tobacco, as well as that is the late blight. This gives late blight resistance to. I will not resistance actually. Resistant means completely tolerance. I will say tolerance, because sometimes in abiotic stresses we call tolerance, and in biotic stresses we call resistance. But I'll call all the things as tolerance. What is the degree of tolerance? Another example of our work is in Cajanus platycarpus. This is a wild relative of Cajanus cajan. And if you increase, and we got the indication from platycarpus, which is actually resistance to this Bihar hairy caterpillar as Spodoptera litura. So if there is increase in epicatechin 3 gallate, then it will generally kill the larva in long run. Again, continuing further, we could inhibit the pathway F3-5H gene, and if you inhibit, then epicatechin 3 gallate production is less, then there is actually insect feeding occur. Again, what is happening inside the insect? If you give epicatechin 3 gallate, so it decreases the lipogenesis and increases the lipolysis. For that reason, the transition phases are being delayed, or they become deformed, or larvae become very weak to produce pupa. So these are the things happening in these secondary metabolites. So a secondary metabolite may be preferred by one insect. It may be repelling some other insect. So a negative correlation between the total isoflavone level and the protein level has been reported in seed, and that's the reason when we breeder go for the increasing in some quality character, then definitely there will be some penalty in the secondary metabolites. Another example, if you increase the cinnamic acid by blocking the pathway from cinnamic acid to comaric acid, the cinnamic acid is diverted towards benzoic acid and salicylic acid, then this induces the artemisinin biosynthesis. Artemisinin is a sesquiterpenoids, and this is a phenylpropanoid group. This is an example of how a pathway influences other pathway. And cinnamic acid, when it is less, then the production of lignin is less. For that reason, the phloem and xylem are defective, and the plant become lanky. And for that reason, it is being infected by jacids and many fungus and bacteria which are not earlier the plant was not earlier the host to them. For that reason, we actually overexpress PAL gene. So we didn't block this lignin biosynthetic pathway, but we overexpress the cinnamic acid biosynthesis. For that reason, the lignin biosynthesis went on, and benzoic acid biosynthesis also went on. So we got the increase in artemisinin as well the plant become tolerant to many insect as well as the fungi and bacteria. This is another gene, viridifloral biosynthesis. This viridifloral biosynthesis actually, if you increase, this is in mentha, if you increase its biosynthesis, then there is inhibitory to insect, the insecticidal in nature that we have proved. Now the paper is under review. Abiotic stress, then allantoin gives stress resistance. So that is also being proved. So we have a genetic resources, genomic resources we have made, and from there we are isolating genes. There is a monoterpenoid genes, and we will be testing one after another how they are giving response. Then this is sesquiterpene genes. These genes are already being cloned. And finally, understanding the pathway vis-a-vis stress tolerance, understanding synteny vis-a-vis similarity of resistance mechanism, understanding metabolic channeling and metabolic engineering, and it will ultimately lead to metabolic engineering for integrated stress response, maybe climate resilience in future. So this is one example of vetiver. See the number of molecules it produces in its root. Believe me, this vetiver is tolerant to many insect pests as well as tolerant to almost all abiotic stresses like waterlogging, water stress, then salt stress, cold stress, many things. So can I ask a question whether the secondary metabolites are responsible for this? And this actually new line of thought, it is going on. We are correlating the secondary metabolites and internal immunity of the plants, and it may be a new branch of science in the future. Maybe who knows? It will give us many clues to fight against climate resilience. Thank you very much for giving me the opportunity to speak in this podium. I thank Das, who was after me to give this talk. I'm not able to come there. So thank you very much. Jai Hind, Jai Bharat. Namaskar.
Yeah, thank you, Dr. Sasani. It's a great lecture. You know, you have many questions to ask, but you cover so many things. Difficult to ask, many things to you. And okay, Dr. Linkar is here. He can answer question to you.
Vaishnav sir. Vaishnav sir.
Dr. Seshan, it was a very nice lecture. Thank you very much. This is Professor Om Prakash Dhankar from the University of Massachusetts Amherst, and we had a very good time in Nabi. You remember that?
Yeah. Yes, sir. Yes, sir. Dhankar sir.
And we don't forget that motorcycle. The photographs, yeah.
Yes.
So my question is, because you are dealing with the secondary metabolites and the flavonoid pathways, these are highly complex pathways and multigenic kind of those, maybe 20 genes or 15 genes involved in each pathway. So changing one or two genes, do you think that can make a outcome in the of the flavonoid increase or decrease, and the ultimate impact on the plant's tolerance? So that because that's even the terpenes, it's a very complex pathway, and changing one of the gene may not be effective enough.
Thank you, sir. Thank you, sir. But there are the pathways are very complex as well as very simple also. So there are single-step terpene biosynthesis many a time, and there are also multi-step terpene biosynthesis like Taxol. This is diterpene. So when the complexity increases, then it has a multiple steps. But ultimately, when we want, and believe me, sir, Taxol is only plant which produces Taxus is only plant which produces Taxol. Similarly, Artemisia annua is the only plant produces artemisinin. So like these specific plants are there, but there are some different metabolites which are produced across the means you can taxa. But when the specific environment the plant is growing, so we need to find out the specific metabolites which are actually giving tolerant or resistance to that particular plant, either for abiotic stress or biotic stress. Once we know that, it is very easy to engineer either the last steps or some of the transcription factor which are controlling the pathway. We cannot go for the whole complex biosynthetic pathway as you told. I agree with you. Thank you, sir.
Yes, Dr. Sasni, I am Vaishnav Tripathi here again. So nice of you hearing from you. Yes, but as Dr. Sasni has said, we plants produce secondary metabolites, not for us, but for themselves. And they are doing it so to protect them from different kind of stresses, including abiotic and biotic stresses. One of the reasons could be because if you give them abiotic stresses, they produce more secondary metabolites. So it is a stress-induced response also. So with these words, we thank Dr. Sasni for getting great lecture. Thank you very much, Dr. Sasni.
Sure.
Missing you all, sir. Missing you. Missing also Dhankar, sir. Namaskar. Thank you, sir. I leave. I take
Yes.
your leave, sir.
Okay. Thank you. Thank you. Bye.
Okay. Bye.
Thank you so much, sir. So that was all for the session before the lunch. Now we will proceed for the lunch. We will resume the session sharp at 12:15. Thank you so much, everybody. Please proceed for the lunch. 12:15. 2:15, sorry, 2:15. Yeah, I'm very sorry. 2:15. So we just have half an hour for this. Please wrap it up soon and be here back on time. Thank you.
ସେଟା କାଟିକିରି ଏପଟକୁ ଆଣ ସନ୍ଧ୍ୟା ହେଇଯିବ। କେମିତି ଗୋଟେ ଚେୟାର ଫେୟାର ଗୋଟେ ପବନଟେ ଦବ ଏଠୁ ଗୋଟେ ପବନଟେ ଛାଡ ଆଗରେ ଆଗରେ କହିଲେ ନାଇଁ ଷ୍ଟେଜ ଉପରେ ପକେଇବେ ନା ତଳକୁ ପକେଇବେ ଗୋଟେ ଥର କହିଲେ ତୁ ଶୁଣିବୁ ସାଙ୍ଗେ ସାଙ୍ଗେ ଦେଇକି ସିଗ୍ନାଲ ଚେକ୍ କରିବ ବନ୍ଦ କରିକି ଦୌଡ଼ିବ ସବୁ ଖାଇବାକୁ ତମର ପଇସା ନବ କୋଉ ଜାଗାରେ ଶୋଇବ ଆଉ ଲୋକ କେମିତି ସବୁ ଗୁଣ୍ଡ ହେଇଯିବ ତୁ ଛାଡ଼ିଦେ ଦୌଡୁ କପାଟ ତାକୁ ଦୌଡୁ ବ୍ୟାଟେରୀଟା ପାଖରେ ଅଛି ତ ଏଇ ବ୍ୟାଟେରୀଟା ପାଖରେ ଅଛି ତ ଅଛି ହଁ ତାପରେ ପାୱାର ଲଗେଇବୁ ବୋଧେ କହିଛି
Hello, test.
Check.
Is it fine? Hello, good afternoon. Is it okay to share the slides or I have to wait?
Check. Good afternoon, sir. So we'll be having this next talk by Dr. P.V. Shivaprasad.
That's
He'll-
me. Yeah, I
Yes.
shared the slide. Can you see it?
Yes, we can see it, sir.
Okay.
You may start now.
Oh. Okay, slides moving?
Yes, sir.
Okay. Should I wait or I start? When I should start?
You can start, sir. You are audible and your slides are also visible.
Okay, good afternoon, everyone. Thank you for the introduction, Dr. Das. I'm sorry I'm unable to make it there, but I'm very much here, so you can ask me anything. If you don't get the time to ask me questions during this talk, please also feel free to write to me in this email address. So I'm going to talk about a new histone variant, so in H4 and what it does. So in my lab in NCBS, which is under Tata Institute of Fundamental Research, we work on all these model systems except Arabidopsis, and all these model systems were new to us when I started my lab 10 years ago. So the central to everything, so we had many fantastic talks in the morning, is DNA. This is the book of recipe. And as we know, though the book of recipe is big, it can code for many things to make many different proteins. At a given point of time, it will only make you a small subset of them. The reason is that many of those are not really making are getting copied, or copied RNA is degraded so that protein is not made. So both these steps at where there is checkpoint from DNA to make RNA or RNA to make protein. So these are regulated by a specific set of regulators called small RNAs. So these are in fact bits of the RNAs, but they can go and degrade other copies of RNA, or in plants very specifically, they can go and interact with the DNA itself and do some small chemical modifications in DNA and histones, and this we term as epigenetics. So the field is very interesting. One small experiment gave Nobel Prize to Fire and Mello long back in 2006. So in our lab, we try to do try to understand different aspects of this pathway. So here the pathway starts from RNA. RNA has to get converted into double-stranded RNA or a double-stranded RNA-like structure, which will make small RNAs. Small RNAs get into organoid. So organoid typically goes and targets RNA into 2 bits. So this is called RNA post-transcriptional RNA silencing. But we work with this pathway because of this branch. Certain small RNAs, slightly longer small RNAs with very specific organoid proteins can go and do epigenetic changes. So that is changes in DNA methylation. They can incorporate one cytosine methylation, which in the right place in the genome can already play a crucial role in regulation, or they can go and influence histone modifications. So the small RNA induced DNA methylation is very specific to plants, and it is called RNA directed DNA methylation, whereas histone modifications was also recorded in Saccharomyces pumbae. So this pathway is very interesting because no matter what happens to the plants, the message is now going to the DNA, and these marks can get inherited in the subsequent generation because all the proteins involved in reading, writing, and propagating, they are expressed during gametogenesis and embryogenesis. This is so unlike animals. That's why many plants can pick up a trait during their lifetime and pass it to the next generation, not always faithfully, but almost faithfully. And these are some of the major traits that we use. These are the ones which influence phenotypes that we all are really fond of. These are also part of our food, so that's why it's important. So last 10 years, we have done several interesting studies, whether it is on small RNA biogenesis or what small RNAs can do, or more recently on how these epigenetic changes take place, who are the players, what are the molecular features of these rules to make these epigenetic changes, and we also work on certain specific stresses. So the work I'm going to talk about is Sundar et al. It is a bioarchive report published or put up last year. So the papers in red are some of the significant studies that we have carried out. So along with this, it also matters what else we did. We have 6 theses, few patents filed, some poster awards, and most importantly, at least 80 different researchers trained. Now, this is how nuclei looks in across plants, across organisms. If you see, I mentioned that most of the DNA is not really read by to make RNA, copied RNA, and that's because the ones which are not copied, they are in a region which is very dense with nucleosomes, and this is called heterochromatin, whereas the loosely packed region is called euchromatin. So there are very specific chemical marks which can designate whether the region should be heterochromatin or euchromatin. And within this heterochromatic region, there is what is called as a constitutive heterochromatin. Imagine that this region contains recipe that will never be read. Whereas facultative heterochromatin, which will be decorated with histone 3, lysine K27 methylation marks, these are sort of recipes which you use once in a while, like when a guest comes. So this can switch back. They can be made or they need not be made. In a nucleus, both these heterochromatic and euchromatic regions, they can coexist. And this is constitute the highest order of regulation in all organisms. And these are regulated by these things which I put up here. Histone modifications, I'm going to give you more details in the next slide. DNA methylation, I mentioned, it is single cytosine methylation is already good enough to influence this trait. Small RNAs in plants, small RNAs regulate both these. But above all, there are histone variants which can take care of all 3 layers of regulation, and they can influence all at once. So I'm going to talk about one such histone variant. Before going there, I want to give a bit more details about epigenetic modifications. So everybody knows DNA wraps around histone octamers. This will be 8 bits of proteins. That will be H2A, H2B, H3, and H4. And DNA is wrapped around roughly 147 base pairs around this. What is important to note here is that N-terminal tail of this protein, they can undergo small chemical modifications. So these are called histone modifications. Depending on the residue, depending on the chemical modification, these can either contribute to heterochromatic regions or euchromatic regions. So on top of this, especially in plants, we have DNA methylation, which is nothing but single methyl CH3 group in the right place. So you get 5-methyl cytosine. One single methylation is already good enough to repress a gene expression. Now, the reason why this is histone modifications play such a crucial role is that these tails in the histones, which are just outside this octamer, they are outside, they are amenable for writing, reading, and erasing by different set of enzymes. And these residues in specific regions in different histones, they are outside, they change, and if they are modified, let's say lysine 9 methylation is a strong repressive mark. In H4, if you have lysine at 5, it is an active mark. It also, one has to remember that methylation per se is not really a repressive mark. It can also be an active mark at certain classes, such as H3 in lysine 4 position. So these are the ones, as I mentioned, they are involved in literally every aspects of growth and development. Now, because these histones are very important, several copies of histones exist in each plant or each organism. And because of their importance, certain paralogs of these variants, these histones have evolved across eukaryotes. And these do not bind everywhere. These decorate very specific regions in the chromosome, as shown in this picture. For example, SEN3 is only very specific to centromere, but not anywhere else. And such variants are very well known in H2A, H2B, and H3, but not in H4. And when these variants are present, in the very active tails, which I showed in my previous slide, they can have different amino acids so that they will first They need not have the specific modification, or they can have a very specific amino acid which is different from canonical to go and make a completely different chemical modifications. So when these variants bind, some of them are shown here, in different part of the chromosome, they bind to very specific regions. So you can note here A to a Z, for example, in the regions which are transcribed, which are upregulated under specific stresses, they have such very nice peaks in the next to the ATG. And if somebody watches 1,000 genes in genome browser, they'll all look almost like this. That's how precise histone variants have very specific regions to bind and regulate in the genomes. Now, how do these variants work? So we know quite a bit, mostly from animal biology. If instead of a canonical nucleosome, let's say you have now the histone variant containing nucleosome, this position can alter. So this is like making makan, or you can have DNA flexibility. The DNA bound to nucleosome can be little flexible, or simply the nucleosome is not going to be bound, DNA is free. So together, these constitute different layers of genome regulation. So several aspects of this is worked out very nicely by Kurmizaka, who is in Japan. Now, when we work on different layers of epigenetics, and we came across a new variant of histone H4. I mentioned that histone H4, nobody has identified a variant so far. So this is the canonical H4 in rice. So it has these folds. This is where it can bind to DNA. This is the tail, and all the modifications are shown in this circle. If you note carefully, in the variant, in the same residues where you expect variations, where expect post-translation modification, you have different residues. If we look even deeply, most of the variations in this variant are present in that first N-terminal bit only, almost 50%. Histones themselves are only around 100 plus amino acids long, 30 amino acids different. That's why if you compare all the histone 4 variants across plants and animals, only in rice or Oryza genera, you have one variant which has 30 amino acids different. So it's only 72% conserved. And rice has 11 copies, only one copy is different. And to see whether it's an authentic histone variant, you fuse this promoter with the GFP and saw where it goes. It went to the nucleus. When we looked into the expression pattern of this histone variant, canonical expressed everywhere ubiquitously, whereas histone variant was predominantly expressed in seedlings and very specific reproductive structures such as embryo, endosperm, and seeds. Now the variant sequence is very well conserved across 3,000 Oryza accessions as well as other species of Oryza. Now, because this tail is so different, we could generate very specific antibody against the variant, which will distinguish variant as well as canonical. When you try to make an antibody and try to see where this protein localizes in the nucleus, so please see this. This is nucleus, and in the middle you have nucleolus, where there will be pericentromeric regions of the chromosome. Most of the variant bound there, as well as in some pockets in the genic euchromatic rich regions. Between certain species of rice, we could see certain differences. I'm not going to dwell too much into it. Then we try to see what kind of DNA this histone variant decorates in the genome. We did a IP experiment, that is simply chop the DNA, see where this variant goes and binds. And we saw it can ghostly bind to distal intergenic region, that means away from the genes. 1/3 of them were also decorated to the genes. The peaks where this histone variant can bind is very sharp, almost similar to certain histone variants known from animals. There were about at least 3,000 different regions in the genome where this variant could bind. Among the histone modifications we saw, wherever this can bind, it overlapped nicely with the repressive mark H3K9 mark. Now to study this further, we reconstituted sort of first time a histone canonical as well as histone variant containing nucleosome core particles. We expressed each of these histones and generated tetramer and dimers of these. And with the serial dialysis, we got the octamer. Then we provided the common DNA called VDOM DNA to wrap around this DNA and got, again, after gradient analysis, we got canonical as well as histone variant containing nucleosome core particles. So these are the gels to show how it was done. It took almost 2 years, but the results were fantastic because for the first time we could show that unlike human nucleosome core particle, plant ones are slightly bigger. So this was sort of expected because plants have 2A and 2B bit longer than animals. Now with these structures in NCPs in hand, we got the first cryo-EM structures for plants. What is shown here in the gray is DNA. What's inside are histone proteins. So on the top, you have the canonical. On the bottom, we have the histone variant containing nucleosome core particle. So what you might notice is in the side view, in the canonical, there is these regions between H3 and H4 are not well resolved. So it's not very compact. Whereas in the variant, the DNA is really nicely wrapped around and H3H4 is nicely resolved. So we went and identified one specific amino acid that is valine at 34%, which could sort of give rise to this change in the nucleosome structure itself. So to see what does this mean at the biochemical level, we got the nucleosome core particles treated with the MNase, which will chop off the DNA and try to sequence these bits of DNA, which is bound on either side. So let's say here and here, and try to see what kind of variation one can see between canonical NCPs and variant containing NCPs. What you can see here is that the variant containing nucleosomes, they can bind longer DNA, much more significantly compared to canonical, which you can see very well in this NGS sequencing experiment, where variant minus H4 containing difference in the coverage of this DNA bound region shows on either side where you have entry and exit site of DNA, you get to see more DNA bound by variant compared to the canonical. So when you look at the surface charge, which is shown with blue color in the nucleosomes, we also compared to human, you can see much more positive charge in variant. That means DNA can also bind better here in this part of the nucleosome. Now, what does this again mean? So what we observed when we were purifying was that in SEC columns, if you simply look at the density or size of these nucleosomes, variant was actually smaller. It was elating faster compared to canonical, which you can see very nicely here. So the variant is much more shorter, more compact than the canonical. To see if these properties matter, especially in internucleosomal interactions, we took the tails of variant as well as canonical histones, biotinylated them, used them as bait, and did a peptide pull down assay. What we can see is that unlike canonical, variant did not really bind nicely to the NCPs, nucleosome core particles, but it bound very nicely to the octamers. So in one of the variant where some biochemistry has been done nicely, that is called H2AZ, what people know is that H2AZ is one of those magic histone variants which mediates heat stress tolerance or heat stress response. The nucleosome has less thermal stability. So we try to see whether H4 variant can have similar property. So what we observed is that in the canonical one, the distability, so when we did this assay for dissociation, it got dissociated at 87 degree, whereas variant containing nucleosomes were unstable at 82 degree. So significant 5-degree difference. If you make a mutant in that valine 34%, which I mentioned, revert back to like canonical, 50% of the thermostability could be recovered. So many of the things which I mentioned, whether especially stable or unstable nucleosomes, these are the ones which tell chromatin remodelers or even histone modifiers to come and do their activity. So these are like beacons for in the genome. Now, when you have a nucleosome, it can occur in two different ways. One, in the octamer, both variant and variant can be in 2 copies. That is called homotypic. Or one of the copy H4 is canonical and another can be variant. So to see which of these is happening for H4 variant, so we fused H2A with the his tag and then serial dilution we did. We provided each of either variant or canonical and try to see how this gets incorporated. What we see is that there is more of almost always homotypic nucleosomes are formed when you have histone 4 variant. Okay, so what does this how does this matter? So we generated CRISPR knockout of this H4 variant, and we have a very nice setup for variant for developing knockouts. So here we went out all the way to T4. We removed the CRISPR containing cassette. So there is only editing as shown here. And in all our transgenics, we do junction fragment sub-analysis to clearly ensure that we have the right transgenes. When we looked into the transgenes, we got two very specific phenotypes. First one is that plants are already sort of stressed. Their roots same as wild type, but shoot is much more reduced, which you can see nicely here. The second important phenotype we saw is that in the knockout, the seed filling was lesser. And when we developed 6 different type of transgenics, I'm not going to the details of it because it's not important here. We developed overexpression as well as chimeric versions of this histone and put under different promoter, native as well as constitutive promoter, and try to assay different phenotypes. So for this talk, it is probably I'm going to stick to this phenotype where we see sort of a stress-like phenotype in knockout plants. And when we performed transcriptome from these plants, and when we looked into the genes which are really changed, we got several transcription factors which one can easily associate with stress. When we looked at downregulated genes, for example, most were closely related to stress responses. We got a hint that it is salt stress by doing this experiment. So this is a PCA. Here we took all the differentially expressed genes in knockout compared to anything else anybody has put up in GEO and look for all the genes and the variation in their expression. When we did that, you can see H4 knockout transcriptome matched almost completely with salt stress transcriptome. So we did this other way around. We took the DGs upon salt stress, put all the data sets, including the knockout, and we saw that's where it is closer. Basically, the plants are almost behaving as if there is under salt stress, which we can again see here. Even without salt stress, the same genes which will go up in wild type are the ones already up in knockout plants. So to check this further, we did this scatter plot. So here the regression curve is here. We are comparing DGs in knockout with DGs that come up during salt stress. There is almost a perfect correlation. There are also 5% genes which behave differently. We know why they are different, but predominantly they all matching almost with each other. Now, if you take the plants and put these plants under stress, now the knockout plants did not do anything worse because they are already behaving as if they are under salt stress. So what exactly is happening? So we try to see the H4 occupancy in the genome. What we observed is that upon salt stress, there is much more localization to the nucleus, which you can also see now some of the spots in the pericentromeric region, they are coming to gene-rich regions in the nucleus. The peaks became many more, so which you can see here, more peaks upon salt stress. Where it is binding, now the bonds regions became little more wider than they were without salt stress. So among the regions which really changed upon salt stress, we try to see which other marks it coincides with. Of course, it matched nicely, all those 3000 peaks matched nicely with the K9, which we already saw previously. And when we, even after looking very carefully, we did not find any specific things changing there. But among all other marks, including A2AZ, which is shown to be important for several stresses, we found none of them are matching except H4K5AC, which is a it's an active mark for several different stresses in plants. So this is where most of those peaks mapped. And again, if you look at the K5 acetylation signal in knockout plants, there is hardly any signal, indicating that for this active mark to come, you somehow need H4 variant. So I'll skip this in the interest of time. So now we did a 3-way comparison. We took all the H4K5AC peaks lost in knockout. looked at the same again differential gene expression in knockout and again in salt stress. We again got somewhat quite a good correlation. One set of the genes very nicely matched. That means if you have to have K5AC marks, you need now variant to be there, and there's the genes which change during salt stress. So what we see is that without salt stress, there is nice decoration in chromatin of H4 variant, and this changes when there is salt stress. Now this H4 variant acts as like a gatekeeper, because wherever it is present, now canonical with the active mark will replace. So these nucleosomes come preloaded. That's one specificity of H4 K5 acetylation mark. Now, because now it can quickly change this, there will be gene expression to tackle the salt stress. So we also put these plants back to wild type, and we saw now loss of again H4 variant in these regions. So why this is matters, why it is important? So rice is one of those plants which have to deal with the high and low water every single day, whether it's even sometimes within a week, maybe there will be too much water, maybe sometimes there will be too little water. So there's a constant change in the salt adaptivity. And where does this fit into the classical salt stress response in plants? So obviously one has to sense this signal. There will be all kinds of immediate changes, and somebody has to transfer this knowledge to the signal to the nucleus, so that all these well-known transcription factors, some of them were mentioned in the talk today, need to come up and do something. And that's exactly where you have the variant. This is the one which tells which of these genes should go and which one not. So I would like to thank the student. So his name is Vivek Harisundar. He's a fantastic student. Actually, this is only one half of his PhD. So this is our lab. All the people who have completed their PhD, these are current lab members. And my source of inspiration are my two supervisor, PhD and postdoc. And so we did the cryo-EM in our in-house facility, but we somehow we didn't have the right carbon grid. We had to do it to the Jayaprakash, JP. Most of the sequencing was done in-house, and my lab is supported with funds from all these agencies. Thank you for listening. Thank you for your attention. I'm very happy to answer any questions if you have.
Thank you very much for interesting lecture. Any questions, please? Okay, if there are no question, I just want to ask you why you are looking this knockout for salt stress. Why can't you look for water stress or any other stress?
Yeah, so I mentioned that we got the transcriptome among the genes that changed. So we did a PCA analysis. The only condition around 300 data sets we checked for rice stress done by anybody out there, it matched only with salt stress. So it is also very surprising, because people think salt, osmotic, and other stresses or drought have overlapping genes. Yes, they have, but it's only tens, but not thousands. So plants are so clever, for each of those conditions, they have very specific set of regulators. And if you look at the 1,000 scale, like 2,000 genes that are changing, salt stress is different from other stresses, and that's where H4 variant plays a role.
Okay, thank you very much. Okay.
Thank you.
Thank you so much, sir. Well, now we'll be having our sponsored session, which is sponsored by State Bank of India. The session theme is Biotic and Abiotic Stress and Crop Productivity. Chairman Dr. Sangamitra Samantrai, Head Corp Improvement Division, ICAR-NRRI, Cuttack, and rapporteur is Dr. Anil Kumar C, scientist. Same. From State Bank of India, our regional manager, Sri Cuttack, Sri Subhash Chandra Behra. Sir, can we please have you on stage?
Good afternoon, everybody. Myself, Subhash Chandra Behera, Regional Manager, State Bank of India, Cuttack region. First of all, wish you all a happy and prosperous New Year 2024. On behalf of SBI, we are here to present banking services by SBI for sustainable agriculture. First of all, we are thankful to each and every one of you, as well as this international level institute ICAR for giving us an opportunity to be a sponsor for this great program. SBI, 1/3 population of India are banking with us. More than 47 crore people's accounts they are maintaining with State Bank of India. We are not only confined to city or urban areas, we are having presence at the remote place of our country. We are dealing with the small KCC loan to financing the industrial like Ambani and Adani. So it is a great thing. It is an opportunity for us to showcase our presence for the sustainable agriculture. And as we are the nationalized bank, our prime objective is to help the poorest of the poor of the society. If you see our State Bank logo, it suggests that there is a hole in that. That means a small, it is a keyhole. That means every customer, be it poor, be it a rich person, everybody is having a opportunity to get the service from us. In Odisha, we are the pioneer in agriculture as well as allied activities financing. We have been financed by Honorable Chief Minister for the best bank in SHG financing, for the best bank in MSME financing. In Odisha, we are having more than 890 branches. And if we see all over India, we are having more than 22,000 branches from Kashmir to Kanyakumari, be it in the top Himalaya or be it at the Kanyakumari. Everywhere our presence is there. We are helping the development of the government in agriculture as well as various activities. This year, we have been as a state, Odisha, we have been given a target of 3 lakh crore agriculture finance, and we are going to achieve it. Major source of agriculture, our agriculture finances, we are financing for direct agriculture finance as well as indirect. Direct finance, we are doing KCC, the farmers for whom myself, you all are present here because they are providing us the food. That's why our late Prime Minister Lal Bahadur Shastri once told, Jai Jawan, Jai Kisan. Without Jawan and without Kisan, we cannot live happily in our country. So for the benefit of the Kisan, we are having several schemes like for cultivation of cereals or pulses, we are having scheme like Kisan Credit Card. We are having scheme for allied activities. We are a scheme like Agri Infra, Agri Enterprising Loan. So from 10,000 to we are financing up to 100 crore in agriculture sector. And for the development and upliftment, because in every family, the family run by our mother, our sister, for their development, not only SBI, but our government in Odisha, they are taking care of by providing SHG loan. They are providing interest subvention for agriculture as well as interest subvention for SHG loan. So we are pioneer, State Bank is pioneer in financing SHG people, and also to provide the service, uninterrupted service to agriculture sector. We are having so many intermediaries also, so many helping hand, like besides our bank employees, we have recruited feet on street because poor customer, he may not able to come to the bank because if any day he come to bank, he may lose his one-day wage. For that, our bank has recruited FOS, that means feet on street. They will go to the village, they will collect the application from the farmer, they will process the loan and disburse the loan at the village itself. The main motto for selection of FOS and providing the service at the doorstep is that our poor farmer who is providing food to everybody, they will not be suffered for getting a loan. Similarly, we are also doing loan, indirect finance. We are having like Annapurna Microfinance. So many NBFCs are there to whom we are also financing to them. And in that way also, we are achieving our, because every bank, they are having a budget to achieve percentage of their advance. They have to achieve their budget in agriculture sector. And that way we are achieving our agriculture budget. And in detail, I request our agriculture officer, FSTO, Mr. Surya, in detail, he will describe what are the schemes in our bank. It is available for the sustainable agriculture. Surya, please.
Thank you very much, sir. I am Surya Kandas, representing State Bank of India for agriculture segment financing.
Oh, thank you.
Thank you. Anything in research field in science laboratory, if we want to take it in the farmers field level, we need finance. If you want to scale up or disseminate this technology into the farmers field, we need some finance. For finance point of view, bank word comes, bank help is required. And among the banks, State Bank as a pioneer in agricultural finance, the name comes first, State Bank of India. As sir told, sir almost covered how SBI is involved in agri financing, and particularly the more focus on sustainable agricultural development. So SBI name, what sir told, involvement of SBI in the periphery for sustainable agriculture. For better understanding what sir told, I will show some pictorial form how SBI is involved in a rural pocket, starting from the urban area to rural pocket in Odisha. Particularly, whatever the pictorial form I will show, those will belong to the Odisha, this state Odisha. So, as sir told, starting from the Kanyakumari to Leh Ladakh, so our presence is there. The year 2,100 ATMs in Odisha, 62,000 ATMs, State Bank ATMs. Wherever you go, you can find the ATM. And one ATM is just outside of this boundary. So, these are the rural branches, regional branches. These are the State Bank's total outfit in different form. So services to strengthen financially in rural farmers, rural household, we have some financial product like insurance, low cost insurance product, Pradhan Mantri. It is mostly government of India promoted insurance scheme. Besides that, State Bank has also its own insurance scheme. So Pradhan Mantri Jivan Jyoti, Pradhan Mantri Suraksha Yojana, most of us are aware about this. And I also appeal in this forum, in this platform, if many students are here, if they do not cover their life insurance with low cost, please cover these. These are very beneficial. To 20 rupees to 440. One product premium is 20 rupees. Another product's premium is 400 around. So per annum. So you can cover 4 lakh insurance policy. So in State Bank, it's own PI, personal accident insurance scheme. You can cover by paying a premium of 100 rupees to 1000. There are 5 variants, 100, 200, 500 and 1000. By paying these 4 type of variant premium, you can cover 2 lakh to 20 lakhs per annum insurance policy. It's very low cost. Pension Yojana is also there, Atla Pension Yojana. The main focus of this product is to cover, to strengthen financial support to rural household in rural pockets. State Bank, not only banking, it goes beyond the banking, like training. It's also giving training to skill development in rural household, rural farmers, giving different training on different activities like livelihood support training, goat farming, fishery, handicraft, handloom, machinery, beauty parlor. If any group of they are preparing, SAG are being promoted by different level, different user groups, different joint liability groups. So those groups can be come to the our RCT, Rural Self-Employment Training Institute. It is being maintained by different banks. We have around 19 RCT in Odisha. State Bank is maintaining those RCT. So we are training every year, every month. We are give training to most of the groups for their livelihood. And once they trained by that particular activity, we also give financial support how they can go scale up their income from that activity. So like SAG, it is very common in Odisha and rural pocket of India. Through SAG, we are financing particularly agri and agri-allied activity. Most of the SAG are doing, and we are financing almost all aspects of agriculture through SAG. It is a very simple, very common and renowned product, renowned financing way of in rural India. So this pictorial form, as I told, how we are reaching in outreach area. This photo can give you a sense of thought how SBI is going beyond the difficulty remote area and providing the service. These photos are very popular. In the middle photo, who is sitting, that our branch manager is sitting on this mule. So that shows how dedicated our field officers, State Bank is dedicated for the commitment of rural development, sustainable agriculture. So these are the public gathering in villages when State Bank is conducting giving any awareness. So these are the gathering in village area. So coming to the transformation in agriculture field, earlier 20 years back, 10 years back, we normally used to harvest the field. First picture, top of the left, first picture shows how we are using to harvest the field. There were a lot of drudgery for harvesting the field crop. So many labor were required to involve drudgery, scarcity of labor. The transformation has been happened. So from manual harvesting to machinery harvesting. Starting from the cow, dairy farming. So the first photo shows how we were doing dairy and present dairy. There is a huge difference. We are also doing the dairy like Switzerland dairy. Irrigation, this pictorial form is also shows how irrigation, there is a transformation in irrigation. So by adopting these type of dairy, machinery, harvesting and irrigation, we require some finance. We require some fund to install in our field. For that, bank is supporting, bank is doing already financing, doing it also in future also in different form of irrigation or starting for any infrastructure will be required. Bank is ready to give a loan. These are the livelihood activity in rural area mostly. Government is also supporting through different schemes for doing these, promoting these agriculture activity. State government and central government through their different various schemes they are promoting. Like you can see here PMGP, the older model is PMGP. Through PMGP, they were promoting rural cottage industry or rural based industry in rural area. That is met by the PMGP. Then National Horticulture Board, National Horticulture Mission come. After that, Pradhan Mantri, PMFME and now Agri Infra Fund. Then Agri Infra Fund, Stand Up India. Those schemes come to promote agri infrastructure in agriculture and allied field. Agri Infra Fund is mainly focusing on how agriculture infrastructure can be promoted. Hassle-free financing can be done in this agri infra area. So Stand Up India mainly focus on allied activity. Agri infra fund is meant for the agri infrastructure, and Stand Up India scheme is mainly for focusing on allied activity, like Mudra, Pradhan Mantri Mudra Yojana for small scale agri and allied activity. Odisha government is also promoting giving financial assistance to this agri and allied area through Mukhyamantri Krishi Udyog Yojana, and Apical, the logo in the middle left is Apical. Apical is the implementing agency. So SBI also encourage and support in this type of activity, starting from the farm pond to machinery, poultry, dairy, goatery, and mushroom cultivation. And Odisha, this pocket of Odisha is famous for mushroom cultivation, and we have very good cluster in this particular surrounding of this belt, Cuttack and Puri. In Odisha, what is the amount of mushroom is being produced? Out of the 100, this pocket of this coastal belt of Odisha contributes around 70% to the total production. So we also, though the financing aspects, finance perspectives of individual activities is very less, but State Bank is very encouraged to promote cluster financing and cluster approach. So to give more focus on sustainable agriculture, changing environment of agriculture scenario, and we also develop some infrastructure as well as some products to give hassle-free financing to this activity. So mostly we developed some branch, we developed some outfit, our outfit to focus on this type of activity. Mostly banking sector means it is normal cash transactions, and most of the people may not be aware that State Bank has developed some specific branch like SCB, Agri Commercial Branch. Earlier we had in Odisha around 28 ADB, Agriculture Development Branch, to focus to finance more on agriculture activity. There were around, out of 200 around branches, 600 branches, we had only 28 agri development branch. Now bank has given more thrust on agri commercial branch. Now agriculture concept, agriculture definition has also changed from the agriculture development sustainability, from sustainable to how we can be a agri entrepreneur or agri commercial way we can go ahead. So like for this, we had most around 30-40 agriculture product. That was very difficult to understand the inside of the customer, our bank people also. To make it very simplified, we developed 3 products. One is Agri Enterprise Loan, AEL. That is particularly meant for the agriculture infrastructure financing. So up to this, in this product, up to we can go 100 crores finance. Starting from dairy to dairy byproduct processing till beverage item manufacturing processing, including wine and auxiliary fermentation food beverage item. So second, we developed a KSR, Kisan Samriddhi Rini. This is only meant for crop production loan, recurring cost for a crop. Is mostly high value. Suppose in polyhouse or high value colored capsicum or polyhouse financing, they required per acre or per 1000 square meter, it may require 5 lakhs rupees. For a small farmer, 5 lakh rupees is not small amount. They may go for the financial help. So bank has developed to cater their need, those type of farmers need. We have developed this case are hassle free. Most of the Odisha agriculture farmers, they do not have that much of amount of security in term of cash, in term of gold, in term of land. So by mortgaging, by giving as a security their agriculture land, we can finance them for this agriculture crop production. This product is only for crop production, high value crop production. So another product is a Pradhan Mantri Mudra Yojana. By this product, we can finance a 50,000 amount to 10 lakh. It is term loan, it is cash credit, it is for infrastructure, it is for cash, day-to-day expenditure of your activity, it is for agriculture, it is for allied. It is mostly allied, dairy, goatery, beekeeping. So through this product, we require some security. as I told earlier slide, agri infra and stand up, they will give a financial support. They will give us a guarantee, bank guarantee. You finance this activity up to this amount to this borrower. We are giving you guarantee. And that guarantee we are getting from these two products, agri infra and stand up India. So as sir told, feet on street word, you might have remembered that feet on street, to giving more focus for this type of activity, financing this type of activity. Bank has recruited, taken some skilled manpower. Those designation is feet on street FOS. Like business associate, the young students can be a business associate for us. You can be a business partner for State Bank of India. By enrolling as business associate, you can give some business lead. Those farmers are doing interested for doing such activity or interested for setting up this type of enterprise. So you can give the leads, or by giving the leads, you can get a commission. That is a business model. So that a bank does not have that much of a manpower, that much of a outreach. Still, we are having a pioneer in agri financing, a pioneer in branch outreach in different rural pocket. Still, we need some financial partner. So bank is also developing inside manpower, inside skilled staff as RMRU, giving a more focus on agri, linking to the agri finance in rural pocket. So bank has developed RMRU, relationship manager rural business. They will focus on only rural pocket agri or SME related activity. So we have developed some poster like this to give more awareness among farmers, like this type of platform. Banner poster, we are giving agri finance, agri enterprise loan. This is the poster of agri enterprise loan. The basic feature of financial feature of this product, those small letters are very small. So up to 100 crore, as I told, up to 100 crore, we can finance this product. So minimum margin is 25, farmers contribution or entrepreneur contribution is 25%. If you will go with 25%, you will 20% minimum margin is 20%. If you will give 20%, you will give 80%. Security, you can cover up to 5 crore as a collateral security. 2 crores. Now it has extended, recently it has extended up to 5 crore. You have to pay some amount of annual guarantee fee. That amount you have to bear as a customer, as an entrepreneur, you have to bear. Then guarantee will give by Government of India through Agri Infra Fund. Mostly Agri Infra Fund is infrastructure development. So what are the activity covered? It is a very indicative activity. Activities are to more than 200 numbers of activity, starting from the dairy, agri commodity, fishery, feed plant, biotechnology, veterinary vaccine manufacturing unit, storage, agri input manufacturing, and in our that biofuel, traditional biofuel, that cow dung cake. Cow dung cake, manufacturing of cow dung cake is also a eligible activity under this activity, Agri Infra Fund. You can go by that activity. So what are the benefits can be clubbed with this activity? As I told, you do not have any security, you can take finance up to 2 crore. Bank will give guarantee. So Agri Infra Fund, Government of India will give guarantee. We will give 80% loan, you will give 20% for the project, and that can be clubbed with another state financial assistance scheme. Like in Odisha, Mukhyamantri Krishi Udyog Yojana, they are giving 50% of capital investment as a subsidy. That can be clubbed. These are the beautiness of this product. You can take the various schemes benefit that can be clubbed under this product. This is bank's own product, agri enterprise loan. These are the basic features: margin money, collateral security. Interest rate is very competitive than other competitors in market. Agri Infra Fund already covered. Kisan Samriddhi loan for mostly for the recurring cost of high value crop cultivation. And digital bank is also adopted in most of the village. Every branch we have committed ki every branch will adopt one village as our village, and we will develop holistically in digital way. Bank has adopted this type of digital village, and we have committed to target to reach 3 lakhs crore, 3 trillion finance in agri segment. So by covering this, by achieving this, we have to touch every aspect of agri and allied finance. So we are that way we are committed. We will definitely help, bank will help every aspects of agriculture need. So in digital way also we are not far, we are much ahead. We are also very competitive in private bank also. Through NO app, you can download NO app. Through NO Krishi, you can take financial, you can register your application for getting KCC, farmer produce loan, or some other bank transaction facilities are also available in SBI NO. Thank you very much for listening us. If any queries, sir, please.
Thank you very much for the nice overview of the SBI financing in agriculture. First, I would like to compliment the SBI.
Thank you, sir,
for the doing this kind of providing the financing for the agriculture and agri infrastructure, because we all know that this next innovation in India after IT and all this is in agriculture.
Yes, sir.
Because agriculture is not going to be traditional agriculture. The farmers used to do that, like you showed the example of the irrigation and all those. Because next revolution in the farming is AI, artificial intelligence, the drip irrigation and kind of those, and protected cultivation. So those are really going to spur the activities of those startups and all this and revolutionize the agriculture. I compliment that.
Thank you, sir. Thank you very much.
Second thing is, I want to ask you a question. So, for example, if someone want to start a startup and in protected cultivation and want to have like several acres of high-end these greenhouses and all those for the all this the controlled temperature, controlled environment. So it's say the example is 5 crore rupees is the capital cost. So how much the bank will fund, and what are the collateral will be, and what interest will be, and then I have second question.
Thank you very much, sir, for your valuable. I have not touched those particular polyhouse related area in this slide. If it is very particular crop product specific, I'm touching. Through I presented a agri enterprise loan, as it is also covered under agri enterprise loan. So bank is very much flexible on financing those activity, because these are the new technology in future technology for the agricultural development. So we require to pay 20% as margin money.
20%.
5 into 2, 10. 1 crore you have as entrepreneur you will pay, and the rest 4 crore bank will finance.
But the National Horticulture Mission is also giving a 50 to 65% subsidies.
Yes, sir. For subsidy point of view, NHB or NHM is providing. So under polyhouse, it is almost 50%. 50% of a capital investment.
Yeah.
So that also can be capitalized into the project. But while financing a project, subsidy is not coming in front of.
It comes up after
it is mostly back-ended. So before though subsidy will come to your account, it will be parked subject to completion of a project. Once project will be completed, then only subsidy can be appropriated into the loan account.
And what
so before that, sir, for completing the project, we have to spend you have to spend money. For money, there is a two source. One is from your source, another is from bank source. Then only 100% project will stand up. After stand up, then we will receive that money. So there is a cushion. Ki we can go for bank and take a call. No, there is so much of a risk mitigated funds are available for this activity. We will go for this activity. So there is a cushion, there is a low risk, so that bank can take a positive call for financing.
Okay.
As that aspects. Another is collateral security. As I told, sir, 40% collateral is required. 40% means 5 into 4 to 2 crores. So 2 crores collateral in term of cash, in term of fixed deposit, in term of gold or LIC bond, or in term of Gharabadi surface compliant land or a land building you can give. But if it is individual, if this activity will be done by the group, SAG, JLG or group of FPO, FPC, then 2 crore up to 5 crore, there is a credit guarantee covered by CGTMSC. You need not to pay any collateral.
Okay.
Will finance, sir.
Okay. How many in the group? How many minimum farmers or participants you need? 10, 5?
Sorry, for SAG, 5 to 20.
Okay.
For JLG, it is 4, 5 to 10.
Okay.
For FPC, it is minimum 10. 10 to any number.
And what's the interest rate will be on those loans?
So if it is a group activity, if it is this activity polyhouse is done by the group, then there will never ask for the collateral security. We will cover through CGTMC cover. If it is individual activity, then you have to pay.
Okay. So what the interest rate will
So
be?
interest rate it will be around, it is case to case. After appraisal, thorough processing, that rating will come.
Okay.
Based upon the rating and some other 2-3 factors, then generally interest rate arrives. It is mostly 9.15 and 1%. So 10.15.
Got it. So my next question is, I'm sorry, but I have to ask him. So now the Pradhan Mantri Fasal Bima Yojana, Pradhan Mantri started that for the benefit of the farmers. Why it has, it's been 10 years already, since around 14 or 15, he started that. So why it's not so popular and why farmers are not adopting it? Because in my area, I originally belong from Haryana and I'm from a farming family. My family is still in farming. When you buy this Fasal Bima Yojana, the companies who are doing this one, they are delaying the payment for several years, sometimes 2 years, 3 years. They are not giving to the farmers on time. So how farmers can sustain that? Second thing is, the clause is as very difficult for individual farmers. You are eligible to get the benefit coverage if the 80% of the crop in the particular village is just destroyed by a particular event, like a hailstones or a rain or something. So if there is 80% of the village, and then you will be eligible, but you are charging on each individual per acre. Why not you cover them per acre basis? Why the 80%?
Sir, I appreciate your concern in this particular crop insurance scheme. But bank does not have any control of taking decision in this aspect. Neither bank nor insurance company. It is a government policy. Government has to declare once any calamity or any crop loss happens in particular village or particular. Mostly village is not a unit area for declaring the insurance point of view. So there is at least one panchayat. This is a government policy. Government has to take a call on that, how they will segregate the unit area to declaring crop loss area. So bank is only debiting the premium amount and paying the claim amount. Bank is the only a facilitator. Bank is a facilitator. In particular, in one district, one state also, different districts are allotted to different insurance companies in any agriculture crop insurance.
Ultimately, farmers suffer.
Yes, sir. But this is not.
All these policies, government is highlighting all of those. Why the Indian farmers are still so poor?
Yes, sir. I also agree with you, sir. I also agree. But this is a policy level decision. We have to take up on different forum, different level to address this particular concern. Thank you. Thank you very much, sir.
If there is no question, then we thank Mr. Sujagandas for giving a comprehensive information to the House, and sir raised many issues, and that should be taken care by Suryakandas, and it will be taken care by the government. Thank you, sir.
Thank you so much, ma'am. The next talk would be by Dr. Sachinanda Swai, ICAR-CIWA Bhubaneswar. His topic is supply chain analysis of fruits and vegetables in Odisha, case study in Kujang block, Jagat Singhpur. Is he here? Is he here? I guess no. So we'll proceed with the next talk. Dr. Sandeep Sehgal from SKUAST, Jammu. His topic is edible wild fruits of Northwestern Himalayas, India.
Good afternoon, everyone. So before the display comes on the screen, I would like to say how much fruits are important in our life. You see, everybody says you must eat 3 to 4 fruits of different colors every day, right? So imagine if a fruit is wild. So if it's a wild fruit, its properties and its benefits, I suppose they may be 2 or 3 times more than what we have the cultivated fruits. So before starting, I would say that edible wild fruits are the fruits which are not commercially grown. And you see, being very attractive, fruits have always been a part of our food system. And what we see is that edible wild fruits basically refer to the species which are not cultivated, and they sometimes do possess many other properties which a common man doesn't know. So in India, you know, people are very much close to nature, and our kitchen is supposed to be a storehouse of medicines. So in our homes, whenever we are ill, we have stomachache. So we have so many remedies already lying in the kitchen. And believe me, they are 100% efficient. And they work till now. Over the recent year, what we have seen that there is a drain in the kind of knowledge which we see regarding the traditional medicines and some other uses of wild fruits, wild vegetables, and all other things which were very easy and passed on over from one generation to another. So, keeping in view we came across so many reports that the traditional knowledge about the use of fruits is going down from one generation to another. Even my kids, if I take them to a jungle, they would not eat anything which they don't know. They would be scared of. And sometimes it's also true they can be turned poisonous also. So keeping this in view and keeping this the drain in the traditional knowledge about the wild fruits and other medicinal plants, we devised a study, and it's in the 3 districts of Jammu and Kashmir, basically which is the Himalayan region. This is the experimental site. And keeping in view the different altitudes, we try to cover almost 2 contrasting agroclimatic regions, say from mid hills to high hills in the Himalayan region. So these 3 districts, the Riyasi, Ramban, and Kishtwar, they all lie along the river Chenab, which is flowing through, and it's also known as the Chenab Valley. But you will see from this slide that there is a difference in the altitude. So at lower altitudes, we'll be having more of the subtropical fruits and subtropical plants. As we go higher in the Kishtwar district, we'll have some temperate type of vegetation. I'll just run through this. This is our methodology, that how we selected the number of respondents. And in all, 5 respondents were selected from each village randomly, And overall, our study had 260 respondents. And we had one condition. All the respondents were over the age of 50 years. So in order to stay very clear about the traditional knowledge and the use, we deliberately selected people with more than 50 years of age. And the data was obtained through questionnaire. And this is some of the statistical tools which we applied to know that what are the relative frequency of citation, what is the importance of the family to which these fruits belong, and what is the use value. So out of 260 respondents, how many would specify a particular species that will result in its higher use value? So these are the results of our study. So mostly the people were of middle education up to middle level, and mostly they did agriculture. And these are the wild fruits in the study area. The names of these fruits, edible wild fruits, are in the local language of that place only, that 3 districts only. Maybe in Hindi, we may be calling them by some other names. But these are, some of the names may be common also. So this is the list of almost all the edible wild fruits reported by the people of that area. Now this is Fragaria nubicola. Now it is also known as the Himalayan cherry. I'm showing some of the photographs of these edible wild fruits. This also depicts that what kind of stature there was, that were trees or herbs or shrubs. Now this is Punica granatum, and if I'll take its common name, most of you will be knowing that it is anardana, right? So it is a very common fruit, wild fruit of that area, which is commercialized, and it sells like anything. Cydonia oblonga, commonly known as bay. It's a very tasteless fruit. It has no taste. It's a cousin of apple, but it is said to possess many health benefits by the local people. Then this is Xanthoxylum armatum, and commonly we call it timru. And you know, the fruits of this tree, it's not a big tree, but a medium-sized bushy tree. The fruits of this tree possess some very nice property which are digestive in nature. And it is also used, the stem of this tree is also used to relieve the toothache. And the stem of this tree, it is also used to prepare chutneys because it is believed that the stem has a flavor which it imparts to the whatever chutney when you grind it. So some of that flows into the chutney, and you will get a very different flavor from that. It is carbonative and digestive in nature. Then this you must also be knowing, Terminaria chabula. It is harad. Now harad is a part of a very important formulation, Ayurvedic formulation, that is Triphala. Harad, Bheda, and Amla. Amla is commercial, so everybody knows that. But these are not so much commercial. This is Bheda. And these are used in different proportions to make Triphala. Then this is Viburnum grandiflorum. This is also a wild edible fruit. Then this is Diospyros lotus. This is also a wild edible fruit, and it is also given in the prasad of the Mata Vaishno Devi shrine. Basically, it has what you call a very good digestive, as well as it induces sleep. Nowadays, we have a lot of sleep disorders. So the skin of this fruit, it is sweet in nature, and studies have shown that it helps in inducing sleep and relieving insomnia. Now, this is the growth habit. Mostly we had trees, followed by shrubs, herb, and climber. Now, I have listed the botanical, ethnomedicinal use of these edible wild fruits, all of them. This also depicts the plant part used, the mode of consumption, what is the application, and then we have what we got from the statistical analysis of that. So one more thing is, it's very important to note that most of the medicines, even the synthetic medicines which we are using today, they have been, they had their origin in the trees. For example, Cinchona officinalis for chloroquine, and so many other drugs also. So the importance of this study is that the results, the uses which the people are mentioning, that can be further researched to find out, to isolate new drugs for that. These are the plant part used. Most of it was fruit. I think traditional people, ripe fruit is naturally justified that it is very easy. You don't need anything to add to it or to prepare some decoction from it. These are results of some statistical analysis. So mostly Amblyca officinalis or Amla was cited by most of the people from the respondents. And as far as the use value is concerned, Ficus carica, fig, a wild kind of fig, that was reported maximum by the those participants of the study. Rosaceae was the highest cited family. Most of the plants belong to this family. So these are the conclusions of this study. Around 32 edible wild fruits were consumed by the people of the area, which were being used to treat so many diseases as listed above. And mostly they were trees. The other two uses things I have told you. And what we need further is that some of these things can also be commercialized. Another thing is conservation, that we need to see that which of the plants are in threatened zone, which are widely available, and what can be done to conserve them. Thank you so much. Any questions? I'm here.
Very nice presentation.
Thank you.
My question to you is that these fruit species which you have mentioned, was it primarily based on a survey, or do you have access to those fruits or the plant material?
No, we went to those areas. We also brought the samples with us, and then we conserved those samples. We confirmed the species, scientific name, the local name, with the many botanists from the University of Jammu. And then using that, we finalized and we properly identified the species. We do have a herbarium of these all species in our department.
So the next question is, have you done any nutrient analysis or the analysis of the samples, biochemical or the actual wet chemistry data on the components of the nutrient? No. Can you do it? Do you have access? Can you do it if it was possible?
some of the people from the biochemistry department, they are in touch with us.
Okay.
So we don't actively do that. They do that, but we provide them the plant samples, the identifications, and the list of traditional uses.
Yeah, that's great. I think I was just thinking if you have the sample and if you can request any of the biochemical or other organizations to help with the complete profiling of the nutrients in the effective parts, then we can actually say what is the chemical which is contributing to those health benefits, and then that will lead to the commercialization if possible. Great, thank you.
Yes, sir. Some of the plants which I have listed in that, these are also being analyzed by the Indian Institute of Integrative Medicine. That is also situated in Jammu. Earlier, it was known as Regional Research Laboratory. So the biochemical analysis and development of new drugs, that part is being taken by the Indian Institute of Integrative Medicine, Jammu, run by the CSIR. Okay. Okay. Right. I got it. So thank you very much.
There is no question. I want to ask you that Sandeep, it is a very good presentation, but have you seen only you have collected from Jammu these wild fruits or any forest?
From the forest.
Only from Jammu.
You can call it a Himalayan region.
Yeah. Have you seen any variation in this morphologically or in fruit size or something? That what supplementing yours, that if you will be going for profiling, because we call it the accession, whatever the plants it is grown in your Jammu, that whatever the nutrient content it might carry, that might not be present here in the plants. That's why what sir is telling, that if you go for the nutrient profiling and any other enzymatic study or something, then it will be very, very valuable approach. And you can compare and you can go for extensive study like different access and collection like, then you can go for this profiling and any morphological variation is there. Thank you, Dr. Sandeep.
Thank you so much. I've taken your points.
Okay, thank you.
Thank you so much. Dr. Rubina Khanam, ICAR and NRRI Cuttack, strategies for mitigating arsenic toxicity in rice cultivation. Is she here? Okay, so we'll have a brief coffee break. Doing this coffee break early, so we'll start the session at 4. 3 more to go, ma'am. 3 more to go, but I have only received 1 PPT, so I don't know how many will happen. Yeah, that will be finished. Yeah. So we already have the next one. If you guys are okay, we can even call her now. Take a break. Ten minutes. Okay.
Bye. Can you please check it?
Registration. ଭାଇ ଯୋଉ ଶାସ୍ତ୍ରୀ ଆସିଛନ୍ତି ଶୈଲେନ୍ଦ୍ର ଶର୍ମା ଶୈଲେନ୍ଦ୍ର ଶର୍ମା ହଁ ଅଛନ୍ତି ଏଇଟା ତ କଣ ପାରାଲେଲ ସେକେଣ୍ଡ ୱାନ୍ ଟି ହେଇଗଲା ଜିନ୍ ଟୁ ହେଇଗଲାଣି ତ ଟୁ ପଟା ଏବେ କୋଉଟା ଚାଲିଛି ଦିଟା ଉପର ଦିଟା ନ ଚାଲିଛି
Okay, so we won't wait further. I'll request Dr. Tanuja S, ICAR-CIWA Bhubaneswar, to please join us on stage for the topic Strategic Climate Smart Approaches in Freshwater Aquaculture and Empirical Analysis.
Respected Chair, Co-Chair, all the esteemed delegates, very good afternoon to all of you. I am Dr. Tanuja S. I represent ICAR Central Institute for Women in Agriculture. Ours is a unique institute which we do research on gender issues in agriculture and allied sectors. And my presentation is a part of an ICRA funded project on gender inclusive homestead integrated aqua horticulture. So the topic is the strategic climate smart approaches in freshwater aquaculture and empirical analysis. So I'll just give a overview about the fisheries sector. So our sector, it ranks second to China in the world aquaculture fish production with an average annual growth rate of around 11%. And we contribute to around 7% of the global fish production. And day by day, the demand for fish is increasing because of the perceived health benefits from eating fish because of the lean meat. And it is rich in all the micronutrients, proteins, vitamins and all. So the demand is ever increasing. And that is why the growth rate is also increasing. So from the sustenance aquaculture, it has already been shifted to the commercial aquaculture. And aquaculture is playing the main role in Indian fisheries because marine fisheries has come to a stagnant point now because of overfishing in the oceans. So now the impact of climate change on fisheries and aquaculture, just like agriculture, it has created a lot of adverse impact on fisheries also, like change in the ocean currents, El Nino, sea level rise, and erratic rainfall, and thermal stratification, storm severity. These are all that is happening in the recent years, and it is affecting the production ecology, fishing, aquaculture, and post-harvest operations, communities and livelihoods, society, and economy. So this is mostly when we say about the climate change impacts, we study the marine fisheries or the capture fisheries, but it's equally affecting the inland fisheries also. So because of the intensification aquaculture practices, these water bodies are getting polluted, and this carbon dioxide emission, that is also increasing day by day from the aquaculture because of the intensification of practices. So climate smart aquaculture, it is a strategy developed to address the challenges and risk posed by climate changes on the food security and the development in a joint and collegial fashion. So the 3 pillars of CSA as brought about by FAO is that any of the practices that it should result in sustainable increase in production income, and it help in adapting and building resilience to the climate change, the risk and shock posed by the climate changes. And it may directly or indirectly reduce or remove the emission of greenhouse gases. So the need of the study was that we are focusing in this particular project, we are developing a model that is we have given the name as GRIHA in that. So that is gender responsive homestead aqua horticulture. That is an integrated aquaculture and horticulture. So gender responsive means it should have the participation of women. So we are focusing on these ponds, that is backyard ponds or homestead ponds, where women are having the access, because in commercial aquaculture, women are not having much access because they are very large fields, and mostly that is very intensive kind of practices that are being followed. So we are focusing on the homestead aquaculture practices where women can participate. And failure to target the climate smart intervention. So according to the local needs, there are many practices, climate smart practices, but we cannot give these interventions to the farmers and they will adopt. So according to the local needs, we have to target these interventions to support the 3 pillars. And this kind of failure will diminish the probability of meeting the sustainable development goals. So we have done an expert survey before like intervening in the farmers field with the practices. So the methodology followed was the freshwater, some of the freshwater aquaculture practices, around 40 numbers. It was selected based on the feasibility of the application in the farmers field, especially in the homestead area, and adoption, ease of adoption by the rural farmers. And online survey of the experts' opinion was collected on these climate smartness of these practices, and some statistical analysis was also done. So these are the 14 practices that we have selected. So I'll deal with it in the next slide. So first is the utilization of homestead pond. So homestead pond, it is the easily accessible resource to any rural household, and throughout the coastal India, this is a common resource in almost every household. So micronutrient rich animal protein source, they can get the if the women are also involving in that, and they involve in homestead aquaculture, they will have a continuous availability of rich animal protein source, and nutrition is also being affected because of the climate changes. So utilizing the family resources will also help them to adapt to the changes that is being posed by the climate shocks, and sustainable farm income and food source in the face of climate change, and minimize the greenhouse gas emissions by increasingly improving the nutrient flow in the system. And it is an easily accessible resource for the women. And according to the expert survey, it showed that it supports the pillar 1 and 2, that is sustainably increase the production, and it helps in adapting to the climate changes. Participation of women. So it has already been studied and proven that the participation of women in this homestead aquaculture, it will improve the decision making, because they take care of the family's nutrition. So their decision making power improves around the consumption and provision of nutritious food to self and family, and equitable power relations. These are all terms which are related to the women empowerment, like decision making and the power relations. These are all terms which are related to that women empowerment, improvement in women's access and control over the resource. Because if they're having the income, they will have a say in the family too. Increased awareness of women about their rights, and also women, they help the communities a lot in adapting to the changes or the risk that is being posed by the climate change. And farmer interest groups. Nowadays, a lot of thrust is being given to women FPOs. So this is being done with a purpose, because working in groups means they gain the confidence and they gain exposure, and this networking, it will help the women a lot, especially the rural women, because lack of exposure is one hindrance to their empowerment. So formation of women farmer interest groups will help a lot. So this has also found to support the 1 and 2 pillars of the climate smart aquaculture. And modified extensive farming. So that is like there are like three types of aquaculture. Like we can say modified extensive farming, then semi-intensive farming, and intensive farming. Intensive farming, like we say, we know that Andhra stands first in aquaculture production, because they usually they are following the intensive type of farming, which is totally dependent on artificial feed and a lot of energy in the form of aeration, fuel, everything is involved. But modified extensive farming means we are limiting the stocking density, we are resorting to natural food for the fishes. So these it is a high efficient food production system where low trophic species mean like the katla, rohu, and mrigal, that is the most popular fish in India. They are vegetarian fishes and they are very low trophic species. They do not need protein, high amount of protein in their diet. So they very efficiently convert the feed into the body mass. And because of the low stocking density and feeding level, we are resorting partly for the natural food and partly we are giving the artificial food. So the pond effluents will contain lesser nutrients and the water pollution will also be less. And it also has the added advantage that it is going as like organic fish. So in Odisha, it is that live fish, it has got much higher demand. So when we see that the fish is coming from Andhra, people have like less likeness for the fishes from Andhra. So that mindset is also there. And polyculture, like only carps is a annual fish, like we harvest it annually. But then there are small species, or species diversification is one of the most important practice that can be followed for climate, the resilience or adaptation to the climate related changes. So we can include that small midterm species which can be harvested within 3 to 4 months, so that the farmer he will not have to face the shocks that is being posed by the climate changes. For example, one cyclone comes, he will lose the whole crop, or even if a flood comes, he will lose the crop. So that he can go for species diversification. Community nursery. So in agriculture or in fisheries, the quality seed is the most important input for any farmer. And in every time farmer faces a problem. So normally the farmers resort to they get the seed from the local vendors who carry it in pots, and they bring to their villages. But there is a lot of mortality, the growth rate is not good. So the community nursery in the village itself, if they can have a community nursery, so anytime they can replenish their own pond from the community nursery. And also if they can stock a higher sized, like around 100 gram of fishes in their homestead ponds, they can get the marketable size fish within the 6 months period. So there is a like a shortening of the culture period. So that will also improve their income. And also because there is like a shift in rainfall or flood season. Nowadays it is said that we stock the fishes in July and August season, but the peak flood season is said to be September, especially in Odisha. So the thing is that in coastal or where there is a threat of inundation, the farmer will lose the stock. So in that case also, he can definitely he can take the seed from the community nursery. Otherwise, he will not have access to seed at that point of time. Late stocking, as I said, if we have a community nursery, he can go for a late stocking with advanced fingerlings towards after the post flood season, he can stock. And then within 6 months, he or she can harvest the fish. Farm-made aqua feeds. In aquaculture, the most important factor contributing to the greenhouse gas emission is the production, transportation of feeds. So farmers, they have access to locally available resources like the oil cakes and the rice bran, which are the two most important feed ingredient for fish. So if they themselves can make the aqua feeds in their farms, they can have a higher profit margin. Feeding management, like that is a lot of research has been done already, because we don't have to feed the fish every day. We can have alternate feeding or like a starvation period and then refeed it. So many studies have been done. These are things which the farmer, because uneaten feed will increase the pollution of water, and also it will increase the cost of aquaculture. So then feeding management is also one strategy or one practice that the farmers can follow. Multiple stocking, multiple harvesting. Now what is followed is that single stocking and multiple harvesting is being followed, or single stocking, single harvesting. Means in July or August, they will stock, and during the month of March, February or March, they will harvest the fish. So multiple harvesting means like that advanced fingerlings if you are stocking. So within 6 months, you can have the first harvest, and then next month, next harvest. So that he will not lose the stock entirely at a time. And also he will have a continuous supply of income and also nutrition to his family. Passive harvesting gears. The gear or the harvesting net used in aquaculture is dragnet. It is a very energy intensive process, and women cannot involve in that because there is a lot of drudgery involved. So passive gill nets, it has been already been studied that it causes the least greenhouse gas emission if we are using gill net, because we have to just set the net and the fishes will get trapped in the net. And use of solar dryers. Solar drying, we all know that there is a considerable amount of carbon dioxide emission reduction. It can be used because in rural areas, they use dried things like dried fish is there, dried vegetables, haldi, spices and all. So then if we can popularize these kind of like energy use based on solar energy, then we can reduce the greenhouse gas emission also. So I will conclude by saying that through expert opinion, because before we are going to like introduce any intervention to the farmers, we need to have a thorough study, and it will always be helpful if we have an expert opinion and evidence-based approach on the prospects of the climate smart aquaculture interventions that can be adopted by the farming community. And then afterwards, we can study how much they are adopting these practices. And this will help along to ensure their livelihood and will aid in achieving the sustainable development goals. Thank you.
Anyone up for any questions?
Arup. If there is no question, but madam, I have a question that in our childhood, that only we are not feeding the fishes, but it was becoming large. How it was being so at that time?
Actually, ma'am, we have also faced this similar kind of a thing. Farmers never dry their pond. So what we are getting, the big fishes, that will be around 2 to 3 years old, madam. So even now the farmers are saying that their fishes are not growing because carp, they have a low growth rate, and within 1 year, hardly it will reach a 1 kilogram size. So it is not possible beyond that, even beyond that. So maximum will get around 700, 800 grams. So what they will be harvesting may be some 2 to 3 year old fishes, madam, because they are not drying the ponds.
Okay. Thank you, madam, for making the woman independent and through this aquaculture. Thank you for your nice presentation.
Thank you so much.
Good evening, everybody. Now.
Thank you so much, ma'am. Well, the next session is again an online session. So we'll have Dr. Vidya Shri Bharti for that from ICAR-CIFA Mumbai.
Hello. Ma'am, next presentation will be by Dr. Shailendra Sharma or Dr. Vidya Shri Bharti.
Hello. Hello. Am I audible? Hello. Hello.
Dr. Vidya Shri Bharti, if you are there, please join us.
Madam, sorry for... myself, Professor Shailendra Sharma. Am I audible? Hello.
Okay. Okay. So, acha, we were not told about this, that Professor Shailendra Sharma is also having an online session. So, Professor Sharma, can we first hear it from Dr. Vidya Shri Bharti?
Ma'am, actually, I have
If she's there.
Is it possible for me to continue because I
Yes,
have
sir, sure.
emergency? My mother is very ill. That's why I have to cancel all the flights to Cuttack. I was very interested to.
Okay, sir. Okay, sir. Sure. Sure, sir. You can take your time. So we are having the session from Professor Shailendra Sharma now. He's from Chaudhary Charan Singh University, Meerut.
Thank you. Desktop one on. Open system. Which one? Click on. Okay. Just a moment, please.
Sir, can you please share your screen? It will show directly.
Yeah, I'm doing that. Just please wait. Yes.
Yes.
Yeah. Are they visible? Slides are visible? Yes.
You're audible, sir, and your slides are visible too.
Thank you, ma'am.
Yeah.
So Thanks for organizers for the Agri Vision conference. Unfortunately, I could not attend this program in physical mode, as I told that my mother is very ill, and I am also suffering from severe cold. Anyway, so once again, thanks for the organizers for giving me this opportunity and accepting my request to deliver this lecture online now. So my title is Dissecting Genetic Resistance of Wheat Towards Plant Parasitic Nematodes. Yeah. So before starting, I would like to give brief introduction about these plant parasitic nematodes. So these are tiny, complex, unsegmented roundworms and are anatomically differentiated for feeding, locomotion, digestion, etc. These species are transparent, L-shaped. So plant parasitic nematodes are very economically important. They have a stylated mouth, they puncture cells with the help of this stylet, and more than 2,000 species of plant parasitic nematodes are known. So they are basically known as the hidden enemies of the farmers, because the symptoms caused due to this damage caused by these plant parasitic nematodes are even confused with the nutritional deficiency in the plants. So the most common damaging PPNs are the root knot nematodes, cyst nematodes, and root lesion nematodes. So basically, I am going to talk about two nematodes on which in my lab we are working on two nematodes, and basically we had a DBT funded project, a major research project. One nematode is Heterodera avena, another is Petilantius thorni. One calls the well-known Molia disease of a small cereal grain species like wheat and barley. Other cause root necrosis. So when we talk about this Heterodera avena, it causes a serious economic problem known as Molia disease of wheat. It can cause yield losses up to 4.3 to 100% in different countries, from Pakistan, India, Morocco, Saudi Arabia, and especially in India, when you talk about the Western Haryana, Rajasthan especially. So we have this severe problem of this Heterodera avena. And there are no significant genetic studies on Heterodera avena. So when we talk about the management, we can go for crop rotation, sanitation, tillage, biofumigation, nematicides. But the most eco-friendly and sustainable management is the development of the resistance varieties. So I'm going to talk about the genetic research that we conducted on these nematodes. So in my lower portion, you can see these are patches. So these are the empty patches in the wheat field. So these are the due to nematode infestations of the roots. Video.
Plans to develop.
I just want to show one video.
Already at this early stage, the root is well developed. In many soils, dark brown, thick-walled cysts lie hidden. Many of them have lain dormant here for years. These cysts often contain hundreds of eggs. Each egg contains an infectious juvenile of the nematode Heterodera shaktii. Substances released by the roots of the young sugar beet plants stimulate hatching of the juvenile nematodes. The nematodes break through their eggshells and then emerge from the cysts. As soon as an infectious juvenile has reached a sugar beet root, it uses its stylet to penetrate into one of the cells of the outer root layer. The roots of sugar beets are suitable hosts for this nematode species. Using the strength of its muscles, the nematode migrates through the root tissue. Supported by numerous cell wall digesting enzymes that are secreted through the nematode stylet, the juvenile advances from one cell to the next, though without feeding.
I think we don't need to show this. So I just gave I gave the video presentation for the Heterodera shaktii. For the Heterodera avena is also little more same. So see, these are the pictures from our lab experiments. So it's showing life cycle of Heterodera avena. So on the right hand side, you can show the number of cysts. And from this cyst, as shown in the video, after hatches, juveniles come out, and these juveniles move into the soil, and then they through the stylet, they infect the roots, they spread in the roots, and they form syncytium in the third stage juvenile, and later on the females become dead, and later it form, there's some tannization, and there's a formation of the brown cyst on the roots. So this is the whole protocol that we follow in our lab. This is not a very expensive experiment. It shows how we collect the cyst. So we have collaboration with the CCS University, Hisar Agriculture University. We collect the infested soil from that region, and then we collect the cyst from the infested soil. And this is a protocol that shows how we we infect the plants, and later on we collect the cyst and we count the cysts. So all the experiments were performed in the controlled conditions. So we have controlled humid conditions, humidity, temperature, etc. So around 60-70 days after sowing, we can find these type of white cyst. So when they mature, they become brown, and inside these we can find a lot of eggs containing juveniles. The first experiment was published in Molecular Genetics and Genomics Journal. So it was based on Jiva's genome-wide association study. The title was Genome-wide Association Scan of Serial Cyst Nematode Resistance in Indian Wheat Germplasm. So we used 143 wheat genotypes collected from different pen regions of India. We obtained this material from National Bureau of Plant Genetic Resources, New Delhi, NVPGR. So basically it's showing, so as I told, it's a genetic study. So it's showing SNP. So we used SNP molecular markers for this Jiva's study. So basically it's a circular Manhattan plot. It's showing the highly significant MTAs. So MTAs are basically regions, we can say that they are the potential regions or potential genic regions, which can tell about the potential candidate genes involved in the resistance against these nematodes. On the right hand side, you can find the chromosomal location of these MTAs. So this study was published in Molecular Genetics and Genomics. The another study was published in the Scientific Reports by Chaturvedi et al. This is the identification of genomic regions associated with the serial cyst nematode resistance in spring and winter wheat. So we have this winter wheat and spring wheat panel from Institute of Plant Genetic Research, IPK Cuttack lab in Germany. Again, this is the Manhattan plot, and these are the potential MTAs. These are the chromosomal locations of these MTAs. And the good thing is that we confirm that these MTAs are real from the previously conducted studies. So some of the MTAs that we found in our study were already co-localized with the already discovered major genes or QTLs. So now we are focusing on these regions. So this is the chromosomal map showing MTAs detected in the present study. This is the expression profile of the candidate genes. So after doing this mapping, we concentrate on the finding of the potential candidate genes in order to find the potential genes. So we have different genes like WD40, F-box gene, NVCLRR proteins, etc. This is also another interesting study conducted by Pundir et al., also published in the Scientific Reports. It's based on the QTL mapping for resistance against serial cyst nematodes in wheat. So I would like to go to. So as you show, this is a major QTL region that we found, and this QTL. The good thing is that this QTL was already reported few years back. And this shows that our experiments were accurate and precise, as we also reached on the same location that was reported earlier. Basically, we use synthetic opata mapping population, DH lines, double diploid lines for this experiment. And now we are focusing to narrow down this region. So this chromosome was this chromosome was located on the 2D region. And we are now interested in to narrow down this region. So we made crosses between these DH lines. This is another study also conducted by Pundir et al. Published in Genetic Resources and Crop Evolution, Grace. So these pink threads are the stained nematodes. So in order to confirm that whether nematodes, there is infection in the root is successful or not, what we do, we stain the infected roots. So after this staining shows different stages. For example, this one is the resistant line M6, and this is the susceptible line Opata. So in the resistance line at 1 dpi, the number of nematodes is almost same like in the susceptible line. But as the time goes, so around 8 dpi, we can found that in the M6 resistant line, you can find only the juveniles, like J2 stage. But in the Opata susceptible line, there is almost formation of the syncytium. So you can find even J4 stages. So we can see that in the resistant lines, there is a slowing of the progression towards the maturity of the nematodes. Again, we also performed a meta-QTL study based on the previous reported QTLs. And we found an interesting reason on the 2A chromosome. Now, currently, we are also performing some transcriptomic analysis of the infected root samples. Again, these are the infected roots. So we perform these experiments in replications. So this work is going on, not published yet. So in the previous slides, we used M6 and Opata that we used in our QTL study. But in the another study based on transcriptomic analysis, we used Raj-MR1. So Raj-MR1 is the only release variety from India which is registered had to have an infection. So we are using this variety for this transcriptomic analysis. So the another nematode that we are working on the Praetelenchus thorni nematode. There are not so many genetic is available, especially in wheat. It can cause up to 62 dies, right? Yugoslavia, Morocco. Again, the stories of development of the resistance varieties. So in the previous nematode heterodyrivina, there is a formation of the cyst, but in this case, there is no formation of cyst. So it's a, you can say it's a moving nematodes, migratory nematodes. So in this cartoon shows the penetration of the root by the nematodes. Again, there's a formation of juveniles in the egg development, and first molting J2Hs from the egg, then to second molting, third molting J3, J4, and there's a differentiation of the adults, female and male, and so on. So this infection, the difficulty with this nematode is how we multiply. So initially we have this collaboration with the ICAR Kanpur, and we faced a lot of problem in multiplying these nematodes. We used carrot disc to multiply the nematodes, but now we are successful. So it takes about 3-4 months to get these nematodes and perform the infection. So this is a carrot callus, and this callus contains lots of nematodes. And again, the process is same, that we collect the nematodes, and in the controlled conditions, we provide the nematodes to the wheat root plants. And then finally, we collect the nematodes. In previous case, we collected the cyst, but in this case, we directly collect the nematodes, and then we count the nematodes, and then we try to associate with the already available genetic markers. In our case, it's SNP, single nucleotide polymorphism, genotyping data. So it just shows that it is a tissue paper. It contains a lot of nematodes, which are visible only in the microscope. You can see it's a lot of hundreds of nematodes are there. So this study was also published in the scientific reports. This is a circular Manhattan plot. And again, we find that some interesting regions that we are currently working on. Again, in the same case, also we are performing some transcriptomic analysis for this nematode in the resistance and susceptible genotypes. It's working. This work is going on. And the other thing that now we also developed in the last 5 years, we also developed some mapping population. So what we did in the first case, we used this only released variety from India, Rajamar1, and we crossed with the susceptible variety WH147. Now it's in F6 stage. And the other one is the WH147. It's a reverse. And the other parent is Rajamar1. So it's a reciprocal cross. So we performed this experiment at Dalang Maidan, Himachal Pradesh, Wellington. It's spelling, sorry for the wrong spelling. And also at, of course, at Perat. Another cross. So these are the publications from my lab in recent 4 years. So 3 publications from scientific reports. One is in molecular genetics and genomics. Another is in BMC plant biology. Another is in genetic resource and crop evolution. Now we are working on the development of the CAS markers. We want to validate the MTAs. We want to develop some user-friendly markers. So now our next goal is to finalize our transcriptomic study and the development of the diagnostic CAS markers. So this is our group. My mentors, Professor P.K. Gupta, Professor Balian, myself, my better half, Dr. Shweta Sharma. These are my students, Deepti Chaturvedi, Deepak, Saksham, Vikas Kumar Singh. And we have collaboration from the Scotland Rural College, Dr. Rajiv Sharma, United Kingdom. And I have collaboration with the IPK Garters Lab in Germany, Dr. Marian Roder, Andreas Borner. And from NBVJ New Delhi, I'm thankful to Dr. Sandeep Kumar. And that is from CCSU Haryana. I have a very good collaboration with the late Dr. Bansa Singh. Dr. Kavar, RS Kavar, sorry. And from the IIPR Kanpur, we have Dr. Bansa Singh. So I am thankful to all collaborators and my students and my mentors for making this research successful. And I think very few groups are working in India on these plant parasitic nematodes, especially towards genetic studies. So once again, thanks a lot. Thanks for your patience and listening to me. My voice is cracking due to severe cold. And once again, thanks for the organizers who gave me this opportunity to attend this conference.
Thank you so much, sir. Ma'am, would you like to ask any question?
I think it's not so cold there, Cuttack.
Yeah, okay.
It's a very severe cold here.
Please take care of yourself.
Thank
sir.
you, Dr. Sharma, for your extensive study in
Yeah.
nematode resistance.
Welcome.
And that my question is that you have gone for that pedigree method for this irrigator somewhere I have seen. And then what is the purpose of it? You want to develop the reel or something?
Yeah, of course, ma'am. So basically, we are working for the QTL. We have to perform some QTL studies. Later on, we want to narrow down the QTL region. So actually, the thing is that besides this nematode study, we have some other traits also in our mind. So we try to select the most contrasting parents, not only for the nematode resistance, but the parents which are showing contrasting traits for other traits. So we selected these parents, especially Rajamar1, another contrasting parents. Yes, mainly for the QTL region. And in the case of in this DH population, we have already made crosses between the DH population to narrow down the region.
Then have you developed some marker for this thing? You have not your future studies to go for fine mapping?
Yeah. That's why I told that we are now focusing, we are trying to establish this caste system in our lab.
Yeah. Then otherwise you can introduce this QTL and you can validate this resistance.
Yes, of course. The ultimate goal is that.
Yeah.
Some high yielding varieties, our target is that.
Okay, thank you, Dr. Sharma.
Welcome.
Thank you.
Thank you. Yeah.
Thank you, Professor Sharma. Now we'll move towards the next one. Please take care of yourself.
Thank you, ma'am.
We have the talk by Dr. Vidyashri Bharti, ICAR, CIFA. And right after this, we'll move for the inauguration. Ma'am, please continue.
Hello, am I audible?
Yes, ma'am, you are audible.
Okay, so thank you very much for giving me this opportunity for the online presentation. So respected dignitaries present there, dear audience, I'm Vidyashri Bharti from CIFE Mumbai, working as a senior scientist there. So let me full screen first. Okay. Okay, so my title of my presentation is the work area which I'm working is the potassium enriched biochar low cost green technology I'm saying here to enhance the production of Litopinus venomai, which is reared in the inland saline water. So we understand that next slide. So we know that in Indian aquaculture, the global fish production is 178.5 metric ton. And out of that, say aquaculture production is separate. And then among Indian condition, we are more focusing on this P. venomai culture because according to MPEDA, we are top shrimp exporter. And we are getting a lot of revenue from this exporting the shrimp. So under that scenario, and we know that shrimp is providing a very good quality food or the protein-rich food to the people. So fulfilling the SDG 17 goals, this global food security is one of the major concern, and we understand this low quality, low cost protein shrimp or the protein diet. No, it will help in mitigating the hunger and providing the good quality food. So we are having the resources, but as we understand, the land is shrinking and we cannot go for the vertical expansion. So in that case, in that scenario, whatever the available resources is there, we have to utilize this. So one of the resources is the inland saline area. So according to Singh et al., globally we are having 953 million hectares and there is no option left. We have to explore this inland saline resources for the production system, for the sustainable production. And in Indian context, it is expected that this salinity or the salinized area will increase up to 16.2 million hectares. So there is necessity to develop the technology which can help in mitigation of this the problem associated with the inland saline system, as well as to pave the way for the sustainable production by adoption of the technology. So one of the technology is the aquaculture in the inland saline water. We understand in the bucket water system, P. venomai culture, fantastic. But coming to the inland saline system, where some limitations are there. So we have to overcome the limitation by intervention through the technology. And one of the technology is the biochar application, which was applied in case of agriculture plot during this NECRA project, which is still running. But most of the success is not achieved. When we see that this biochar is having much more potential for the remediation of the different problems associated with the agriculture as well as the aquaculture. So this inland saline aquaculture ponds are experiencing few problems I'm just mentioning here. So seepage issue is there. Then nutritional deficiency is there in terms of particularly this water is having very low potassium, high calcium, and then variable magnesium content. So these are the few limitations I just indicated here. So this potassium and magnesium ion, which is required for the molting and as well as the immunity built up in the shrimp. So which is very much crucial for this shrimp culture. And then in that scenario, if I'm enriching this biochar with the potassium, it will be very much helpful in supplying the potassium, which is the limiting nutrient in that condition. So keeping in that view and the problems associated with the inland saline system, so by application of biochar, which is enriched with the potassium, we are taking care of the limitation, one of the limitation, and then at the same time, we are utilizing the resources, which is having a lot of problem. So by what we see that from the literature that this L. venomai culture in the saline water, because of this deficiency of this the potassium as well as the magnesium, the productivity is very less. This ionic imbalance will adversely affect the physiology of the species cultured there. And then to maximize the growth and survival of the shrimp, particularly people are going for the amendment or the I would say the application of KCl as well as the magnesium chloride. So the major problem of this nutrient imbalance, we can say, the potassium and the magnesium, as well as I mentioned, the seepage loss. So the biochar, we find that biochar is very much helpful in improving the water holding capacity of the sediment, as well as mitigation of this magnesium, as well as the potassium issues, because we know this paddy straw biochar and other biochar is very much rich in the potassium ion, and at the same time, the biochar is very much known, or we can say, because the properties are similar to the charcoal. So biochar is very much known for the removal of the ammonia absorption. So this keeping these benefits of this biochar application, and as well as the amendment with the potassium, we attempted to apply the biochar which is rich in the potassium, as well as by enhancing the potassium content in the biochar by the engineering part, and then we applied in the aquaculture pond in the inland saline scenario. So what we find that this is the biochar we are producing. We can see the picture here, and this biochar, if you see at the molecular level, so biochar is having this is the scanning electron microscopy of this sugarcane bagasse biochar. So at the molecular level, the biochar is having the very high surface area. So this surface area will help in the removal of the ammonia, as well as this site provide the better exchange of the nutrient which is available in the sediment in the water. So coming to the methodology part, what we actually had done, we had taken the agro waste available, the sugarcane bagasse as well as the paddy straw, which is abundant in the northwestern part of the country, and it is leading, because it's a burning issue, it is causing the air pollution also. So keeping this thing in mind, locally available agro waste, we are converting into the biochar, then adding the potassium, the salt, the KOH, and then activating it, making it as the potassium-rich biochar, and then applying in the aquaculture pond, and then cultivation of this shrimp. So what we find that, and then this activation, we attempted earlier 0.5%, 1%, 1.5%, and then 2%. So what we find that 2% application or 2% enrichment of biochar with the potassium is very much helpful in amelioration of the deficiency of this potassium. So these are the few parameters. We stock this PL10, which is procured from this Andhra Pradesh, and then before stocking in the main pond, 30 days it was done in the rearing pond. And as we know, in the inland saline water, the salinity is also the very good condition. So we are using the salinity for the culture of the shrimp. Otherwise, it is not feasible for the inland saline system. And then feeding is done. So all the parameters which is practiced in the coastal aquaculture or the brackish water aquaculture, we are implementing in the inland saline system also. And then most of the measurement or the analysis was done with the standard methods. So we had done this sediment parameter, the water quality, as well as the fish health parameter, particularly these enzymes, we can say, and the growth parameters. So coming to this physical and the chemical characteristic, so biochar, everybody understand, as compared to the agro waste material, biochar is having a very high surface area, water holding capacity will be very excellent. Then the porosity will be higher as compared to the normal agro waste. And then at the same time, the issue, this water holding capacity, as I mentioned, it is very much higher, though it is activated or non